So, as you know, one of the passions of WACCBIP is connecting with African scientists who are in the diaspora. And if you read the stories in our profile, you realize that we have actually managed to bring a good number of African scientists who were in the diaspora, and we facilitated their return home to work at WACCBIP or other institutions. In keeping with that, we continuously engage with our friends and colleagues who are in the diaspora. And whilst it is always desirable that we bring them home, some of them are very useful to us being where they are and where they are situated and the capacity they have where they are to be able to support us in different ways, instead of being here. So we normally say this is what brain circulation. So it's not brain drain, it's brain circulation. So the brains go there and they circulate knowledge and resources back to us and vice versa. So today we are very pleased to welcome a Ghanaian scientist who has distinguished himself in the world out there, specifically in the US, and specifically in the field of drug discovery. He has, made a big name for himself in leading innovative drug discovery work, first in the University of Maryland, and now in Purdue, and probably going somewhere else. So, ladies and gentlemen, I'm pleased to introduce Professor Herman Sintim. He obtained his BSc from the University College London in medical chemistry. He did his DPhil in organic chemistry at the University of Oxford. After that, he did postdocs at University of Oxford and also Stanford. Then he went to University of Maryland in 2006 as an assistant lecturer. He got his tenure in 2012 and became associate professor. In 2015, he was promoted to the rank of professor. Same year, he moved to greener pastures when he was awarded an endowed professorship in drug discovery at Purdue University. So he relocated his group there. Whilst there, he has co-founded Kinarix, which is a private company. He served as program director at the National Science Foundation for their chemistry section. In 2022, he became the Richard B. Wetherell Professor of Chemistry and Drug Discovery. And in 2023, last year, he became a distinguished professor of chemistry. He has won numerous awards, including the National Science Foundation Career Award, Camille Dreyfus Teacher Scholar Award, Sigma Psi Distinguished Lecturer. He has also served as a permanent member of the NIH Study Section on SBCA, which is a great privilege to be on study section as a permanent member. He currently serves on the editorial board of Bioorganic Chemistry and is on the editorial advisory boards of the American Chemical Society journals, which are the ACS journals, including Medical Chemistry Letters and ACS Infectious Diseases. He has attracted over 20 million in external funding from diverse sources, including the National Science Foundation and the National Institutes of Health. Ladies and gentlemen, let's welcome my friend Herman.
Thanks, Gordon, for such, I would say, a very flowery introduction. It's always a pleasure to come back here. I did my A-levels at Presbyterian Secondary School just around the corner, so I used to come to campus a lot to enjoy the campus and also to study here. It's always a pleasure to come home and to meet my friends. I'm gonna set my timer so that I don't go over. Okay. So I was trained as a medicinal chemist, and the goal of a medicinal chemist is to take in all the insights in biology and medicine and to come up with new therapeutics that could help extend life, as well as solve some of the serious diseases that there are no drugs available. And so COVID has elevated medicinal chemists to the forefront. Now the general public know what we do. So I always make the joke that without medicinal chemists, COVID will still be rampaging through the society. It's a really bad joke. But the point here is that, doctors and pharmacists and the health professionals will not be able to cure diseases without medicinal chemists developing new and innovative therapeutics. And so my group, we are interested in two general areas. One is infectious diseases, and then the other one is cancer. Okay, so we are motivated by what I call perplexing issues in medicine. And so infectious disease is something that touches most of us. It turns out that although there are many antibiotics available, there is a rise of antibiotic-resistant bacteria. So these are bacteria that are resistant to the majority of available antibiotics, okay? And so this is almost like a ticking bomb. Here is an example of a patient who had infections from a drug-resistant bacteria, and so the available drugs didn't work. And so we can see that progressively, from day 1 to day 9, the infections and the sores were moving all over the body. And then unfortunately, by day 34, the patient passed away from sepsis, okay? So this is where there is uncontrolled inflammation, and you end up having organ failures. And so despite the availability of antibiotics, drug-resistant bacteria is still one of the very, very key and major challenge in medicine, okay? The second perplexing problem in medicine is the issue of tuberculosis. It really boggles my mind that TB has been with us forever, yet we do not have very good drugs that actually tackle drug-resistant TB. Oops. Looks like we're having a problem. Okay.
So what's up? Come again? Oh.
Okay.
Sorry. So they are rebooting the machine?
They are rebooting the machine. Okay. Okay. Well, so I will probably still talk about the TB until it comes up. Um, I remember when I was a kid, I was told that there was someone who had TB and he used to cough, and we were all advised not to get near that person because we all know that when someone has TB and they cough, it is very easy for one to also get that TB as well. Um, So, the machines are not taking over, right? We always get scared that, the machines will take over and artificial intelligence will, but we forget one thing. If you plug, if you plug the stuff of the machines will not have any soul anymore. So I think that we've still got some juice to go, okay? Is this where I bring up the whiteboard and I do a chalk talk? Yeah.
While they're rebooting, maybe you'll talk about this. I'll just break the silence.
Yeah.
Drug-resistant TB.
Uh-huh. Yeah.
Some progress been made in pediatrics. I'm just curious about waiting on the slides.
You mean in pediatrics, you said? Yeah, so you actually bring a very good point. Pediatric drug discovery is one of the most difficult areas because before you test drugs on children, the safety level has to be like way better than you test it on adults, right? And so the advances in pediatrics it's not better than in adults, but the TB Foundation has now come together to try and come up with new modalities, to try and come up with new drugs against TB. The problem with TB is that the funding to do TB drug research is just simply not there, right? Most often, it costs more than $500 million to take one single drug to the market, right? The clinical trials are very, very expensive. And so drug makers are always looking at the profit margin. And TB is more prevalent in the parts of the world where profit margin is not the largest, right? And so you have the TB Foundation, the Bill Gates Foundation, and some benevolent foundations that have taken on the TB issue. And so one would have argued that if the economics of TB was as lucrative, which is not a very nice word to use, right, when you are talking about drugs, but if the economics was as lucrative as, let's say, cancer, maybe probably we would have made more advances in trying to cure TB. But yes, drug-resistant TB is still a big problem as well.
Thank you,
Yeah.
sir. I think we're back.
Okay, so we're back. Okay, so you know the statistics with TB is really outstanding, right? It actually, you know, 25% of the global population actually harbor some form of TB, right? And in 2022 alone, more than 1 million people actually died from TB. So this was towards the tail end of COVID. And so TB was the number 2 killer, but TB has actually gone to now become number 1 killer now that COVID is not killing that many people. Okay. So it is really sad that for such a devastating disease that still claims a lot of people, the efforts to actually come out with new drugs against TB has not garnered pace as one would really like it to be. The next or the third area that my group focuses on is cancer. And it turns out that 1 in 3 of us, if we live to our 70s, will get some form of cancer. 1 in 7 women would get breast cancer. Okay, so these are really stark statistics. It also turns out that every year there are about 20 million people who are diagnosed with cancer, right? So the incidence for cancer is about 20 million per year. And it also turns out that every year across all cancers, the mortality, the number of people who die from cancer-related problems is almost about 10 million. So when you really think about it, 20 million people are diagnosed with cancer each year and about 10 million die, right? So we are looking about a 50% chance across all cancers. But there are some cancers which are even worse. So for example, if one gets cancer of the brain, then the prognosis is really terrible, okay? More than 90% of people who get cancer of the brain are going to die. More than 95% of people who get pancreatic cancer will die within about 2 years, okay? There are some cancers such as cancer of the thyroid gland that is now almost curable. Over 90% of patients who get thyroid cancer can be cured. And then also, it turns out that because of screening and other advances, breast cancer has also seen a very good therapeutic option. So over 90% of breast cancer patients can also now survive, okay? But cancer is still one of the big areas. And so the big ticket cancers are lung cancer, breast cancer, and liver cancer, ends up killing a lot of people per year. So these are what I would call the big areas in medicine that despite the advances that have been made, we are still very far from actually dealing with these. So I started my group in 2006. So in 2 years, it will be like 20 years since I started my group. And when I started my group, I asked myself, which area did I want to move into that maybe a few decades later, some of the advances that we make and also people in the field would actually make a big dent in our efforts to deal with drug-resistant bacteria as well as cancer. And so I was trained as a chemist, and so I always look at things from the molecular level. And so the question was, which molecule is going to be the star molecule of the future, or which signaling axis did we want to tackle? And so I made a bet. And the bet that I made almost 20 years ago was that the molecule that you see on the screen, which is called a cyclic dinucleotide, was going to be one of the star molecules 20 years later. It was an interesting bet because when I started my career, I had to convince a a lot of academic departments to give me a job, right? And so cyclic dinucleotides were not actually very popular. And so trying to convince an academic department to give a job when that area was still young was a little bit difficult, okay? So it is almost with pride that I look back 20 years later and realize that that bet actually paid off. And so why do we call these cyclic dinucleotides? You can see here that there is almost like a cyclic structure. So that is where the name cyclic comes from. And then that is called a nucleotide. So you call that nucleotide 1. And the second is also called a nucleotide. And so when you have two nucleotides, you have a dinucleotide, right? So that is where the name comes from, cyclic dinucleotides. And so one of the greatest physicists of all time, called Richard Feynman, once said that what I cannot create, I do not understand. So this very interesting molecule had been described, but as a chemist, I felt that for us to really understand this molecule and understand the signaling, we had to come up with a way to create it, right? And so it turns out that my first paper as an independent scientist was the synthesis of this molecule. It was actually part of the Emerging Investigator issue of Molecular Biosystems. And it was a very good bet, because when we were able to synthesize this molecule, going by what Richard Feynman said, we would then be able to understand this molecule and apply it to so many areas that I'm going to go through. That allowed us to get our first NSF Career Award. So the take-home message or the advice that I will give to young people in this audience is that you have to be bold, you have to make a bet, and when that bet pays off, the payoff is actually going to be quite huge. And so we got a 5-year grant, just over $750,000 to study this molecule. And so that started our journey. And then in 2014, in collaboration with Claudia Gravekamp at Albert Einstein School of Medicine, we were actually one of the very early groups in the world to demonstrate that this bacteria signaling molecule is actually a cancer. You can use this for cancer immunotherapy. So there is a very prominent paper that we published in 2014 that has actually now gained a lot of citations. And then in 2023, as a chemist, we were able to develop a very potent cyclic dinucleotide-based immunotherapy, and I'm gonna go through these. But just wanted to highlight how we made this bet, and then looking back, we've seen how this bet has really paid off. So in science, we all stand on the shoulders of giants. And one of my heroes and one of the biggest giants in science is this guy called L. Sutherland. So in the late 1960s, L was the first person who revealed to us the structure of a molecule called cyclic AMP. So for those of you who do biochemistry, you are aware of cyclic AMP. It's one of the most important signaling molecules. We call them second messengers. And so here, this molecule, you can also see that it's cyclic. You see the adenine, and so it's a cyclic adenine monophosphate. And so for the discovery of this molecule, which has only got one nucleotide, and the demonstration that this molecule is important in biological signaling, Sutherland was awarded the Nobel Prize in Physiology and Medicine in 1971. And it turns out that the pathways that are involved in this molecule has now led to the development of many drugs that are used in cardiovascular diseases, erectile dysfunction, okay? And so the Nobel Committee were actually very, very insightful in giving him this Nobel Prize. Now, there has always been the joke, what is the quickest way to get a Nobel Prize? Is there anyone in the audience who knows that answer? I mean, of course, you can always come out with some seminal discovery that would impact medicine or science. That would become very obvious. But it turns out that the quickest way to get a Nobel Prize is actually to work for a Nobel laureate. Okay? So, it turns out that the student of L. Sutherland also got a Nobel Prize, right? And that is Farid Murad. He got a Nobel Prize also for a molecule called cyclic GMP. So it's very, very similar to cyclic AMP, right? Okay. And he was given this Nobel because it turns out that cyclic GMP is really critical for a lot of the vasodilations and so forth. There has been numerous other people who have been given a Nobel Prize in physiology and medicine for identifying the proteins that are involved in responding to these signaling molecules. I would actually make a very bold statement that you will not find any field that has resulted in as many Nobel Prize winners as cyclic mononucleotides. Okay? So, it turns out that in the late 80s, a gentleman called Moshe Benzeman in Israel discovered another molecule. Now, this molecule, which he published in a Nature paper, is a cyclic dinucleotide, right? So I have mentioned that cyclic mononucleotide has led to so many Nobel Prizes. It's really led to advances in medicine, right? And when Moshe Benzeman published his paper in the late 80s, he showed that this molecule was important in a bacteria that made cellulose. So that bacteria was relevant for agriculture. Almost no one paid attention to this paper. So for more than a decade, Moshe's lab was the only lab in the world working on cyclic dinucleotide. So when I was applying for an academic job in 2005, I was looking for areas that were new that I could make my bet. And as a chemist, I had an appreciation that cyclic dinucleotide is di, 2 is better than 1, right? So if 1 had led to all these great discoveries, then 2 was going to lead to at least twice those discoveries, right? Now, I would even argue that it was going to lead to not twice, but a square of the discoveries, or maybe the multiples. So it turns out that cyclic GMP is made up from 2 molecules of GTP that condenses to form this linear molecule, and the linear molecule cyclizes. So you have this molecule, and we call it a signaling second messenger. It then binds to a cellulose synthase, and it regulates the enzymatic activity. But in biology, everything has to be terminated by an enzyme, right? So the enzyme that terminates the action of cyclic dinucleotide is called a phosphodiesterase. So the phosphodiesterase would break one of these phosphate bonds to the linear form. So we call it phosphodiesterase A. Looks like this is very, very happy, trigger happy. So we call that phosphodiesterase A, PDEA, and then a second phosphodiesterase breaks it down to the mono, right? And then the action is terminated. So this is a very regular feature of biological processes. Now, in Benzeman's paper, he postulated that there should be the existence of this phosphodiesterase, but they didn't characterize it. Okay, so this was in the 80s. Now, I always say that life, sometimes you just have to be lucky. So when I started my career at the University of Maryland, a very good friend of mine, who was also a colleague called Vincent Lee, also has started his career, and Vincent Lee is a microbiologist. He had done his postdoc at Harvard, and he was also interested in cyclic dinucleotides. So what are the odds that we would both meet at Maryland? And so we formed a team, and in 2015, several years after Benzeman's prediction, we actually showed in this PNAS paper that an enzyme called oligoribonuclease, which was formerly characterized as an enzyme that cleaves short RNA, was actually the elusive cyclic dinucleotide PDE. Okay, so this actually got some press. I'm going to show you that cyclic dinucleotide is now the real deal. Unfortunately, Benzeman actually passed away in 2004. If Benzeman had not passed away, I would have made a prediction that he was going to be awarded a certain prize, name not to be mentioned. And if I was to nominate, I would have given the citation for the discovery of cyclic dinucleotides, ubiquitous signaling molecules in all forms of life, which spared the development of novel cancer immunotherapies. Okay, unfortunately, Benzeman passed away before the beauty of these molecules actually became apparent. Okay, so fast forward, we now have different forms of cyclic dinucleotides. So these were the mononucleotides, and these are now the dinucleotides, right? So we now have so many different forms of cyclic dinucleotides. And the cyclic dinucleotides, this is a very busy slide. The cyclic dinucleotides, some of them are bacteria-derived molecules, right? And bacteria-derived molecules are interesting molecules because the immune system has evolved to recognize bacteria-derived molecules as danger signals. That's the reason why you have sepsis, right? When you get an infection and you get a high fever, it's not always a bacteria that is actually directly giving you the high fever, but it's the immune system trying to counteract the bacteria that leads to the high fever. And so there are all these receptors on the cell that actually sends different components of the bacteria. It could be the LPS, it could be the DNA from the bacteria, it could be the RNA from the bacteria, but cyclic dinucleotides are also one of these pathogen-associated molecular patterns that the immune system recognizes. And an interesting protein, which I would expand a little bit, is called STING. It sits here in the middle, so you can see that they feed into this interesting protein, and then that leads to a lot of interesting signaling pathways. Okay, so now let's expand that, right, and see what happens. And so it turns out that when you get infected with bacteria, viruses, once they get into the cell, the DNA of the bacteria can be exposed, or during aging, when mitochondrial DNA gets introduced into the cytosol, the cell basically recognizes the cytosolic DNA as a danger signal, right? After all, DNA is supposed to be in the nucleus, okay? And so what then happens is that this DNA binds to an enzyme called cGAS, and once cGAS is bound to the DNA, it becomes activated. It takes ATP and GTP. It cyclizes it to form something called a cyclic GAMP. That is the human second messenger. The bacteria, in addition to releasing the DNA, can also release cyclic dinucleotides. Remember that Benzeman discovered cyclic dinucleotides in bacteria. So both the human form of cyclic dinucleotide and the bacterial form of cyclic dinucleotide can now bind to this very interesting protein called STING. Once STING binds to these second messengers, it leads to downstream cascades that activates the synthesis of cytokines, okay? And so it can lead to inflammation, and inflammation can be a bad thing, right? But inflammation can also be a good thing because inflammation can clear cancer, okay? And so let me just update you again. This is cGAMP, where there is a linkage between the phosphate and a 2' position and a phosphate and a 3' position. So we call this a 2', 3' cGAMP. It's a mammalian form. The bacterial form is a 3' linkage. Okay, so we have the bacterial pathogen-associated molecular pattern and the mammalian pathogen-associated molecular pattern. They can all bind to sting. Okay. So we like to ask questions because it turns out that sometimes you get very interesting insights if you actually ask a question. So I started this talk by talking about TB. Okay. Now, TB is an awful pathogen. It kills so many people. But TB is also a very interesting and sophisticated pathogen, and I'll tell you why. You know, TB actually infects macrophages, right? Macrophages are your immune component. So to me, it's almost like a thief coming into a city and going into the army barracks. I mean, these are the soldiers, right? It actually goes into the army barracks and it hides there, okay? And so we became really interested with this key question: how do intracellular pathogens evade the inflammatory response via sting, right? Okay? And it turns out that when you have bacteria that gets into cells, they release their cyclic dinucleotides, it binds to sting, and that should lead to immune response. And so if TB is able to infect macrophages, then it means that TB has found a way to switch off macrophages, right? So that was the understanding. Well, that was a prediction. And so what I did is that I combined with a guy called William Bishaya at Johns Hopkins University. So William Bishaya is one of the world's preeminent scientists in TB biology. So we were working on cyclic dinucleotide, and we worked with William, and we disclosed in 2017 that TB actually has developed an enzyme, a phosphodiesterase, that actually cleaves the human form of cyclic dinucleotide. So the analogy that I like to give is like a thief comes into your house and you have all the security cameras, right? And so you are sat somewhere taking wine, just looking at your screen monitor. The thief goes and just switches your camera off, right? So that's what the TB is doing. It's got an enzyme that basically switches that off. And so now, as a drug developer, how do you take this insight and make a TB drug? It means that the phosphodiesterase that a TB makes is what we call a virulence factor, right? So as a drug discoverer, we now started looking for drugs, small molecules that would inhibit this phosphodiesterase, okay? And so we did that. So this is the model that we have. The TB releases the DNA makes cyclic GAMP, but at the same time, the TB produces a phosphodiesterase that breaks down the cyclic GMP, and so there is no immune response to it. And so recently, we were able to develop or identify small molecule drug-like molecules that actually inhibit this phosphodiesterase. And it turns out that these molecules can actually tamp down inflammation. No, I misspoke. They can actually increase inflammation because they inhibit the TB enzyme. Okay. So this is an example of how we go to do fundamental biology. We actually identify targets that are important in infection, and then we go and we identify small molecules to actually inhibit these. Okay, so So the second part is cancer, right? Cancer is really important, but cancer and inflammation are always linked. I'm really beginning to appreciate it, that the biggest weapon against cancer is actually not a drug that we make. The biggest weapon against cancer is our immune system. So ultimately, if we can activate our immune system, we will be able to basically cure cancer, okay? So I'm not going to go through the whole immunology, cancer immunology, because this will take several talks. But I just want the audience to appreciate that we have the adaptive immune system and the innate immune system. Okay, so cancer immunotherapy is now a very, very big area. And cyclic dinucleotide is a pathogen-associated molecular pattern, right? Comes from bacteria. The immune system has evolved to be inflamed when it sees bacterial molecule. Inflammation is bad news for cancer. Not always. Sometimes too much inflammation is actually good news for cancer. Because if inflammation is chronic, then you actually get metastasis, okay? So this is, one example where, again, I collaborated with Claudia Gravekamp, one of the preeminent scientists in cancer immunotherapy using bacterial pumps. And in 2014, we published this. So this is one of the very early papers that shows that a STING ligand can actually lead to cancer vaccination, okay? And so this is cyclic dinucleotide. This is the natural molecule. And it turns out that since this publication, so almost 10 years ago, right, there is this 2004 update, progress in the research and development of STING drug targets. There are now 35 different indications that involve STING, right? Cancer, inflammation, infectious disease, 72 organizations. There are 53 clinical trials, right, that are involved with entities that are targeting STING. And this number really boggles my mind. More than 12,000 patent applications have now gone out, okay, with over 5,000 papers have been published. The CGAS STING field has exploded. And that is the reason why at the beginning of the talk, I mentioned that I made a bet. And it turns out that the bet was right, okay? So, there is a limitation to our discovery. Let me sort of go back here to point this out, right? So this is the bacterial signaling molecule, and for those who have biochemistry background, you will notice that this is a phosphodiester bond, right? And there is an enzyme called phosphodiesterase. Phosphodiesterases are everywhere in your body, okay? So even though it is generally recognized that this is one of the most seminal papers that showed that you can use cyclic dinucleotides to activate STING, this was never going to become a drug because the molecule would be chewed up in the body, okay? So a year after we published this, a company called Auditorium Bioscience actually beat us to it. Now, for the medicinal chemists in the audience, they'll be looking at this and said, you had to make this more stable, right? If we had made this molecule more stable and we beat Auditorium to it, we would have had $750 million from Novartis, because that's what Auditorium actually did, okay? So it turns out that the molecule contains a natural phosphodiester linkage, and this is the mechanism. It gets cleaved, and so therefore, it's not going to be a drug, okay? So what is the solution? You take a phosphodiester bond, you make it a phosphotioester. You just change oxygen to sulfur, right? Nature deals with oxygen, so you change it to sulfur, it becomes resistant, and that is a drug. That is what a company did, and they took many molecules to clinical trials, right? That's simple. But remember that this is sulfur which is going out of this ring, okay? So, sometimes in life, you just have to really understand your opportunities, right? We were looking at fundamentals. Let's show this for the first time. So we published our paper in 2014. And as we were doing the fundamental stuff, look at that. 2015 is when Auditorium published a similar thing, but they replaced the oxygen with sulfur. and their compound was better. Okay, so Novartis actually acquired Aduro for a lot of money, millions of dollars. They took their molecule to clinical trials for cancer, right? And well, we couldn't argue because we didn't make the sulfur analog, okay? Okay, so, at that point I was scratching my head. All right, okay. Maybe I might not have been here. I would have been somewhere on the beach somewhere, retired. Okay, all right. But I want to draw your attention to the fact that this sulfur is actually exo. The sulfur is coming out. And many companies, so Merck also has a compound that is out, right? Many pharmaceutical companies have jumped onto this, okay? And, at some point, I was just thinking, I'm a chemist. Why didn't I latch onto the analog, okay? And I missed out on this. But it turns out that there is a limitation to this molecule. So they have to inject the molecule into the tumor, right? They have to inject the molecule into the tumor. If you talk to a lot of oncologists, they say, well, that's a dumb thing to do. Why? If you can access the tumor, why don't you take it out? Right? Okay? So you don't want a molecule that you inject directly into the tumor. That is called intratumor injection. What you want is that you want a molecule you can take, you can do an IV, you can do a subcutaneous, right? And so we thought, there is still a challenge. Their analog, which the sulfur comes out, is not a good solution, okay? I'm gonna speed up a little bit. So this sulfur is exo, we call it XOS. These are very difficult to prepare, to scale up. And so I went to my chemistry background and I said, it looks like people have done many modifications, but there is one modification that no one has done, right? That is, instead of making this the sulfur, what about this oxygen? We call this the bridging oxygen. No one has made that into a sulfur, okay? And we had a reason that if you make it into a sulfur, that is actually a superior approach, right? Well, so to cut a long story short, let me go through a little bit quickly because I think I've run out of time. Oops. This is the crystal structure of cyclic dinucleotide bound to. Okay, so to cut a long story short, we actually made this analog where the sulfur is not the one pointing outside, it's the one inside. We call it the bridging, okay? This actually solves a synthetic bottleneck. This is now very easy to make, right? So we solve the synthetic bottleneck. And for those who have a chemistry background, you would also realize that the phosphorus is not chiral. If you put the sulfur exo, it is actually chiral and it's very difficult to synthesize, okay? This has been published. It actually became the cover of RSC MedChem in 2024. Okay, so let me move on because this is not a chemistry department, but the key point is that, does it work, right? Okay, so looks like this is a little bit slow. Okay, so let me quickly get to the cancer part because it's really exciting. Okay, so now we wanted to test that when we put a sulfur inside, it's actually more superior, right? And you don't have to inject into the tumor, you can actually use it without. So the way that we do this in the lab is that we take cancer cells from patients and then we do a xenograft in mice, right? We grow the cancer on an animal and then we treat it with our drugs, okay? And then afterwards we see whether our drug is working or not. So this is the idea. Okay, this is beautiful, okay? So this is the animals that were treated with this cancer cells, but they didn't get any drug. You see that the cancer grew very quickly. And then these are the molecules that we injected into the tumor. You can see that it reduces it, but not as much. And this one is the dosing that we did, a subcutaneous dosing. We didn't do intratumor, we just did a sub-Q dosing, and it works beautifully. And so let me show you the picture. We then, oops. No, okay. So let me show you the picture. So we grow the cancer, we grow the tumors, right? And then we treat it with our molecules. So this is vehicle without the drug. Look at how big the cancers became. This is when you add our molecule, but look at the subcutaneous dosing. So what I want you to do is I want you to consider this with that, right? So you can see that our new cyclic dinucleotide significantly shrinks those cancer. We've patented this molecule, and we are hoping that we'll be able to make versions of this and maybe hopefully take it into the clinic, okay? Okay, so I wouldn't go through this because I think that I'm now out of time, but inflammation is good for cancer, but when you don't have cancer, inflammation is terrible, right? Because inflammation can lead to arthritis, it can lead to Alzheimer's, Parkinson's, diabetes, and all of that stuff. And so now, instead of activating STING, we want to inhibit STING, okay? So again, we've come out with ways to inhibit STING. I won't go through the process, but just to show you that we have a molecule that inhibits things. So to summarize, we made a bet on cyclic dinucleotides and STING signaling. We think that this can lead to anti-infectives because it's important in bacteria. It can lead to anti-cancer, but it can also lead to anti-inflammatory. This field has been so generous to us in terms of research fundings, publications, and then hopefully a company. And all of this work would not be possible without funding from various places. We have really been blessed that the community has really bought into this program and generously funded us. But I always say that I'm the one who talks about the work. It's actually my brilliant graduate students and postdocs who actually do the work, right? Okay. And so all of this will not be possible without these fantastic people. So this is my current group. It's a very, very diverse group from all corners of the world. But I started my group in 2006. And so I always want to give a shout out to people who started the group. It turns out that these are the pioneers. So I started in 2006. These are the pioneers. So you've got students all over here who are all, 3 of the students are actually academics now. And some of these former students have all gone into academia. And it was a previous talk that I gave recently that I also had to mention on mentorship, because after close to 20 years, I've now begun to realize that the people that you leave behind or the people that you train sometimes are even more important than the science that you do, because they then go on and do really amazing stuff. And so some of these students have gone to work for FDA, NIH, pharmaceutical companies, and some have also gone into academia where they have their own group, and some are doing like really fantastically well. So I want to give a shout out. It looks like we now have like the next generation of the former CINTIF students who are also becoming professors. Jackie is an associate professor, but we say Jensen Wang started at the University of Kansas, and he did fantastically well that he was last year recruited to University of Chicago Medical School. So Ken Oyendip is at MESA, and I believe he's at MESA. I don't really know, but he's at MESA. And then Allison Kempin just started at Loras College. So she started only like about 4 weeks ago, but she is gonna do amazing stuff, okay? And then it turns out that there are also postdocs in the lab who are also academics. And so I really owe a lot of the stuff that the group has done to these fantastic students. They are really, really a pleasure to work with. And it's been a pleasure also presenting this, and I'll be happy to take any questions from you.
Wow. I think what we've had is actually a teaching session, not a talk, right? So the students, I hope you really enjoyed that, yeah? Yeah. And then he ended on a powerful note, which is that you guys actually do the work, right? We just talk about the work, right? We just go and talk about the work, but the work is actually done by you guys, which is really, the fact of the matter, really. Okay, so that was outstanding, Herman. Thank you so much. If we had time, we'll keep going, right? We could listen to this the whole day, but we'll continue another day. Anyway, so we'll take a few questions. I know we are behind time, but I'm sure there are a few burning. I see a lot of hands going up already. And one of the things that Peter and I were just talking about, when you mentioned Aduro, I think in 2019 or so, 2018, the founder of Aduro was here on this same platform. He spoke right here where you are speaking. Stephen Isaacs. Yeah, and he told us a story about Aduro, but who knows that he took 700 million from you? I didn't know. So it's a small world, right? Anyway, you get it back. I'm sure you make $1 billion very soon. So Yao, yeah. Yeah, Yao actually brought Steve Isaacs here. Please get a microphone so that we can hear you.
So yeah, I was just going to say that it's sort of an interesting cycle because Steve Isaacs, I met Steve Isaacs in Berkeley, where my brother is faculty at University of Berkeley. And Steve is very interested in biotech in Africa. So I think 2018 is when we invited him to speak. And it was actually his talk that pushed me to start Yamachi. And Steve is actually on my advisory board, together with Prof and a few others. So when you said Aduro, it was, and Aduro in tree means medicine. So that was something that I was like, wait, is this the same Aduro? And I knew about their sting work. So I'll send him a message and say, I know where he got his sting from. And I think Aduro has since he sold Aduro. So Aduro has been sold. And actually the company that bought Aduro has now also been acquired by Pfizer. And so it shows, where your sting is going. So it's pretty cool.
All right. DJ is at the back.
I just wanted to make a quick comment on the Nobel laureates and working with one. I wanted to apply a little bit of public pressure here. So I had the distinct privilege of working with a Nobel laureate. His mentee got a Nobel Prize. Then there's me. But Gordon has done amazing things. So thank you.
Well, this is my Nobel Prize right here. There's a hand here.
Yes, thank you very much. Many years ago, I worked with a chemist in England, Michael Blackburn, working on these diadenosine polyphosphates, and he was making thiol analogs. I was wondering if you came across those compounds in your work, because the interesting thing about them is that they make cyclic structures because the energy minimized structure of these diadenosine polyphosphates is with the base pairing. They actually cyclize in nature, but they aren't a hard cyclic, but in nature they are.
Yeah, I know of those polyphosphates. Absolutely. Yeah.
All right. There's another hand over here.
So considering the fact that you have chemistry background, you could synthesize all these structures, and you also knew that there are interactions with receptors and so on. I realized that somehow you may have been focusing on use of in vitro, in vivo methods. And I was just wondering, to what extent has computer modeling or docking, to what extent did this play a role? I don't know whether that's, if you had been using that, the Japanese may not have beaten you to that.
Yeah.
I don't know.
Yeah, no, excellent question. We we use docking and also molecular dynamics a lot. My personal thought on computational drug discovery is that if you have a lead, if you have already found something that works to a certain extent, then it's maybe easier. But typically when you go into a new, like if you go into a new fold, new active site, and there are not many molecules that are known to bind to that active site, it's very difficult for computational dockings and these generative models to come up with molecules that actually work, right? I mean, some people would argue that generative AI and machine learning ultimately would come up with de novo drug discovery, but I haven't seen that yet. I haven't seen that yet. But when we have a lead, if we have a hit which is not as good, then docking and molecular dynamics are really perfect tools to optimize. But usually getting it from the scratch, I'm still a believer of going through the library, where you take a whole library of compounds and then you first get a hit, and then you then optimize the hit.
Yeah.
Okay.
Yeah, so thanks for the interesting talk. So I may have missed it in the details, but I'm just wondering how your molecule will be able to prevent the thief from hiding in the barracks. So essentially, how will it prevent the MTB from hiding in the macrophages and evading the immune system? So do you probably go in that direction?
Well, so we need a cell permeable, right? If we get a cell permeable small molecule, then that would get into the macrophage, right? And then inhibit the TB enzyme so that the sting gets turned on. So that is the reason why we went for screening and we got cell permeable molecules. At the beginning of the talk, I did highlight that getting funding to do TB work, drug discovery, is very difficult. And so sometimes with TB work, you get a hit, and then it's almost impossible to get, the millions of dollars that needed to actually do hit expansion and hit optimization. But ultimately, that is what you would want. You want a cell permeable small molecule that can go into the macrophage and into the TB. There is an issue here, right? Because you wanna transverse the macrophage membrane and then also the TB membrane, right? So you have two big barriers to sort of think about, but it is still possible. Yeah. Okay.
All right. So more questions. Okay, let's take the two questions at the back. Professor Wellington, do you have the microphone? Okay. Harrison has the microphone. After that, then Professor Wellington. Harrison, go ahead.
Thank you very much. I mean, that was really interesting. Incidentally, one of my collaborators actually is in University of Maryland College Park in the molecular and biochemistry department. So the obvious thing is that there are modified nucleotides used for treating HIV. And I was kind of wondering, are there any plans to remodify some of these things to treat other diseases like HIV and hepatitis, because they are natural substitutes for nucleotides in the system? And the possibility, of course, selectively dimerizing HIV-RT, for example, because if we take the NNRTR binding site, you can easily have two of these molecules stick out, bridged by the diphosphate, and selectively prevent them from working. So I don't know if it's come to your attention and whether you'd be interested in future looking at some of these. Thank you.
Yeah. No, I mean, excellent question. As we speak, we have someone who is evaluating our molecules in Lyme disease. HIV, we haven't thought about that. There are other groups who are looking at sting agonists for viral diseases, but here the target is actually activating the immune system. But if I got you correctly, you were suggesting actually going after the bug itself, right? Yeah, it is actually plausible. I just haven't spent too much time thinking about that angle, and that is always why conferences like this are great, right? because there might be someone who is also looking at a different direction, and we can always have a chat over coffee or something if there is mutual interest. So absolutely. I just have not thought about it to give you a very intelligent answer. Yeah.
All right, last question. Oh, you have the microphone also.
Yes,
okay,
I have
two last questions. Two last questions. Then we'll continue the discussion during the break. Yeah.
Yes, I think so. So mine is to congratulate you, and because you really have walked the circle. We're thinking we should be working in R&D. You are situated there. So not just doing the research, but you focus in development. Of course, as a medical parasitologist, we look at the parasites when they are in the macrophages, and just listening to you and how you try to look at molecules that can get into the macrophage and to do describe some of the outcomes you're talking about. But I was just wondering again, we're looking at the possibilities of medicines, of course, in helminths, bigger worms. By the time you try to look at targets where you have to get the worms, the medicines either the molecules become, they go wary because they cause lots of adverse reactions. How do you target those bigger parasites? And then the second one, if you have some thoughts around that, is looking at how to get molecules into the brain. Because as they go across the blood-brain barrier, you begin to see. So I read recently that toxoplasma, one of the tissue parasites, could be used as a vehicle to take, molecules across the blood-brain barrier for them to be helpful. Just some thoughts. Of course, we can continue over the discussions in the corridor, but congratulations. Thank you.
Yeah, no, thanks. You just hit the point. So I didn't get the first one very well, but the second one, I did get it. So getting molecules into the brain is one of the holy grails, right? If you are working on, let's say, diseases where you want your molecule to be at the periphery, then of course you don't want it to get into the brain, because then that will give you a lot of side effects. But when you are working on certain diseases, so I actually have a current R01 developing a kinase inhibitor that has been shown to work, but it doesn't go into the brain. And there are some cancers that metastasize into the brain, like lung cancer, breast cancer, right? And so there is a lot of interest in getting molecules into the brain. I tend to be a big fan of small molecules. I know there are a lot of strategies with people using alternative strategies to get into the brain, but I am a small molecule sort of. I'm a big fan of small molecule, because I think that when you use small molecule, you don't have to do a lot of engineering, right? Whereas when you go into the nano realm, you have to tinker your molecule, you have to do engineering, you have to do targeted. But getting into the brain is one of the holy grails. At the moment, I would say that we do not have very clear rules. So it's mostly you make your molecule and then you test whether it gets into the brain. We don't have any good formalism as to strategies that you actually use to get your molecule into the brain. Yeah.
Okay, I'm here.
Yes, please.
I was anxiously following the compound with the tumor cell, the cancer cells, and how it would terminate. Unfortunately, it reduced, but did not terminate it completely. So which means you still need a surgeon and maybe the radiologist at the end of it.
Yeah.
So what comes to mind is, what advantage does it have, especially in terms of toxicity, as compared to current existing cancer drugs?
Yeah. No, excellent question. You didn't let me get away with that, right? So we didn't get a regression, right? We didn't get a regression. We got a reduction. So it turns out that the Aduro compound is now being used in combination with the PD-1s. If you look at infectious diseases, like HIV, for example, is now tackled with combination therapy, right? And so the goal is that if we take STING agonists and then we add, let's say, a PD-1 immunotherapy, and then we add a chemotherapy, right? Because this was done as a monotherapy. And so as we speak, we actually have ongoing studies that are now trying to do combination, because ultimately, you really want to regress the tumor, right? There are some tumors that are gonna be inoperable. And so for those tumors, you would really want to have, a small molecule too. But absolutely, you did catch that, and that is the limitation. Yeah, thanks. Okay.
All right, so thank you. So big thanks to Herman, and then I hand over back to Peter Kwashi, and hope we can catch up on time.
Thanks. I think that fantastic talk deserves another round of applause. That was just fantastic. And I think it's a sign that we get great keynote speakers, who have done great things and will go on to do even greater things. The fact that we had Steve Isaacs here and now we had Herman Tim here. So the chairs for this session are Dr. Yawaniwe, and I'm going to ask Dr. Anastasia Akins to join him because Dr. Constance Koko actually has traveled. I forgot about that. And I'll hand over to them to take us to the next session of the day.
Please turn on the microphone.
I believe that everyone will catch up strongly. And we have great scientists, so without much ado, we'll invite Professor Labs to take us through this topic.
Thank you so much. This is a hard act to follow. But maybe it will serve to show that to do research, it takes all types. So I think I'm pretty much everything that Herman is not. I'm not an African. I'm kind of old. I have a really small team, and I have very little money to work with. But I mean, so you should not, just be too discouraged, even if you can't fill all the characteristics of Herman. I think you might still find something to do. So I will also just focus on some very recent developments in my lab, done on work in collaboration with the University of Ghana, where I have worked over decades now. So, and as some of you know, I am one of the true believers in the virtues of the PFEMP1 molecules that are characteristic molecules of falciparum parasites. I will not delve in too much detail into those. I've talked about those over the years. So, as a parasitologist, you have to show a life cycle. I will not spend much time on it. Actually, the only thing that is really important here is that even though there is this 48-hour multiplication cycle in the red blood cells, if we are looking at parasites in blood films, we only see the ring stages. So those are the only ones that you will see in slides. And the reason for that is that the mature forms of the parasites are sequestering in tissues. That is where they will cause a lot of inflammation, endothelial inflammation. And so that is a really important element in the pathogenesis of falciparum malaria. And it will also allow the parasites to not go to the spleen, which is the place that parasites in red cells, malaria parasites, really do not want to go. So only the very young parasite stages where the infected red cell still looks pretty innocuous will have to go through the spleen. So this very brief summary of the pathogenesis of malaria is the background why a lot of the efforts to try to develop therapeutics against malaria through immunization against PFEMP1 the idea, these molecules mediating this sequestration are the PFEMP1 molecules, which are parasite-encoded molecules inserted into the surface of the infected red cell. And a lot of the efforts for people working on PFEMP1 has to be to induce immune responses that will prevent this interaction, that will disengage the sequestered infected red cells from the endothelium, reducing inflammation, and also make sure that the infected red cells, which are now opsonized, go to the spleen where they can be dealt with. All right. So the important point to remember here is that PFEMP1 molecules come in many different kinds, and the different kinds are linked to different disease outcomes. So PFEMP1 molecules that can bind to host receptors such as ICAM1 and EPCR are thought to be really important in the pathogenesis of severe malaria in children, including cerebral malaria, probably also severe anemia, although we don't know much about that. As you develop immunity to those kinds of PFEMP1, you grow bigger, and adulthood here is characterized by uncomplicated or even asymptomatic infections. And those are probably to a large extent mediated by PFEMP1 molecules that are allowing the parasites to stick to molecules such as CD36. So malaria, severe malaria in a place like Africa is mainly a childhood disease. The great exception to this rule, of course, is pregnancy. Pregnant women, again, become super susceptible to malaria because they become susceptible to parasites that can sequester in the placenta by binding to a molecule called CSA. This molecule is mediated by a particular PFEMP1 called VAR2-CSA, which is all these PFEMP1 molecules are very big molecules. The binding to CSA is mediated by a smaller part of the molecule called the minimal binding region. And we have, over the years, been able to convince ourselves and some other people that this is an important process. The acquisition of VAR2-CSA specific IgG is what really mediates the acquired protection against placental malaria. We know that some of these monoclonal antibodies that are generated against VAR2-CSA are broadly cross-reactive, and therefore there's been the idea that maybe it should be possible to make a vaccine that could generate antibodies that will prevent this placental sequestration and therefore prevent or protect against placental malaria. 2 clinical trials were published on this idea fairly recently. Unfortunately, I think results were not as promising as were hoped. I certainly was one of the strong believers in this and very, very disappointed. One of the big problems, Nikes will talk about this not on Friday, but this afternoon. I realized my slides were made some days ago before I realized this. One of the big problems with these molecules as vaccine molecules is their enormous polymorphism. I will not talk about this at all because, I mean, I will not steal Nikes' thunder, and I have only 15 minutes. Instead, last year I talked about, when I was giving a talk at the conference last year, I was talking about an additional complication, which is maybe that the epitopes that are recognized by the vaccine constructs, such as those used here, may not fully reproduce the epitopes that are present in the native molecule, and that in itself could be a problem with subunit vaccination. We discussed this in a paper that was published last year, so if you wish, you could spend 15 minutes reading that paper. That's quite an interesting one. I will not talk about that either. I talked about that last year, but I promised that if I was invited back, I would talk about another complication. And so I was very happy that I was actually invited back to talk about that, and therefore I will talk about this additional complication that we see. So most people working on vaccines are malaria vaccines aim to induce what we call neutralizing antibodies. And in malaria, that translates into inhibition of infected erythrocyte sequestration. But so that's taking away the parasites from the sites of sequestration. That, of course, is not only in pregnancy, but also could apply to all sorts of severe malaria. But there is another way that antibodies can actually mediate their effector function, which is sometimes a little bit overlooked maybe, and that is opsonization. So that is using antibodies to engage cells of the immune system. That could lead to activation of the classical complement cascade. Whether that is really important, I think the jury is out on that. We published a paper saying that we don't think it's very important. The Australians published another paper saying that we were wrong, and I think it's a little bit up in the air at the moment. I mean, it could go either way, I think, at this point. Phagocytosis, mediated by phagocytes carrying receptors that can recognize IgG, is probably quite important. We all assume that, although there is not that much literature on this, actually. But recently, there has been a renewed interest in other effector cells, such as NK cells, and I was happy to see some NK cells in one of Herman's first slides. NK cells and other cells can also engage in fighting diseases like cancer, like viral diseases, but also malaria, it turns out. Published first by a paper from the group Aurora et al., and then recently that is actually clinically important from Prasad Yaganathan at the Stanford University. So what is happening here is that the NK cells can recognize the opsonized infected red cell, and it will undo this, the ADCC response, what we call antibody-dependent cellular cytotoxicity, which will destroy the infected red cell. This is dependent on recognition of the Fc part of the antibody molecule mediated by the Fc gamma receptor 3 carried by NK cells and other cell types. So here we have an antibody molecule, we have the Fc region. Interestingly, the ability of the Fc gamma receptor 3 to recognize the Fc part of IgG molecules is very much dependent on the glycosylation of a single glycosylation site in the Fc region of the molecule. I've made it a little bit bigger so you can see it in the back also. The glycosylation tree looks like this. It comes in many different kinds. But almost always there is a fucose moiety in the proximal part of this glycosylation tree. And that is critical when it comes to recognition of IgG by NK cells. If the fucose is there, then the NK cells don't bind really to the IgG molecule. And since essentially all IgG has this fucose, so this total IgG, and you can see about maybe 90–95% of IgG has this fucose, NK cells are not generally considered very good at recognizing IgG, and we call the Fc gamma receptor a little bit, I think, derogatory. We call it the low affinity Fc receptor. It's the good for nothing kind of Fc receptor. But that also goes for malaria antigens, such as the myristoid antigen GLOB. But a couple of years ago, we could show that if you, from this population of highly fucosylated IgG, purify the IgG that can recognize the PFE-MP1 molecules, we have the VAR2-CSA that I already mentioned, and another PFE-MP1 that is involved in severe malaria in children. Much of this IgG is actually lacking fucose. So this is a subpopulation within this. And if this is lacking, then the Fc receptor very well recognizes this IgG that lacks the fucose and can kill the target. What is even more amazing is that even though this happens in the Fc part of the molecule, which is supposedly conserved, this is an acquired phenotype. So when we looked at women here and sorted them according to pregnancies, women in their first pregnancies, most of them have highly fucosylated VAR2-CSA specific IgG. But as you move on with more and more pregnancies, the VAR2-CSA specific IgG become increasingly afucosylated. So it is an acquired phenotype. We also noted that if we, in the same women, looked at another PFE-MP1, the VAR6 molecule, which is involved in severe malaria in children, we didn't see this. So all these PFE-MP1 molecules were markedly afucosylated, even in primigravid women, and we were wondering about what is going on here. But we suspected that maybe this is because this is a childhood-associated PFE-MP1. Maybe these women, when they were girls, also did this, but with the VAR6, so we could see this. And once you go down, you stay down. And we were recently able to show that actually using a cohort of children instead, that if you take children, now we're looking at VAR6 here, you can see that actually in very young children, they have pretty much fully fucosylated VAR6 specific IgG. As they grow older, it goes down. So it kind of mirrors this in the pregnant women and VAR2 CSA. That only happens later in life because you only acquire your VAR2 immunity once you get old enough to get children. Yes? So when you combine the data, you see that this phenotype is acquired, and once you get to the afucosylated state, you stay afucosylated. So this is, I think, is interesting from an academic immunological point of view, but does it also matter? It's a big question, of course. So in children, susceptibility to severe malaria decreases with age, but levels of, if you believe in PFMP1 specific immunity as I do, the levels are going up, but the degree of afucosylation is also going up. So what is important here, levels or afucosylation? And similarly in women, susceptibility to placental malaria goes down with increasing parity, but levels of antibodies go up and the degree of afucosylation also goes up. And again, and here we have the addition that we know the adhesion receptor, so we also know that women are acquiring more and more anti-adhesive antibodies. So what is it that really cuts the cake? Is it levels? Is it anti-adhesion? Or is it afucosylation, and thereby engaging cells of the immune effector cells? So now I'll move to my punchline, and so because the answer is afucosylation is really important, we believe. Here we have a group of Ghanaian children, and we looked at these children and at the levels of VAR6 specific IgG and also the degree of afucosylation of this IgG. We corrected the data for the levels, differences in levels, and now you can see that children with markedly afucosylated VAR6 specific IgG are much less prone to develop anemia than children who have highly fucosylated VAR6 specific IgG, suggesting that that the degree of afucosylation is actually clinically important. In a similar way, children with afucosylated IgG is also much less likely to become infected, to be parasitemic, than children with fully fucosylated VAR6 specific IgG. In pregnancy, we see a similar thing. Again, we have women here. Now this is a study done in collaboration with Nikes from Benin. A group of pregnant women here. We looked at IgG levels to VAR2 CSA, the ability of this VAR2 CSA specific IgG to inhibit adhesion to CSA, and also the degree of afucosylation of these antibodies. And you can see that neither the levels nor the anti-inhibitory capacity or indeed the afucosylation levels could tell us much about how anemic the women would be at delivery. Probably that because pregnant women tend to be anemic in general, and there are many reasons for that. When it comes to placental malaria, we had hoped to see a big difference. We didn't really see a difference here in just, do you have parasites in the placenta or not? But what really is important here, I think, in terms of low birth weight, which is really the main complication of placental malaria. I mean, the risk of delivering really underweight babies who will be at great risk of dying in infancy. Then levels as such doesn't really matter much. The ability to inhibit adhesion in the placenta certainly matters. I mean, and that is really the rationale for the anti-adhesion vaccine against malaria in pregnancy. But amazingly, even if you control for the other ones, the degree of afucosylation, so thereby the ability to attract immune effector cells, really matters. And you can see here this is a log scale, so it is quite a dramatic reduction in the susceptibility of these women to placental malaria and negative birth outcome. So I presented to you the first evidence, I think, that the ability to engage these immune effector cells is really an important component in protection against malaria, in addition to the ability to block adhesion. And I think that actually calls for perhaps even a need to reconsider the ways we think about PFEMP1-based vaccines. Everything has been about neutralization, the blocking of adhesion so far, but maybe we should also think about how can we create vaccines that will engage effector cells in a better way than previously. And that is important to mention here that if you do subunit vaccination with the molecule VAR2-CSA in the vaccination experiments that I just showed you, these subunit vaccines will induce IgG that is completely fucosylated. So in marked contrast to the IgG that the women acquire in response to natural infection. Clearly, follow-up studies are needed. So there's a lot of room also in this room for important studies to be conducted. We need some independent verification. Maybe this is just a one-off fluke. We need additional antigens. Frederica Party, who is in the audience, is now already looking into this. And then there are, of course, additional cells in addition to the NK cells that are important here. Certain gamma delta T cells, various subpopulations of macrophages, and so forth that also carry these molecules. And then, of course, we need longitudinal studies, which we haven't done so far. So let me just end by acknowledging some people who have been instrumental. Mike Ofori, who has been my brother in arms here for decades, still is. Nikeas, who is another very good friend and close colleague and an intellectual sparring partner when it comes to this. Mary Lopez Perez, who works with my lab. That is about 50% of my lab, this person. So I have a really small group. But she has been responsible for doing the lab work here. I would like to, of course, also thank a lot of other people who have been involved in various studies that we've done, the sponsors that we have. And then I'll just end by also acknowledging and commemorating Francis Nkrumah, the former director of Noguchi Institute, who was director when I started here more than 30 years ago now, and welcomed us warmly to come and work here. And he just passed away a month ago, and I just want to pay my respects to him. Without him, we would not have been able to do this work, and that has been fantastic. Some of the greatest years of my scientific life have been spent here. So with this, I will stop and take questions.
Okay. Yeah, we already have 3 questions, so take.
Who's going first?
DJ.
Yes, wonderful talk as always, Lars. I just have a really quick question with regards to endemicity and afucosylation. Have you looked in different areas of endemicity and been able to find some rough correlation of how endemicity maps with age and the pattern of afucosylation?
No.
Okay.
I think no, I mean, I mentioned the need to do a lot of more studies. That obviously is one of them. I mean, the only studies that we have so far are data from Ghana and from Benin now. But we don't, the microphone. Sorry, the only studies we have here are from Ghana and Benin so far. There are ample opportunities to try to do this in other places of the world also, obviously.
Yeah. And just a really quick follow-up. In terms of pathogenesis, how do you think this impacts on the actual development of let's say, severe malarial anemia in this particular context? Thank you, sir.
I really have no informed response to how it impacts the pathogenesis of severe anemia. I know which is, I think, your pet topic. That's something maybe we can discuss over a cup of coffee or something, but I mean, I have no really, that will be speculation on my part.
That was great. I'm not going to criticize it. I have a question though. Why fucosylate in the first place?
Why fucosylation in the first place?
Yeah.
I think the reason that most of our IgG is fucosylated is to prevent NK cells from going around and killing our cells. So an antibody may easily land, I mean, say in autoimmunity, but in general, antibodies can still land on our own cells, and it would be disastrous, I think, if those accidentally landing antibody molecules would trigger an NK cell response. So I think that is a reason why.
And so then in a normal immune response or a normal antibody response, presumably what's happening is you are gradually fucosylating as the antibody response matures?
Well, it doesn't happen to all antigens. And that is a really fascinating story that I didn't have time to go into, and I won't go into now even in answering, because it's a beautiful thing. Because this was really found out in terms of alloimmunity, with rhesus immunization of rhesus negative pregnant women against their rhesus positive offspring, where it turned out to be a really bad thing. I mean, because then you also see this afucosylation, and you thought, why do they do this? And I think that is because the alloimmunization is collateral. I think this evolved in response to malaria. I think. So I'm beginning to convince myself that the primary job of NK cells, even though we always herald them as cells that are involved in antiviral and anti-cancer responses, were actually as anti-malaria effector cells. That may be stretching it, but why not? I mean, we can talk over that, and I will have much coffee to drink here to talk to various people about this.
Yeah, go ahead.
Thank you for a really great talk. So easy to follow, so clear. Okay, you did say now that afucosylation is not good for everything. But if you were to try and induce these types of responses with a vaccine, how would you drive afucosylation?
That is an excellent question. I don't know whether everybody got the question. Is if you think that an afucosylated IgG response is a good thing, how can we modify our vaccination strategy to drive the antibody response to include or be dominated by afucosylated IgG? I cannot take credit for that. That is really the work of good colleagues in the Netherlands. They hypothesize, and there is no firm evidence yet, that what really drives the afucosylation of the IgG is a post-translational modification on the part of the B cell, but that they hypothesize that there is a system in the B cell where the B cell can recognize where the antigen sits. So a response, a B cell responds to a soluble antigen, or a B cell responds to an antigen on a foreign pathogen, a bacterium, whatever, will be fucosylated. That an additional signal that the B cell can sense some kind of, that the antigen is sitting on one of our own cells. That could be a virus-infected cell, that this cell should be killed. And that it could also be a malaria parasite-infected red cell. This cell should be killed. Or it could be a cancerous cell that are now expressing various neoantigens, that this cell should be killed. But exactly how it operates, we don't know. But how to generate such a response is also a bit of a tricky question in a way. But it could be that if we could make vaccines where the foreign antigen is presented on, say, an enveloped virus that is covered in host membrane, maybe that can work. But it's speculation. Nobody knows at this point, I think.
Hello, this is a great talk. Thank you. You know, it takes time to develop good IgG antibodies. But in the early stage of the infection, the body will produce large amount of IgM, right, to combat the infection. Is afucosylation, right, specific to IgG, or is also involve other immunoglobulin isotypes?
So the question is whether afucosylation happens with IgG only. As far as I know, yes. In any case, the only Fc receptor that is affected by this afucosylation is the Fc gamma receptor 3. So other Fc gamma receptors, such as the Fc gamma receptor 1, carried by phagocytes, they don't give a shit whether the IgG is fucosylated or not. They will just engage it. So that is why we call it the high affinity IgG receptor. They just bind anything. So it's only the Fc gamma receptor 3 that is carried by the NK cells and various other cell subsets that are affected by this, which I think is kind of smart on the immune system, but I've come to realize in my old age that the immune system is actually pretty smart. I think I should stop now.
Yes. Thank you very much, Lars. Round of applause.
Thank you. I believe we are having a very exciting morning session of talks. So our next presenter is Collins Misita Moranga. He's one of our WACCBIP alumni that we are very proud of, and he's going to talk to us about genetic diversity and structural variation of Plasmodium falciparum in complex malaria infections.
Good morning, everyone. As WACCBIP celebrates their 10th anniversary, I just want to say that I'm very proud to be one of the alumni from the WACCBIP system. And today I'm going to divert a little bit from what last feeders talked about and talk to you about the genetic diversity of Plasmodium parasites in complex malaria infections. And today I'll probably focus a lot on the multiplicity of infections. So globally, at least 3.2 billion people still are at risk of malaria infections. And this stem widely from people of all races and geographic locations. But in West Africa, if you look at the total number of cases that were present back in 2010 was about 30.6 million cases. But this number has shot up to 73.1 million in 2022. So it's a stark increase, despite the fact that there's been a lot of funding being pumped into malaria control programs, as well as interventions such as, treated nets, SMC, and the rest. Now, still the parasite rate persists at a very high rate in this West African region, and Ghana is part of the West African region. So one of the reasons is that the parasite is highly diverse, and most often it evolves, and it uses these mechanisms to probably evade drugs. The drugs we use are developing resistance, evading the host immunity, as well as other control mechanisms that we are using, such as SMC. And on a short or large extent, it's already impairing our future vaccines that we are already developing against this parasite, even before we start using them. Large collaborative efforts led by MalariaGen have sequenced for over a decade. They've already sequenced about 20,000 parasite genomes. And one of the things that they show very clearly is that the parasites in different geographic locations, they express or they are genetically diverse. And this is a principal component analysis, and you can see that the parasites that are stacked in this cluster are mostly from the African region, while all these others are from Asia and Southern America. So you can see that there's already a separation in parasites from these different regions. And if you zoom into Africa, one very good research done by Prof. Alfred Ungwa is that he looked at the subpopulations of Plasmodium falciparums in sub-Saharan Africa. And you can see even within Africa, although there was a cluster of parasites within Africa, there's a huge separation of parasites when it comes to different countries within Africa, and especially separation of Eastern Africa parasites and West African parasites. Now the question is, does this continue all the way down into the complex infections within these parasites? And one of the measures that he showed in his paper is using FWS. FWS is a measure of, is an indicator of inbreeding, and mostly we use it to show clonality of infections. So parasites that tend to be close to 1 up here are basically monoclonal parasites, And then parasites that are below this demonstrate that you have multiple genotypes within that particular sample. And you can see that across all the different countries, there's presentation of multiclonality of parasites. If you zoom in down to just West Africa and look at parasites in different endemic regions, you still find a similar or high level of genome complexity. And one research that was done here by Prof. James Abugri and Gordon and David Conway and the whole team is that they showed that even if you zoom into, this is still FWS measure. If you zoom into two high transmission areas, this is Kintampo and Navrongo, which are high transmission areas. Of course, you get monoclonal parasites up here, and this line is usually at 0.95. But then you still have a high population of samples that end up being multiclonal. Now, the question we are asking is that, or the problem we see here is that we still don't have complete information on the complexity and diversity of this parasite, Plasmodium parasites. And this could be, an indicator or assist in eradication of malaria or elimination as set out. So in this work, I'm going to zoom into one small region, which is the Upper East region. This is Kasena-Nankana district, and which is right on top of Ghana. It includes Navrongo. So we went to Navrongo and we sampled community cases and clinical cases. And from most of the research I've shown you in this previous work, I would say majority of them is that they've been done on clinical samples or samples collected at health facilities. And here we used a few community cases to compare with clinical samples to see whether there's actually a difference between these particular parasites. And this was our research question, whether there is diversity differences between the community and clinical parasites. So we aim to show the differences in multiplicity of infection, as well as show the differences in population structure and genetic diversity of parasites through whole genome sequencing. So we obtained ethical clearance. We did a malaria test before administering a questionnaire, and we sent some of these samples to MalariaGen for generating a genetic report card. This is basically to check for drug resistance genotypes within the population. I also did whole genome sequencing and SNP and genome mapping and SNP identification. So what we've done so far is to also check on multiplicity of infection of these parasites as well as population structure. So we had 154 samples that were successfully sequenced, and 75 were clinical cases and 79 community controls. These are some of the demographics that we collected. The parasite density was significantly different between the two populations, and we grouped our individuals to different age groups. So we basically said we are going to look at the differences in children below 10 years and young adolescents all the way to older adolescents, as well as adults more than 18 years. And then we also checked on participant education and occupation, because socioeconomic differences also could determine on the behavior of mosquito infectivity. What is very clear here is that we have near equal populations between the three different groups, and you can see that the parasite density is completely significantly different. So the community individuals, these are box plots showing the distribution of parasite density between the age groups, and you can see that the community controls significantly had lower parasite density. So we go on to check for the different drug haplotypes, and this is just analysis of the genetic report card. And we've so far looked at the clinical samples, and you can see that majority have the wild type CRT haplotype. Pregnant women mostly receive SP for intermittent preventive treatment, and you can see that the frequency of this triple mutant RNI is very high, as well as for SP, you can see that the double mutant is also gaining, in terms of frequency. We also look at MDR1 haplotypes, and you can see that there is quite variation. This is the wild type haplotype, and all these haplotypes that are show resistance to malaria drugs. Midazoquine is also one of the drugs that is used in this region for SMC, and I have to say that Navrongo is one of the regions that SMC has really been applied to, and you can see that we still have the wild type. We don't have the KELCH mutations that are causing resistance in Southeast Asia. So back to the multiplicity of infection. What we show here is the FWS measure, the clinical cases, and the community controls. This is a box plot, and you can see that the clonality of parasites, or the median differences between the multiplicity of infection between the parasites is quite different. And I have to say, probably this is the first time we are showing that community controls have a high level of multiclonal infections compared to the clinical samples. So, and if we tease out some of these populations that are monoclonal, you can clearly see that there is single genotypes. But when you section out some of the parasites around FWS 0.5, you see more than one genotype. If we break this down to different ages, the same multiplicity of infection, you can see that the community controls significantly have lower multiplicity of infection, or the median, compared to the clinical cases. And it's interesting that only a few adults tend to have had monoclonal infections, but majority of the adults have multiclonal infections. And these community individuals probably could just be as reservoirs for transmission of malaria parasites. And we tried to correlate the multiplicity of infection with parasite burden. And this is very interesting. What you can see is that this is parasite density on the y-axis and the multiplicity of infection. So as parasite density increases, or ideally you'd hope that the multiplicity of infection will probably have some level of correlation with parasite density. That is, monoclonal infections causing higher parasite burdens compared to multiclonal infection because of competition. But then you see that both the clinical and the community controls have generally no correlation, not significant in these populations. And this is interesting because it throws in a spanner on why some parasites, have high parasite density and they are monoclonal, yet you have others that have low density and they are multiclonal, and vice versa, on parasites that are in this region, high multiplicity of infection, and low multiplicity of infection, that is multiclonal infections with very low parasite densities. So these are things we hope to tease out and look at into these samples and see what could be causing these differences. So we also did principal component analysis of the population to try and see what is the population structure in these samples. So this is each dot here just represents an individual case, and we have both the clinical and the community samples here. So really what we are trying to do here is to reduce the SNP, calculate the distances between each individual SNP, and then reduce it into a lower dimensional space to be able to observe the differences between the samples. And you can see that there is very low, the population structure is a little bit conserved, which means these parasites share or have a good ancestral history, despite the fact that they are causing differences in clinical cases. We did LD pruning so that we remove SNPs that are in linkage disequilibrium to see whether there will be any separation in SNPs that are not in linkage, and you can still see that they still stuck together. So, and this is as expected because we've sampled from a very, within one population, and you expect that all the parasites circulating within that geographic location will probably be highly related. We also do some pairwise distances, that is to try and do identity by state. Each of these is a correlation matrix for each individual SNP, and you can see that it's a lot, but if you reduce it to using multidimensional scaling analysis, which is basically a method to instead of using a PCA, on secondary generated data, you use this MDS to be able to reduce it to a dimensional scale. And interestingly, you can see there's a slight separation of the clinical cases and community controls, although they are still stuck together in this. We looked at FST, which is basically a measure of the differences between populations, and it was very low. You can compare this number to, generally in the different geographic population, the range is between 0.2 to 0.4. So 0.03 is basically showing there is very low difference between the two populations. And this is as expected because all the samples are coming from the same geographic location. So what we are trying to do now is to try and tease out and see what could be accounting for the differences in terms of which genes or which SNPs could be accounting for the differences between these populations. And if we take the population level allele frequency, most of these are low frequency alleles, so they will not be having any much impact. But think of it this way: if you pick the SNP that is here and its allele frequency is down here in this other population, you can find the difference between the two and see whether it's significantly different. So we did that, and we picked SNPs that at least there was a difference of more than 0.2 between the two samples. And if we pull only those SNPs, and we could do this vice versa, but at the moment we did between clinical and community cases. We could do it the other way around and see which SNPs are in the community and not in the clinical cases. But what I'm presenting here is from clinical to community controls. And you can see there is a significant difference between this. There is a difference between these two populations, of SNPs. What we try to present here is the raw allele frequencies of these two groups. The scale is slightly higher because I stacked them together, but most of these, if you look at them carefully, you will see that the frequency of this compared to this is more than 2 times. And I still present it as a table here. So one thing that we did, we used Fisher's exact test to test whether the allele frequencies are different between the 2 groups. And you can see that we have a few genes. So these are only a representative of genes that were significantly different between the 2 populations based on the allele frequencies. And most of these genes, for example, I'll use the chromosome 9 SNPs. Most of them come from one region of MSP1. And all these others come from, surfins, etc. But MSP1 is of interest because lots of studies have gone in to show that they can differentiate between different allelic genotypes of parasites. So we are still teasing that out, but I would like to summarize that, multiclonal genotypes dominated in community cases compared to clinical cases. But then generally, both groups have highly multiclonal parasites, and this could be due to being in a high transmission area. And the parasites in and MSP1, basically is one of the genes that we've already teased out to be, different between the 2 groups. We are still doing a lot more analysis into this to be able to understand the differences. The other things we are trying to do is to develop, analysis pipelines to be able to sequence samples from more populations. And this is basically using long reads, nanopore sequencing to try and generate, and this is one of the pipelines we are trying to develop to be able to understand the genetic variation. Because long reads allow us to study, because Plasmodium has long stretches of 80-rich genome. And if you're able to sequence the long tracks of these parasites, you can be able to study most of these haplotypes much more carefully. And we could do this with combination with short read sequencing, and that one too. So this is a sample of some of the genomes we've already generated, and you can see this is coverage, 10x, 60x. You can already see that we are generating very good coverage genome-wide for some of these parasites. These are lab isolates, but then for clinical isolates, you can still see we get up to 10x in terms of coverage for these parasites. And we are trying to also develop methods or analysis pipelines for analyzing whole genome samples from diverse sample populations to be able to understand the genetic variation in parasites. I would like to acknowledge my supervisors, starting with Prof. Godona Wandari, Dr. Lucas, Thomas Otto, Prof. David Conway, Prof. Alfred, Dr. Victor Asiola, who assisted in sampling collection in Navrongo, as well as the entire Navrongo Health Research Center and all the study participants. I also like to acknowledge all these people, including the Department of Biochemistry. Thank you very much.
Thank you, Collins. We'll have time for only one question. Okay, DJ.
I'll ask a long question that you can try to summarize, okay? So when we think of multiplicity of infection, right, in very high transmission regions, it can be, because of this particular clone that I have, it's highly variable. It can also be because I have multiple clones circulating in my system. So my question is, I wasn't quite sure what the definition of multiplicity of infection is. Maybe you can help me. And just really quickly, the reason I ask you that question is more than 20 years ago, I sat around with some theoretically intelligent individuals, and across time we've been working on this with a very simple question. In severe disease, does one have a, quote, more complex infection, thereby the immune system can't respond to it? And we've looked repeatedly and not actually seen that. And so that part is a comment, but phasing and the definition of multiplicity of infection, I'm curious how you did that to get at, I have a complex infection versus I'm complex because I'm polyclonally infected. Thank you, sir.
Thank you very much. So the FWS metric, what it does is that, you know, once the mosquito picks up the parasite, depending on the complex genotypes that it will pick, there is a chance of recombination events happening within the mosquito. And if you have a few diverse parasites within the mosquito recombining, and this re-injects back into the human, you already have a complex genotype. Now, if an individual gets multiple mosquito bites with different parasites, it increases the complexity of these parasites. So what the FWS does is that you calculate within the host heterozygosity, and you can find the ratio of that using the within population heterozygosity. And this heterozygosity is basically because Plasmodium is a haploid genome. So if you're able to measure the level of, you know, heterozygotes within this population, you can determine whether there was multiple clones within the sample or not. So yeah.
Okay. Thank you, Collins. Let's give him. Okay.
Okay. We now have Muki taking us into another great talk, which is the only gap between you and your coffee break.
Okay, thank you, Chair, and thanks to the organizers for giving me the opportunity to talk about the work that we do. So when I looked at the program, I thought I was going to be the only big TV guy in the room, but our keynote speaker took it from under my feet. So our research is basically trying to understand natural immunity to MTB infection and how we can harness that for the development of the next generation of TB vaccines. So the keynote speaker already introduced this slide, so I'm not going to waste any more time. But what I'm going to point out is also the incidence of tuberculosis in the past 2 years or in the past year. So South Africa, where we do our research, is considered to be a high endemic country with the incidence of per 100,000 population of about 468. And Cameroon, which is my home country, has an incidence of 179. So this morning I put Ghana there just to try and impress you. So the incidence of about 140 per 100,000 population. So so what happens is that when people are exposed to TB or to index cases of TB, in the short to long term, a lot of people become infected. So they might have the latent form of the infection, which is called latent TB infection, where your body is able to contain the bacteria in the form that is not replicating. But some people also go on to develop active TB, right? But we do find that despite this sustained exposure, despite this heavy exposure over a long period of time, there are still people that do not develop any sign of infection at all. And these are what we term them the resistors. And that is my interest, my research interest. And the question is, what is contributing to this resistance to infection? Is it the immune system? Is it the genetics? Is it both? Or is it even none? So to try to understand some of these questions, so we had an interest in cells that are called MET cells, which stands for mucosal associated invariant T cells. So classic textbook immunology will tell you that for T cells to be activated, they require three signals, which are antigen presentation, cytokine signaling, and co-stimulation. But these MET cells are interesting in that their activation pathway is quite different. So they can be activated by either cytokines alone or by antigen presentation. And the antigens that are recognized by MET cells are from the riboflavin pathways, which are presented on the MR1 and recognized by T cell receptor called V-alpha 7.2 on the MET cells. And once these MET cells are activated, so they become very effective MET cells, very producing cytokines and cytotoxic molecules that are able to kill bacteria-infected cells. And some studies have shown in the past that these cells are important in preventing infection, both in humans and in mice. So a few years ago, I wrote a grant to the Wellcome Trust to try and understand whether these cells do have a role in protection from infection. And the simple questions we were asking is whether protection or resistance is associated with higher MET cell function, or whether this was also associated with a high ability of MET cell to be functional or to be cytotoxic, as well as an increased capacity of activation, or whether there were genetic polymorphisms in these MET cell pathways that are protective or associated with resistance. And obviously, writing a grant, you have to be focused. So we focus on the MET cell, but once you have the money, you can pretty much do what you want within limits. So to start answering these questions, so we approach a cohort of individuals that we consider to be very highly exposed. And these are healthcare workers who work and treat TB patients on a daily basis. So we know that they are exposed on a daily basis, but we then looked at healthcare workers who have worked in a TB treatment facility for a minimum of 5 years. So we considered 5 years to be a long enough period that even after 5 years of exposure and you are still not infected, that means that there must be something that is protecting you. So we also tested these individuals for HIV infection. And those that were eligible were HIV infected and worked for a minimum of 5 years. So we tested them for MTB infection using the two classical tests that are being used at the moment. So one of them being the tuberculin skin test, and the other being the interferon gamma release assay. So once we had these two tests, we then combined them. And too quick. So we then combined them, and the individuals that were double negative for both tests were considered to be resistors on your right. And those that were double positive for both tests were considered to be already have the latent TB infection. And our positivity was defined by a TST value of more than 10 millimeters and an IGRA value of more than 0.35. So once we identified this group of people, so we then collected a lot of samples for the different analyses. Some of the samples that we collected were plasma for cytokine analysis. We also collected PBMCs for cellular analysis. We collected RNA for our gene expression analysis, as well as a DNA for genotyping of polymorphisms. So all in all, we screened over 780 participants. And out of these 780, so we identified 129 that were resistors and 145 that already had the infection. There were also other groups that we were interested in, such as the people with previous TB, so those who have had TB in the past, but with successful TB treatment, as well as those who were discordant, positive for one test, negative for the other test. So we also wanted to look at their immunological profiles. But for today, I'm just going to focus on the LTBI and the resistors. So this is just a table of characteristics for the participants, or demographics for the participants that we had in our study. So we collected a whole lot more factors, but I'm just going to present a few here. But the take-home message is that there were mainly no differences between the resistors and the LTBI. So they were mostly similar age, about 36. Majority were female. Most of them had worked between 5 to 10 years in the TB treatment facility. as well as there are still others that have worked for more than 15 years, but the numbers were pretty much similar between the two groups. And in South Africa, of course, we had different ethnic groups in South Africa, but majority of them were black Africans, but we also had a few in the different groups. And this is the significance here was mainly driven by these few numbers on the other ethnic groups, but majority were in the black African population. And in terms of masks, so most people said they were wearing surgical masks, but the N95 masks, which are the most protective masks, so there were also people who said they were wearing them, but they were higher in the resistor group compared to the LTBI group. So to be able to start answering the laboratory questions or the immunological questions, we focus on a group that we define as extreme phenotypes. So from the resistors, who were the 129, so we isolated a group that we call them extreme resistors. So with a TST of 0 and an IGRA of less than 0.2. And we also had extreme LTBI, TST of more than 15 and an IGRA of more than 1. And the rationale for choosing this extreme phenotype was that if we are to observe differences between the two groups, this is where we are most likely to see those differences. So the first question that we were asking was whether med cell frequencies were different between the two groups. And as a reminder, that med cells were identified by flow cytometry by a combination of CD161 and the MR1 tetramer. So the 5-OPRU is a riboflavin metabolite used to recognized by med cells. And the first thing was that there were no differences in med cell frequency or numbers between the two groups. Then the next thing we did now was look at mixed functions, and we stimulated PBMCs with whole mycobacteria, live bacteria. BCG is the vaccine that is currently used for prevention of TB, which was developed over 100 years ago. And we also used the laboratory strain of the MTB. And what we observed was that whether stimulation with MTB or stimulation with BCG, expression of granzyme B, which is a cytotoxic marker, was significantly increased in people with LTBI compared to resistors, as well as the expression of interferon gamma was also higher in LTBI compared to resistors. But TNF alpha was similar, and the total cytokine was similar. So this is opposite to the hypothesis that we had. So then we looked at the activation of med cells by looking at three markers: CD38 expression, HLA-GR, and CD27, which are classical markers of T cell cell activation. And what we observed was that there was no difference for CD38, but there was an increase in HLA-DR expression in resistors, and a trend towards the increase in CD27 in resistors compared to LTBI. And this was what we hypothesized at the beginning. And then now we then looked at med cell memory. So in vaccination, basically what you do in vaccination is to try and induce a memory response. So we decided to look at med cell memory. And what we observed was that majority of the med cells were in a transitional memory phenotype, which is opposite to classical T cells, where majority of them will be a central memory, but the central memory med cells were lower. And the take-home message is that there was no differences in the memory phenotype between the two groups, except for effector memory med cells that were higher in LTBI compared to resistors, which are also still consistent with the function of med cells that we observed. So I'm just going to pack the med cell story here now and also talk to you about some of the other analyses that we are doing in the cohort. So the first is looking at gene expression analysis. This is bulk RNA sequencing from whole blood. So when we did the bulk RNA sequencing, we identified over 18,000 differentially expressed genes. And of these 18,000, 26 of them were highly expressed or enriched in resistors compared to LTBI, while 65 were highly expressed in LTBI compared to the resistors. So these are just, if you look on the left side of your graph, so these are the 26 genes in the resistors and the 65 in the LTBI. So moving, so we also did a 65-plex where we looked at 65 different analytes using Luminex assay. And then we tried to then look at the genes that were highly expressed in resistors and LTBI, whether there was a correlation with some of the cytokines, just to see whether the gene expression was also influencing the cytokine production. So there are still other analyses that we did, but I'm just presenting the correlation here. And the top 4 cytokines that were mostly correlated, either positively or negatively, with these genes were the MEP1 alpha, which is also CCL3, as well as the macrophage inhibitory factor proteins. And these are also involved in innate immunity, innate immune responses. But looking back at the gene expression and doing immunodeconvolution, so I don't also understand this, so bioinformatics will understand. So what they did, or the bioinformatician did, was looking at this differentially expressed gene and looking at the cellular pathways that are associated with these genes. And what they found is that, or what we found is that the genes that were highly enriched in resistors were mostly associated with the monocyte pathways, while the genes that were highly enriched in LTBI were mostly associated with the CD4 T cell pathway. And then lastly, so zooming in again on the cytokine analysis, so we first looked at the cytokine and compared them in this heat map, comparing them between the resistors and the LTBI. So the LTBI to the right and resistors to the left. So when we looked at the plasma samples, so there was no discernible differences between the resistors and the LTBI. But when we looked at from quantiferon supernatants stimulated with MTB antigens, so what we observed is that most of the responses were in the LTBI group, so most of the cytokines were in the LTBI group. But when we stimulated with PBMCs with BCG and MTB, so there was some sort of a shift towards increased cytokine expression in the resistors. And most of the cytokines that were mostly enriched with the stimulation were still either involved with the innate pathway or with the IL-17 immune response pathway. So I hope I've convinced you that in resistors, so MET cells frequencies were similar between LTBI and resistors. And the function of MET cells was higher in LTBI, which was opposite of our hypothesis, while the basal MET cell activation was higher in resistors, which was in the direction of our initial hypothesis. And gene expression and cytokine analysis reveals that monocytes and the IL-17 immune response might be important in resistors, and we are still trying to do further analysis to try and tease this out. So I will end by acknowledging, something cut out, all the participants, supposed to be all participants there, and then a welcome trust for the funding. So, and most of the data that I presented was generated by Norma and Abuyonke, who are PhD students in the lab. So please, I don't have them on speed dial, so don't ask difficult questions because I can't get to them. So I will end up there, and then I will thank you again. I'm happy to take any questions.
Yeah, thank you, Muki. Very interesting presentation, so I'm sure you're going to have a lot of questions. Yeah.
I'll
sorry,
start.
DJ is up.
Thank you. Really nice job, beautiful work. A couple of suggestions to help, and you don't need to phone a friend at the bioinformatics place, okay? The CyberSort analyses that you did, the deconvolution analysis, that's a signature matrix of 20 different immune cell phenotypes. So here's the reason I say that. When you look at some of the immunophenotyping that you did with the specific cellular markers, if you go into that CyberSort data and look at the matrix, that will correspond with genes that will be within those networks that you saw. So suggestion number one, it'll give you some level of redundancy and additional discovery. And the second thing you can do, the cytokine analysis you did was quite nice, but there's another package which is called BloodGen3 that we recently started using. That's a similar matrix file where you take your sequencing data, run it through this matrix, and it'll give you cellular populations associated with genes within those, that begin to give you informative information about which pathways are activated. So good job. You don't need to phone a friend.
Thank you. I will come to you after this. Thanks for the suggestion.
Yeah.
Thank you for your talk. I had a question about your whole blood transcriptomic analysis. So the assumption is you took blood from your two groups and then, so basically did RNA-seq or something. But how is that TB specific? Because these people probably have a lot of other things going on. Your quantiferon was PBMCs that were re-stimulated, so that is clearly NTB specific response. But for your whole blood, how do you know that what you're seeing or not seeing is TB specific as opposed to anything else that's going on in these people?
No, that's an interesting question. Yeah, so that is one of the limitations for the analysis. So we try to collect as much data as possible on the comorbidities and everything else, where these people were considered healthy because they were HIV uninfected and didn't have any other signs or symptoms of any disease. So, and most of the factors that we collected were similar between the two groups. So we are sort of convinced that whatever we see might be related to TB, but we can't say that for sure. So because we didn't, especially for the whole exome sequencing, RNA sequencing, as well as the cytokine in plasma. So we can't say for sure that it's related to TB, but because we try to control for the other confounders, and also most of the comorbidities, hypertension, diabetes, and all of those that we collected were similar between the two groups. So yeah, we can't say for sure that it's directly TB, but if the differences are there, it might still be related to TB.
I mean, I'm wondering whether it would be more informative to do RNA-seq on your stimulated PBMCs. So, looking at your TB-specific response in your PBMCs. And then maybe at the whole blood level, have you considered looking at genetic differences? So do you think there may be genetic differences between resistors and people who are latent? I suspect those will be easier to see than transcriptomes that vary based on what is going on in the individual, and probably even with the comorbidities balance, there'll be a lot of background that I worry you won't be able to tease out. So just whether you looked at the genetics and then transcriptome of stimulated PBMCs versus unstimulated to see whether the TV specific response.
Thank you. Yeah, we are currently doing that. So one of the students in the lab is stimulating the PBMCs and either doing bulk sequencing or isolated with med cells, focusing on the med cell, isolated med cells, and also did bulk after stimulation with whole mycobacteria. So that is ongoing. And for the genetics, because I didn't have enough time to present, so we did a whole exome sequencing on the extreme phenotypes. So we've identified some interesting polymorphisms involved in the fatty acid synthesis pathways, as well as platelet activation pathways that we identify them in the extreme groups, but we're now genotyping them in the whole cohort just to see whether that stands out. So yeah, we are doing a big genetic analysis. We've done whole exome sequencing already, but we're currently doing genotyping on the SNPs that we've identified from the whole exome sequencing. Yeah, but that is ongoing.
Okay. Thank you very much, Muki. Thank you, everyone, for your audience.
Okay, so would like to thank our moderators, Dr. Akins and Dr. Aniwe, very much. We are running slightly behind schedule. We have time for a coffee break right now, so I'll invite you to move to the coffee area. But I have one small announcement. If any of you are still holding on to the orange or red lunch coupons, please can you kindly give it to me as you are exiting. Thank you so much. And be back in 15 minutes, please.
Hey, this place. I'm not going to. I'm going to look like person here, Hey, mommy.
All right. So we are about to start the last session before lunch. Well, the last of 2 sessions before lunch. And I'd like to call the chairs for this session our very own Dr. Jerry Jo Harrison, and Dr. Ajwa Padiki Nate. I hope everyone got their coffee, and they will take us through the last of the 2 sessions before lunch. Okay.
Okay, welcome back. Let's get started. It is my honor to invite Jonathan to the podium to discuss importance of lysosomal stress response to the pathogenesis and potential treatment of HIV-associated neurodegenerative disorder.
Thank you very much. Thank you for the invitation to speak. It's a real pleasure. Halfway through the meeting, I've learned so much more about infectious disease. So the title of your institute is Cell Biology. You're going to hear a fair bit of cell biology, more specifically organellar biology, mostly in the context of neurological complications associated with HIV, a condition that isn't so widely known, although 50% of people living with HIV have neurological complications. So I always like to which button? Not that button. Which button? Oh, there. So it's this button, this button. So I always like to start my talks by acknowledging the people that do the work in the lab, as well as the funding sources. 2 particular people I'd like to call your attention to is Kofi Lakpa, an Ivorian who got his PhD with me a few years ago and now is a medical student in Wisconsin. And then your own Darius Kwanzaa, who's finishing his PhD in my lab. And so if you could please clone a few more Dariuses and send them our way, we'd be very appreciative. So endolysosomes, I'll refer to them in composite, endosomes and lysosomes, are acidic organelles and are very important for physiological functions. I'm going to be talking mostly about pathological significance, but clearly they are of physiological importance. We sometimes consider them to be garbage bags. That's why they were called lysosomes. Nobel Prize was given for that. But we oftentimes think of them as stomachs because they are acidic. And you can see that they're fundamentally important in many things, including cell death pathways. So in the context of brain cancer, glioblastoma in particular, most of the cell death pathways are mutated around, and the last one that's standing and that med chemists are coming after right now is lysosomal membrane permeabilization. So, which is why they're specifically targeting the pH of endolysosomes to make the cells more susceptible to temozolomide and other chemotherapeutic agents. So our interests are in aging and age-related neurological disorders, HAND, which is what we're going to be talking about mostly here. We've done brain cancer work, opioid analgesics and other drugs of abuse, and SARS-CoV-2. And so in the context of all these things, we do a number of insults to our cells, to our neural cells, and including in the context of HAND, the HIV proteins, transactor transcription TAT, GP120. The organelles we look at are endolysosomes, mitochondria. You'll hear me probably say that I think cell biologists are way too mitocentric in cell biology, and we like to think that the endolysosomes don't get the respect that they deserve. Oxidative stress and the entire reactive species interactome. A PhD student just defended last week in my lab about the reactive species interactome. And the antiretrovirals themselves. Clearly very helpful, keeping people alive, but they are also have their detractors and can be neurotoxic. So what this has evolved into is really the concept of lysosomal stress response. If you look in the literature for organelle stress responses, there are thousands of papers about endoplasmic reticulum stress. There are thousands of papers on mitochondrial stress. But if you, we did this review 4 years ago, published it 4 years ago, and looking in the literature, there was only about a dozen or 2 dozen papers that even mention lysosomal stress, let alone only a couple of them made any attempt whatsoever in defining what it is they're talking about in terms of a lysosomal stress response. So in the center of that circle, the lysosome, you'll see 9 points that we have thought would be very important. Central amongst all of those is the pH. So these are acidic organelles. They like to be acidic. In the context of neuroscience, de-acidification is a bad thing. Acidification is very neuroprotective. And so you can see that we've done a lot of different things published that nanoparticles de-acidify. LDL cholesterol, which is a model for sporadic Alzheimer's disease, de-acidifies. The HIV proteins, TAT and GP120. The SARS-CoV-2 S1 spike protein de-acidifies. We just published a big paper in Journal Neuroscience about the human endogenous retrovirus K envelope protein, which is implicated in the pathogenesis of ALS. That de-acidifies. Also drugs of abuse, methamphetamine, opioids, alcohol, nicotine, all de-acidify. And then as a positive control, we oftentimes use chloroquine, a drug that I think you're quite familiar with here in this region of Africa. And we finally got smart and realized that as a weak-based drug, this, and as a pharmacologist, our research might have even more implications. I'll talk a little bit about that story. So this is basically what you see. This is HIV TAT protein. It deacidifies the endolysosomes, and it makes the endolysosomes, changes their number, changes their conformation, changes their movement in the cell. Under normal conditions, endolysosomes are perinuclear, but under insults and deacidification, they move to the plasma membrane. So the frequency and the sizes changes, and not only does the pH change immediately upon putting on a basic protein like HIV TAT, it's a polyarginine protein, but it also changes the pH in the long term. So here you're seeing with that bar diagram that after 2 days of putting one treatment of TAT on these neurons, the pH goes up and stays up. The same sort of thing happens with GP120, the coat protein of HIV, showing that the pH changes on the upper left in real time. You can see that the peak response is quite similar to the bifilomycin, which is a positive control. You can see how the shape and change, the endolysosomes, the shape and movement in the cell changes. And then the volume, they clump, they get bigger, and so it's quite typical in the lab to do not only function but also structure, morphology. So then we also, and this is our work and work of others, have shown that these HIV proteins depolarize the mitochondria. Of course, mitochondria are important for cell life and death. And if you look on the left, the A, the mitochondrial membrane potential, it depolarizes over time, 24 hours, 48 hours. And then if you look at reactive oxygen species, that increases as well. If you look at other reactive species, carbonyls, nitrogen, sulfur, those change as well as the oxygen. So we knew enough about reactive oxygen species to know that iron is important. And we've been doing a lot of work with a different divalent cation and the pool of calcium in the endolysosomes. It's a readily releasable pool of calcium. The concentration of calcium in endolysosomes is actually equal to that of the endoplasmic reticulum, and yet very few people talk about that as a readily releasable store of calcium inside of cells. But we also got interested through the reactive species about iron. And iron is very implicated in neurodegenerative diseases, probably the most common of which is Parkinson's disease, most well-known link between iron and neurodegenerative diseases. But it's also important in HIV. There's a vast literature. So iron is required for HIV replication. The iron levels are associated with a faster progression of the infection. Higher levels of iron inversely correlate with survival. So higher iron, decreased survival. And iron chelators have anti-HIV effects and have been used as adjunct therapeutics. And so what we have done is we've done many things with iron at this point. I'm just giving you a very small snapshot of the kinds of things we found. Here we show that GP120 deacidifies the top, and it causes the iron levels to come out of the endolysosomes. So you see a decreased level of iron in the endolysosomes, and that iron pool is sufficient, once released from the endolysosome, to account for the increases in iron that we measure in the cytosol. The same sort of thing is true if you look at a consequence of the iron. So if you look at the reactive oxygen species, not only in the cytosol on the left, mitochondria on the right, you can see that GP120 in this case increases the levels of reactive oxygen species both in the cytosol and in the mitochondria. If you put in the chelator deferoxamine, DFO, by itself, decreases those levels, so is somewhat protected and completely blocks the GP120 induced increases in reactive oxygen species. So there's two reasons why deferoxamine, it's a clinically used drug, why deferoxamine does this. One, it chelates. We know it, we refer to it as a chelator. The other is that it acidifies the endolysosomes, and because of the enzyme STEAP3, it keeps the iron locked up as ferric 3+. The only way iron can get out of the endolysosomes is if it is in the 2+ ferrous state. So deacidification leads to more ferrous and encourages its release out of the endolysosomes. So this brings us to the thing about the weak-base drugs. So as if maybe, already I think probably some of what I've said is of relevance to your lives as scientists. But then it dawned on us as pharmacologists that 75%, we've been using positive controls in all of our experiments, such as chloroquine, very common diproic, weak-base drug. And 75% of drugs on the market are weak-base drugs. They have the physical chemical property of being weak bases. I can talk a lot about why that might be, why that is, but the fact remains that these are the most common weak-base drugs. And so we decided to take a number of weak-base drugs. So in the United States, let me point out, in the United States, we are a drug-taking culture. I don't know about Ghana. People come to see their doctors in the United States. They expect to walk out the door with a prescription. When you're 60 years of age and older in the United States, on 2/3 of the population are on 7+ drugs every day. So if 75% of these drugs are weak-base drugs, that means 2/3 of the population, 60 years of old, or an age of older, are taking 5 or more weak-based drugs. So what do these weak-based drugs do? We took 5 very different drugs. We just published this. We took atropine, anticholinergic, azithromycin antibiotic, fluoxetine antidepressant, metoprolol, beta-adrenergic, beta blocker, tamoxifen estrogen receptor blocker. These are very different drugs, completely different chemicals, completely different mechanisms of action, given for completely different clinical reasons. And what do they have in common? The physical chemical property of being weak-based drugs. And if you look in the literature, in the neuroscience literature, these things kill neurons. Amazingly enough, because many people are on these drugs, but they are toxic. So we did concentration-dependent studies. We picked concentrations that were of therapeutic relevance to humans. So we did the IC50s, EC50s, and we picked concentrations that were relevant to those that are measured every day in human beings. And you can see in the bottom that it not only does it deacidify, but it kicks out iron from the endolysosomes. If you look at the weak-based drug-induced increases in cytosolic iron and in reactive oxygen species, they all increase. That pool is sufficiently large once it's kicked out of the endolysosomes to increase the levels in the cytosol. The reactive oxygen species, all of that is blocked by pretreatment with deferoxamine. It also does the same thing if you look at the mitochondria. The drug-induced increases in mitochondrial iron and reactive oxygen species. It all occurs with all of those weak-based drugs. All of those responses are blocked with deferoxamine. And then if you look at the consequence on the mitochondria, clearly mitochondria are important. It's kind of an end game thing for cell survival. If they depolarize, that's a bad thing. The mitochondrial permeability transition pore opens up. And because mitochondria are not part of us, they have many things inside of them that the cytosol never wants to see. And when the mitochondrial membrane, the permeability transition pore opens up, these toxic apoptosis-inducing factors come out, and it kills neurons. So here you're seeing mitochondrial membrane potential. Yes, these responses to these clinically used drugs is sufficient to cause depolarization of the mitochondrial membrane potential, and ultimately it causes cell death. And all of that is if you control that upstream pool of iron coming out of the endosomes and lysosomes, you protect against all of those toxic reactions. So we think that the model is quite simple to view here. These weak-based drugs and many other insults, in some cases they get stuck in endolysosomes. Hydroxychloroquine, for example, the half-life of hydroxychloroquine in endolysosomes is 70 days, 70, makes pharmacokinetics very difficult. So these weak-based drugs get into and get stuck in and deacidify the endolysosomes, kicks out iron. That iron is sufficient to cause all kinds of other problems in the cytosol and in the mitochondria, up to and including causing cell death. And so this, of course, has relevance to HIV as well, because a lot of the drugs that are used, this is from about 10 years ago now, so the drugs have changed, but our work has been confirmed and extended by some of our colleague friends around the world. And what we found is that a couple of things that this slide illustrates. One is that the antiretrovirals that are weak-based drugs cause these kinds of changes in the endolysosomes, but not the ones that are acidic. And this has been confirmed and extended with the newer antiretrovirals. In this case, what we're seeing here is another thing that's very interesting in terms of the pandemic, and that is that the neurological complications have changed over the 30 years or so. And that is that early on in the pandemic, the neurological complications were more serious, more Parkinsonian. The virus stayed and congregated more in the basal ganglia. But now it's more Alzheimer's-ish. It's more hippocampal, cortical, and the symptoms are milder. And so mechanistically, what we showed in this is that the HIV TAT deacidifies, and the BACE1 enzyme, which is the rate-limiting step for misfolding amyloid beta protein, it causes an increase in the amyloidogenesis. And that's an endosomal enzyme, whereas the gamma secretase, the second enzyme in the scheme, is a lysosomal enzyme. So this helps mechanistically understand why the neurological complications have changed over the 30 years or so. And so ultimately, I'm just going to leave you with 3 thoughts. And hopefully the sketch artist can summarizes all these things in these 3 thoughts. And that is that endolysosomes can contain readily releasable stores of divalent cations: calcium, iron, zinc, copper. Less has been done with zinc and copper. We're pretty focused now on iron. That the iron released from the endolysosomes accumulates in the cytosol mitochondria and changes the reactive species interactome. And we think this is a really important point for cell biologists. It's not that we love mitochondria as much as the next cell biology lab, but we are now fully convinced that the endolysosomes are upstream in virtually every way that we can think of, upstream of what's going on in the mitochondria, and that might help explain why virtually every trial that's been invented for any disease in human beings has failed miserably when mitochondrial reactive species have been targeted, whereas in every cell culture model, every animal model, those same strategies are incredibly protective. And we think that's because of this reverberation and the crosstalk between organelles. And so ultimately, that's what I'd like to my talk. And yes, there are many questions you could ask about this photograph. Thank you.
Okay, so we'll take questions.
Yes, you did. Very quick question. Something like rapamycin, right, which inhibits mTOR. Have you used any of those compounds to see? I'm trying to figure out what the mechanism is, right? I understand Fe2+ is leaving and creating reactive oxygen species. Is this related to autophagy pathways? Is that what's happening? So when iron gets out, it in and of itself can be toxic, but is it the iron in heme units for critical enzymatic function that's causing this problem, or is it just the free iron per se?
Right. So great question. The way I'd like to answer that is that the greater lysosomal system, including autophagy, including extracellular vesicles coming from multivesicular bodies, many of the same compounds that people use as positive controls and insults affect those processes as well. So almost certainly that all these things will affect the greater lysosomal system, including autophagy and autophagosomes. Clearly very important. We've done a little bit over the years with autophagy, but we have stayed out of that field because we think it was hard enough to do the iron story.
Hello. Yeah, also really interesting. I work on autophagy. But my question was more about, so the lysosomal pH is maintained by the VATPAs, right? And so there are direct inhibitors of that. But what I'm really interested in is if you have these
A little louder, please.
if you have these weak bases that are getting, it's really weird talking when you can see your face on the screen. I don't know if anyone else is finding that. But so these weak bases, when they get into the lysosome, clearly the VATPase must still be pumping protons in to the lysosome when that's happening. So how is the calcium and the iron getting out when those weak bases are interacting with those protons that are being pumped into the lysosome?
Right, so excellent question. And we've done a lot of that. We've done a lot of characterization of the channels and transporters through which calcium and iron exit and under what conditions. The thing to keep in mind, though, is that the thing about weak base drugs is that they, and the basic proteins that we've done as well, they get into and get stuck in the endolysosomes in that acidic environment. So it's not just an immediate deacidification, it's a longer term deacidification. There are probably multiple reasons for that. And so it's, even though, so the other thing I'd like to point out is that even though all these different insults deacidify, the ones that I've mentioned, they don't necessarily deacidify the same way, the same mechanism. So for example, some of these things interact with, like the S1 protein, actually binds to and affects subunits of the vacuolar ATPase, the main hydroton pumps. Some of them are affecting second messenger systems. There's many different things that are going on and many channels that are activated. But in this short period of time, I just didn't have time to go into that kind of story. Thank you.
We'll have one last question.
Thank you, Jonathan. Very intriguing talk. I'm just, you mentioned S1 protein. I'm just thinking about GP120. What kind of mechanism is it? Because TAT I get, but how is GP120?
Right, so my colleague with the S1 protein really showed that it's subunits of the vacuolar ATPase that's being affected. We didn't, so our stories and our targets and the molecular mechanisms, we don't do the exact same thing with the exact same, with yet another insult. We think that'd be rather a boring way to approach science. So our targets evolve with the story, and so we don't know, for example, we haven't specifically tested whether GP120 affects, has the same effect mechanistically as, for example, the S1 protein of the SARS-CoV-2.
All right, thank you very much. We'll hand over to Peter. Okay, so the next session is table talks. Okay, please, when you see your slide up, you can come give us a talk. Same as yesterday.
Is Aisha here? Aisha?
Okay, can we move on to the next slide? Previous slide, yes, please.
Okay, thank you. My name is Benedict, and my work focuses on examining the effect of antibiotics on the production of cytokines in breast and prostate cancer cells. So, breast and prostate cancer cells remains a global health challenge, mainly because therapies have failed to achieve have failed to be effective in these patients. One of the mechanisms include inflammation, which drives the progression of the cancer and the resistance to therapy. So my work focuses on this inflammation. Cytokines are released by the cancer cells and other immune cells, and then they make the tumor microenvironment suppressive. So this leads to the resistance of cancer therapies, and then influence proliferation, migration, and angiogenesis of the cancer cells. So approximately 50 to 60% of cancer patients, cancer deaths are related to infections. And antibiotics plays a crucial role in the management of these patients. So studies have shown that antibiotics can impact the progression of tumor and then also impact the efficacy of therapy. So it can either enhance the therapy or reduce the efficacy of the therapy. So my work focuses on how these antibiotics affect the production of cytokines. So the question is, will it be a positive or negative impact? So I'll be on poster 38 tomorrow, and I'll be looking forward to you to discuss my results. Thank you.
Please, next.
Yeah, please, next one. Once you see your poster, you rush up stage so we can have you talk to us. Okay.
Dora.
Good afternoon. My name is Dora Hokomensa, and my work focuses on purification and characterization of IgG antibodies against the spike and nucleocapsid proteins of SARS-CoV-2 utilizing ELISA. So since the outbreak of COVID-19, or since the outbreak of SARS-CoV-2, we've had over 700 million COVID-19 cases, and out of the 700 million COVID-19 cases, we have over 600 million recovered cases. And we asked ourselves, what do these recovered individuals or convalescent individuals have? And we all have IgGs or antibodies against the spike and nucleocapsid proteins of SARS-CoV-2. So we asked ourselves, what do we know about these antibodies? And as you can see, I have some questions on the slide. Stuff we like, which of the 4 proteins of SARS-CoV-2 induces the most immune response? Like, do these antibodies have neutralizing effects against the different strains of SARS-CoV-2? And which of the subclasses of IgGs is responsible or is produced in response to the virus? So, and most studies, once they work on SARS-CoV-2, they actually deal with plasma and serum samples. But for my work, or for my method, I first purified IgGs or antibodies from convalescent patients before I did my assay. And I had some pretty interesting results, as you can see there. So kindly pass by poster number 98 to learn more about this work. Thank you.
Thank you. Apparently, some of you don't recognize your slides, so we have to mention names. Harriet.
Okay, good afternoon. I'm Harriet Kamandeku, and my poster presentation is on the functional characterization of gametocyte-specific genes of Plasmodium falciparum as a novel candidate for transmission-blocking vaccines. So malaria has been a major public health concern in Ghana, and this occurs through the transmission of the Plasmodium parasite to the human body, which affect the human body system and then cause clinical symptoms, which leads to high rates of morbidity and mortality in Ghana or in the world or sub-Saharan Africa. So there is a need for intervention tools, and some of these intervention tools that have been produced against malaria are the RDTs and then the drugs, but this Plasmodium falciparum parasite have developed resistance towards this intervention tools. Hence, there is a need for a vaccine production. Researchers have worked on this, and then there have been a number of vaccines in the pipeline that are undergoing clinical trial. But then, these vaccines are about 30% and 70% effective. Hence, we need a vaccine that is fully effective and long-lasting. Therefore, this is the aim of my study. The Plasmodium falciparum genome has a large number of protein coding genes which could be investigated. But out of the 5,300 to 5,400 protein coding genes, only 30% of these genes have been investigated so far. So there's a need to explore more on these genes so that we can see if we can get an antigen that could give us a more potential or a vaccine that will be fully effective. So we identify some selected genes based on surface localizations and uniform expression systems. So to know more about what we've found so far, please come to poster number 12. Come to poster number 12 to learn more. Thank you.
Jessica.
Good day, everyone. It's a pleasure to see you all. My title is missing, but it's there. I don't know. Well, do you know that by 2050, an alarming 10 million lives could be lost if nothing is done about antimicrobial resistance? We certainly do not want that to happen. So in our study, we utilized in silico methods to identify and understand genes that cause resistance to antibiotics in Salmonella, and we discovered a vast array. Remember that these tiny genetic warriors are not easy to fight, but together we can defeat them. Say together we can defeat them. Together we can defeat them. So meet me tomorrow at poster number 34. It's on the second floor. Thank you.
Mabel.
Hello, everyone. I am Mabel Apiej, and I'm here to briefly present my work titled Establishing a Breast Cancer Cell Line Stably Expressing microRNA 4725 and Validating its Putative Target. So breast cancer is the #1 cancer among women worldwide. And then microRNA, which is a single-stranded non-coding RNA that regulates genes, have been found to be implicated in breast cancer. So we employ this workflow, that is cell culturing, transduction, pyrimycin treatment, and gene expression, to establish a breast cancer cell line that stably expresses this microRNA, and we determine the role that this microRNA plays in some putative target genes in breast cancer cell lines. And amazingly, we found that most of these putative target genes were upregulated by this microRNA. For more detailed discussion, please come to me. I'll be right by poster 127. Thank you.
Hello, my name is Magdalene Sarah Ofori, and I'm sure most of you have heard of the ongoing dengue outbreak that we're experiencing as a country. But did you know that in 2022 and 2023, we detected some sporadic cases of dengue fever? Now, the thing about the virus that causes dengue fever is that it has 4 distinct serotypes. An infection with one serotype does not confer protective immunity against another serotype. And spoiler alert, we have 3 serotypes circulating in Ghana as of last year. So if you want to find out about the circulating serotypes we have and then the strains that we detected, you can see me at poster number 110. Thank you.
So just by way of announcement, the even numbered posters are going to be up tomorrow. And so just get ready and visit the various places to have an interaction with the presenters. Thank you.
Good morning, everyone. Good afternoon, everyone, sorry. Let me make this taller, good. I'm Kwamina, and I sought to solve a problem with surveillance of zoonotic diseases and also figure out how we can get hold of those pathogens that are not common. Because of that, nobody is concentrating on them. So to do that, I proposed a One Health molecular approach in what I consider the best human-animal interface, which is captured beautifully by Corinth, one of my favorite impressionists. And then, as a downstream analysis, I did AMR. So if you came, I'll share this approach with you, my results with you, and then in return, you will help me see if it's economically feasible, it's practical, and then if we can adopt it as a preliminary study for nearly every zoonotic study. Thank you very much.
Thank you. Do you recognize your... Okay, I think...
Hi everyone, my name is Josiah, and the poster you see is on a malaria immunology and vaccinology study. And here we inform on the protein sequence, structure, and antibody profile of the apical membrane antigen 1 of 3 Plasmodium species. So in this study, as you have heard time and again, malaria remains a disease of global health concern, and various intervention tools are put in place to help curtail this issue. And under the auspices of vaccinology, this study, the underlying hypothesis of this study is that various antigens or various epitopes are conserved across evolution with respect to Plasmodium species. Various epitopes are conserved across evolution in sequence and in structure across and are shared in antigens of several species. And in this light, in this study, we try to compare the sequence and structure of 3 Plasmodium species, and we do a cross-reactivity test with functional antibody testing and provide information on the antibody profile with 3 or 4 cohorts, with several cohorts, plasma obtained from several cohorts across the country. Your inquisition and your possible contributions are welcomed at poster number 2 tomorrow and Friday. Good afternoon.
Samson Kafui, right? Are you here? Not here. Next. Okay, I think that'll be all. All right, so a recap of yesterday's table talks. 1 minute each, quick, and then we can go take lunch. And please, pass by the posters. There are very incredible posters over there. I have taken a look at many of them. I'm sure you will learn quite a few things and be able to get some few collaborators in the process. So please, take some time and visit the posters. They're an integral part of the program, and I'm sure you enjoy interacting with the presenters. I mean, as you can already see, many of them are very interactive, and it's a joy to be with them. So please visit them. Timothy, are you here? Timothy is not here, so next one. Ernest Afari, present, out, or absent? All right. Next. Poster number 5. So take note of the posters, the ones that you might be interested in. If you recognize your poster, you come up here. Right.
Okay. Good afternoon, everyone. Okay. So my name is Comfort, and the title of my study is Unveiling the Immunodominant Portions of PF-CELI1. So actually, PF-CELI1 is a new antigen, and it
Shall we go to the next one?
Hi, I'm Precious Kumi. My work focused on looking at the longevity of booster immunity. So what I did was I measured T-cell and antibody responses to two COVID-19 vaccines that we had used as boosters. So if you want to know more about what we did with respect to the longevity of the boosters that you might have received during the pandemic, please come to poster number 3. Thank you.
All right.
Okay, so I guess since there are no more presenters, it's time for lunch then. I'm hungry. Thank you.
All right, Peter, take it over.
Thank you very much, Ajwa and Jerry. Let's give them a round. Before you file out, some of you may have received an email about scoring oral presentations. I hope everyone logs on to that Google form and scores that. And before the day is out, everybody is going to get for the posters. So please, as many scores as possible would be really beneficial. Thank you. All right, bye.
Is it on? Okay. All right. Welcome back from lunch. I hope everyone had some food. Good. All right. So we're going to get going here. We are falling behind time again. So we're having our second keynote speaker for the day. You'll notice that we had to do a swap. Janelle was supposed to present now, but Nikes, who is going to come now, was supposed to have been, or he was scheduled for Friday, but due to his flight arrangements, he has to leave tomorrow. So Janelle kindly agreed to swap with him. So we're having Nikes right now. So it's my pleasure to introduce Professor Nikes Indam, who is a close friend of ours and a colleague. Nikes is actually a pioneering director for IRD. IRD is the French International Research and Development Agency, right? Did I get it correct? Anyway, he'll tell you, he'll give you the correct name, but we know it as IRD. Is a research partnership that is funded by the French government. Nikes was the first to open an office for IRD here at the University of Ghana. He was based at Noguchi for a few years, and now he's moved to Benin, where he is basically the regional director for Benin, Nigeria, Togo, and Ghana. And he continues to work with us and co-supervise students with us and write grants with us and so on and so forth. And he's really passionate about what we do here at WACCBIP and the capacity building efforts that we are doing here, and he's always been supportive. His research, like Lars mentioned, is also along the lines of Lars, and he and Lars have done a lot of great work together. And one of his key contributions was in characterizing the role of VAR2 CSA that Lars mentioned as a key determinant of immunity to placental malaria, and subsequently now a key vaccine candidate. So Nikes will tell us more about this as we welcome him. So Nikes, please take over.
Thank you, Gordon. Thank you, everyone. I'm very pleased to be here. I'm honored to be here to participate in this celebration, the 10th anniversary of WACCBIP. WACCBIP is like a family to me, and I've been working with you guys for a few years, and I experienced Gordon's leadership, particularly in bringing together the ACEs within the WANIDA network. And yeah, so Gordon, I'm happy having you as a friend and as a colleague. And I hope we will continue together to find solutions and build capacity for the next generation research in the WACCBIP in Africa, because your leadership goes beyond Ghana, and in the Western Africa. So many people know what you've been doing, and even at IRD. So I have to extend the greetings of Valérie Verdier, the CIO of IRD, to you. Okay, so you introduced me as the regional director of IRD, for Ghana, Nigeria, Togo, and Benin. And I'm based in Benin. Just it's an administrative function to me to facilitate IRD activities within the sub-region. And by profession, I'm a director of research and a professor of parasitology at Université Paris-Cité. And I am in Benin for a few months before I go back to Paris to take over my department of international health, mother and child. So my research interest has been around this placenta malaria story. As the title clearly indicates, yeah, and as also Lars said this morning, we failed in developing an efficacious vaccine. Even this would have resulted in almost 1 million low birth weight occurrence. And we know about the consequences of low birth weight. So it's an important risk factor for neonatal mortality and morbidity. And so it's of major public health concern, combating placenta malaria. And the prevention of these conditions relies on the use of IPT-PSP. So it's not an optimal drug. It's monthly given, it's monthly administered, but only from the second trimester of pregnancy because of safety. And it's also practical to start on the second trimester of pregnancy because we never see women visiting ANC before the second trimester of pregnancy. So the other complement of this tool is the use of impregnated net, mosquito net. And these has proved to not work very well to prevent placenta malaria or malaria during pregnancy. And based on the epidemiological and surveys in many countries, it's like the coverage is still very low, and we definitely need a vaccine that can cover, especially the first trimester of pregnancy, to offer a better protection and reduce the occurrence of low birth weight and save many lives. So we go back to this parasite life cycle, just to mention that there have been some stages, vulnerable stages, where vaccine could be developed. So the strategy for developing vaccine were identified. So targeting the liver stage, and a couple of antigens have been published, and one of the most famous now is the CSP. And we know that there are R31 and RTSS that were pre-qualified recently by the WHO and now being deployed in many countries. And the other strategy will be to target the blood stages. And some leading candidates here are the RH5 that we know, and the EMP1 that Lars approached this morning. So that can be used to prevent tissue tropism. So it's a kind of disease-preventing strategy vaccine. And those vaccines seem to be similar in terms of efficacy, even though the initial efficacy was pretty low, about 40%. When they are combined to other prevention tools or other control tools, they can prevent around 70% of malaria episodes. And this was described in there when they were combined with SMC strategies in Western Africa. So these vaccines are destined to be given to children, and children who were vaccinated were followed above even after the intervention was stopped. And what was observed is that there is still a significant burden of infections after the intervention. So when we stop vaccinating, so after the age of 5, we still have a significant burden of parasitemia in the population. So this vaccine cannot prevent placenta malaria because this comes later. So anti-disease vaccines are still needed. There is still a room to develop and complement this basis vaccine strategy. Tissue tropism is the key of malaria pathogenesis. And as Lars showed this morning, it is mediated by PFEMP1 parasite. So they have the capacity to bind to several tissue receptors and lead to sequestration, parasite sequestration. And depending on the organ, this can be fatal. This can complicate the malaria and completely change their clinical presentations. So the PFE and P1 are expressed in a mutually exclusive manner. We have like 60 copies per genome, haploid genome, and only one is seem to be expressed at a time. So when parasite has depleted, so when we knock down this gene, family genes within the genome, it's like we significantly decrease the recognition, so the amount of antibodies that recognize the surface of infected erythrocytes. So it is an important target of acquired immunity. So it plays a major role, and if we can target this, there is a possibility to limit the disease. So the parasite can switch, as I was saying, and in some diseases like placenta malaria, we now have a clear picture of which type of PFE and P1 is expressed. So this is the VAR2-CSA. And in the genome, the PFE and P1 differ among genomes. So when we see that we have 60 copies per haploid genome, and it doesn't mean that in a different genome we have the same 60 copies. So there is a great polymorphism that makes it very difficult to identify a protective target or protective correlate with these antigens. So in this graph, we can see like many epidemiological studies indicate that the malaria problem is basically in young children. So there is like a gradual acquisition of immunity, a kind of immunity over time without any intervention. And we can see here that most of the severe cases are associated with placenta malaria, with cerebral malaria or complicated malaria in children occurs here. And those parasites that can bind to specific receptors like protein C endothelial receptor, like let's say an ICAM1, seem to be more specific. And gradually there is an acquisition of immunity to those type of PFE and P1 until the adulthood when women become pregnant. And when she becomes pregnant, because of this source of asymptomatic infections, so they become susceptible to a new form of malaria with a particular phenotype of parasite binding to the placenta. So yeah, we have those parasites that can accumulate in the placenta and affect the functions of the placenta, resulting in essentially the growth intrauterine growth retardation, leading to low birth weight. So the mechanism is now well known that those parasites bind specifically to CSA. So the VAR2-CSA, the ligand of this, the ligand that the parasite expressed to bind this receptor has been clearly identified, and it was initially demonstrated by Eli Celenti in Copenhagen when he was playing with 3D7 parasites, and that was selected to bind to CSA, and he found that one of the PFE and P1, or one of the VAR genes member, was specifically upregulated when the parasite was selected to bind. And this was a combination of work from what Lars initiated in Copenhagen. And at the same time, I was working I was doing my PhD at the same time with Eli Celenti. And I went to Senegal collecting parasites from the placenta and characterizing them. They were specifically binding to CSA, as Durfee has published initially. And we tried to look at the transcription profile. It was very difficult to define a primer that would target those. I tried to use the primers of Celenti early developing in Copenhagen, but they were not amplifying anything because of the polymorphism of VAR2-CSA. So it took us like a year to accumulate different variants of VAR2-CSA, identify some conserved region, and develop and design some kind of universal primers that we use, and then saw that the VAR2-CSA was clearly upregulated in those parasites. So this contributed to confirm the importance of VAR2-CSA in driving placenta malaria. So from there, we also conducted several studies, and in Benin, a number of a cohort study, and a particular one was this one. So we recruited women who were with the intention of becoming pregnant. So, and follow them until pregnancy and throughout the pregnancy, and characterize their sample, the sequential sample that we were collecting. We characterized that a posteriori. So they work hard. We recruited like 300 women, and 100 of them became pregnant. It was very, very important, 30% working hard. And when we characterized those parasites, we confirmed the hypothesis that much of the infections occurring during the first trimester of pregnancy were not novel infections. They were infections that were already present before pregnancy, meaning that, yeah, those invisible minority population could simply take advantage of the placenta to propagate. So it wasn't linked to new infection. It's not because there was no relationship with the seasonality. So the evil was already in. So that's why using mosquito nets couldn't help, especially in the first trimester of pregnancy. So from here, we understood that more than ever, there was a need to develop a novel strategy to prevent these infections in first trimester of pregnancy. And in the same study, we also showed that infections in the first trimester of pregnancy were also having the same impact on pregnancy outcomes. So placenta tropism can start very early. And when we characterize those parasites, binding to CSA were occur very, very early, as from the 7th week of pregnancy. So it's an early thing. So when we also set up another cohort and follow the pregnant women of various gravidity, what we noticed was that there was a rapid acquisition of cross-reactive antibodies to the VAR2-CSA. So as from the second pregnancy, women already had a high level of those antibodies, similar to what Michaela Fried and Patrick Duffy published early in 1998, when they collected parasites from different geographical areas, like Kenya, Malawi, and Tanzania, and they collected serum sample from different geographical areas and used them on Kenyan parasites. And they were able to show that as from the second pregnancy, women already have acquired those antibodies with the ability to interrupt binding to CSA. So this opened the way to try and understand how VAR2-CSA could be used to develop and induce this kind of immunity. And then the concept for the VAR2-CSA vaccine came in to try and reproduce this acquired immunity that prevent multigravity of having placental tropism. So the idea would be to use the VAR2-CSA and induce specific antibodies that would interrupt placental tropism, and then send the infected erythrocyte to the spleen where they will be filtered. So the rationale was very clear. We based the rationale on this, that if we are able to induce this type of antibodies, so the interaction, blocking the interaction, the neutralizing antibodies, so the job will be terminated in the spleen. And VAR2-CSA is, of course, a very large molecule, more than 350 kilodalton. So developing a vaccine with such a large molecule was not technically possible at that time. So we had to imagine how to cut the VAR2-CSA and identify domains that concentrate the inhibitory epitopes. The structure was not there, and the initial work was to truncate the VAR2-CSA and then we did that with, at the same time with, I don't know if this is showing something completely different. Okay. We truncated the VAR2 CSA, and in my lab we were using the genetic immunization. And in Copenhagen, Ellie and the team was using, they were making outdoor recombinant protein with those small small constructs. And we came to the same conclusion that the N-terminal part of VAR2 CSA was the fragment containing or concentrating the essential inhibitory epitopes. So we could shorten it and then find a minimal binding region. And this was later confirmed by the structural observation that the CSA bind in this N-terminal close to the DBL2. So if we cut like just 2 domains, we can make it, and this is feasible technically. So from this, 2 vaccine candidates were developed. And the PAMVAC, together with the University of Copenhagen, so this was our construct, the minimal binding domain, and the PRIMVAC by our colleagues from INSERM in France, who propose a longer segment from DBL1 to DBL2. So all these constructs could efficiently bind to CSA with an affinity close to the full-length protein. So both vaccines have been tested in 2 different clinical trials in Europe and in Africa. So the phase 2 trial, 1A and the phase 1 trial, A and B design was similar. So the randomized, double-blind, placebo-controlled, dose escalating with 3 intramuscular vaccination. It was the same, and you can see the detail on this clinical trial identifier. So the results that we got was that the formulations were safe. So that's the first thing we look at. So there was no adverse effect with the 3 different formulations that we use. With this formulation, GLASA, driving a high antibody response. And this also was able to induce functional response. So functional here in terms of interrupting binding to CSA. So we were happy that we're almost there until we try to inhibit heterologous parasite and then realize that we failed, we couldn't. So it was disappointing because the rationale was clear enough that if you identify the binding region and you have the structural function evidence, you should normally induce antibodies that could interrupt all binding to CSA, independent of the variabilities out of the binding pockets. So we were very, very disappointed. We couldn't understand. And this was like, we need to reevaluate our vaccine approach based on VAR2 CSA. And how? So the first thing was to go back to the structure and look at the structure. So the cryo-EM structures. This is the structure where we see that it's like a 7 in number. And that isolates a core protein and a kind of arm. And this arm is composed of the 2 DBL, DBL5 and DBL6. So probably these are not interfering with the binding. So if we concentrate the effort on this, we probably could make something more powerful. So now this is the schematic where you see. And when we look at the binding pocket with the CSA, there is 2 channels of binding. So this major binding pocket between the DBL1 and DBL2, and a minor binding pocket here, just because it's a putative binding pocket, just a monomer of CSA bind to this, just after the DBL2. So we were like, no, we normally have this. in the minimum binary domain. So maybe the conformation is different, because here in the conformation we have the DBL4 coming close to the DBL1 to form a kind of pocket. Maybe we need to now reevaluate and try to reproduce this kind of conformation of construct. So several efforts. I'm sorry that you cannot see it here. So several efforts were now initiated to design parasites, to design VAR2CSA, different construct of a VAR2CSA. It's a pity. So different constructs were made, like, you can see this. Me too, I cannot see. Let's go back to this. So trying to design this one, the DBL1, DBL2, and remove the immunodominant domains like DBL3, cutting the arms too, and trying to bring this, keeping the DBL4, trying to form this. So several constructs were prepared based on this, and we also tried to remove the ID1 and induce antibodies in small animals, in rats, and test them. So these are the results that were recently published in PLOS Pathology this year. And the results clearly show that 3 of the constructs, so the construct engaged in the major and the minor binding domain, so the DBL1 and DBL2, DBL4, without the DBL3, induce functional antibodies with properties similar to the full length. And we also have the same thing with another construct, which is the major and minor domain here. Removing the ID1 was like completely disturbing the structure, as we couldn't get the functional antibodies just by removing the ID1. And it was surprising. We said, okay, let's keep it that way, and let's go and test it. Now that we have several constructs, we driving high inhibitory antibodies, high-level inhibitory antibodies similar to full length. And then the result was like what we got with the minimal binding domain. So strong inhibitory capacity, but no strength transcendent again. So the problem is not on the structure. So we need to think about other things. That's where we had to go back to the polymorphism, the sequence, the antigen variation, and try to understand what is the message behind. Why do the parasite modify some of the antigens or some of the nucleotides that are not in the binding pocket? How can this affect the conformation? And the first thing was to reanalyze our sequence from placental parasite collected in Malawi and Benin. And the first thing was that VAR2CSA polymorphism seemed to be linked to variants. So some of the variants were causing much damage, low base weight. And in this study it was the 3D7 variant that were associated with an increased risk of low base weight. Maybe this is a message that we should consider in developing a vaccine. So the most important one was this observation. When we sequence the N-terminal part of the VAR2CSA from NTS to the DBL and reanalyze them, we identified 3 different clades of the VAR2CSA variants. So the 3D7, the FCR3, and a new clade that was striking because they were only found in multigravity, who already had immunity to VAR2CSA. So the idea was, how is this possible? So these variants were representing 5 to 20% of the isolates, depending on the study. So it's like these clades, variants belonging to these threads, preferentially found in multigravidae have the capacity to divert or to escape the immunity to other variants. So they already have immunity. They are in the second, third trimester of pregnancy, and they can, when they are infected with these variants, they have the same poor pregnancy outcomes: high parasitemia, low birth weight. So there's probably a need to understand this. So these variants are here, and probably we will need to think about adding this in the vaccine development. So polymorphism is probably of something in the redefinition of the antigens. So this is just to show you how the ID1 of this new cluster differ from the other ID1, 3D7 and FCR3. And you remember in the structure, when we remove the ID1, we are not able to induce functional antibodies. So ID1 is probably play a major role, a very important role in the antigenicity of VAR2-CSA. So now, how can we consider this? We define 3 different clades with a novel strategy, like to combine these 3 different clades, a representative of the 3D7 of FCR3 and the WR80 that preferentially infect multigravidae, and trying to combine them together to make a multi-allele vaccine. So these are the 3 strategies that we are now focusing on in the lab. And using the capsid-like particle, also to consider the observation that Lars presented this morning, that maybe the presentation of the antigens on the surface of something might drive some effector properties that we were not able to have with the soluble proteins. So this is the way forward. So the second generation that we are now trying to push forward will be based on this. And we know that capsid-like proteins, capsid-like combination is also associated with high antibodies induction. So the preliminary result that we get, because when we prepared this capsid-like proteins, I sent it to Justin and Patrick Duffy in their lab to test. And we observed for the first time this cross-inhibitory using several parasites having different backgrounds, VAR2-CSA backgrounds. So the antibodies could block them efficiently, more efficiently when we only use one variant like the 3D7 FCR3 or the 2053. So now we are working on this, trying to test different combinations, 2, 3, or sequential vaccination with the different constructs. And the way, another strategy based on this observation is that if the parasite persists from before pregnancy, throughout pregnancy, probably if there is a strategy to vaccinate before pregnancy, we should also think about maybe a multi-stage approach that can play on this parasite before pregnancy, before they get into pregnancy, where placenta will select a different variant, and then the VAR2-CSA will come out and knock this tissue tropism. So we are also involved in a project of developing a multi-stage malaria vaccine with the NIH and the Serum Institute of India. We're now working on these 2 candidates, the PFS 230 and R21, to see if this vaccine that would be normally administered to adults could serve as a basis also to add other blood stage antigens on this, like the VAR2-CSA. So, in summary, it's not a success story, but we think and we believe that the immunogenicity of VAR2-CSA can be improved, maybe by combining a small number of variants, like maybe 2 or 3 variants, and considering also what Lars showed this morning, to use this and to develop a strategy that can also consider post-translational modification like the FCF cosylation, maybe by presenting the parasite on the surface of a capsule-like virus that can then, though we definitely need to induce antibodies with these properties, neutralizing and opsonizing properties. So the vaccine development should be guided by basic research, like what we show here, using the structure to redefine, but there is also an implemented by European vaccine initiatives. And Horizon Europe is also funding the translation of this into a GMP manufacturing and to move it into clinical trial. So this project has been extended to 2025, because and this one would probably go beyond the 2027. And I want to thank all my colleagues. These are people from my team. So Femin is our parasite culture expert. So she has been able to grow like a dozen of parasites from the different clades and have a stable CSA binding phenotype of those parasites. And yeah, all these guys contributed in protein expressions, and Nadine played a very critical role in field studies in Benin. And I also need to acknowledge these friends, Heli Salanti, the pioneer, Morton, we are together in the vaccine trial, and last, in the developing this new generation of R2CSA. Patrick Duffy and Justin, who did his PhD with me and who is now based in Patrick Duffy Lab. So thank you very much, and I will end by inviting you to Cotonou to this upcoming meeting in December. Thank you very much.
Okay, you can stand here for the questions. Oh. All right. Thank you, Nikes. Yeah. Peter, are you looking for the microphone? Okay.
Thank you, Nikes. I think this was quite interesting and it was a great talk. As a non-malaria specialist, just thinking about some of the talks yesterday, Malari or Valley, if within falciparum you have this level of variability affecting your vaccine responses, what do you think that sort of, if you think in a, let's say, post-falciparum world, how effective would these be in those other strains, other variants?
Yeah, that's an interesting question. Thank you very much. So the question is also for, not for the VAR2CSA, for all malaria vaccine targeting falciparum, right? Like the R21 and RTSS, so which is being deployed in the field now. Those vaccines are specifically targeting falciparum, and we know why, because falciparum is the species causing the much mortality and morbidity. So this was a priority. And malaria and ovale, so the vaccine are not supposed to target them, but we should take it like something that will fit in a toolbox. So vaccine, if we target the falciparum with the vaccine, and we should also consider putting it in a context that there are other strategies that will control non-falciparum. Otherwise, we'll open the way to the malaria or ovale to let them jubilate, as Yao was saying yesterday. Yeah. So yeah, this is specifically targeting falciparum. Yeah. So the CRAMER is just the strategy that Serum is promoting now. So they just want to use it to fuse the CSP. So it's a protein. They just want to use it to fuse it on their system. Yeah. Thank you.
Yes, Collins.
Yeah. I think that would be the most easiest way for us to do. But the problem at the end is the cost. Because if you want to develop as individual antigens, you will spend the same amount 3 times. Or if you combine them and you are able to purify as a single molecule or a single combination, you will just pay 1/3 of the amount to develop your vaccine. It's purely economic.
Yeah. Okay, last one. Daniel.
Yes, thank you for the presentation. So my question is about the paper you presented about the persistent infection. Would you call them the asymptomatic ones, or why do you choose the word persistent?
Did you hear? Yeah. So the question is whether the persistent were asymptomatic. Initially, they were asymptomatic because they were there before pregnancy. And women were not sick. So they didn't seek for any treatment. They were feeling okay. There was no need for them to seek for treatment until they become pregnant. And then they select in this reservoir parasite with a different phenotype that will bind to the placenta, propagate, and outgrow the other one and become symptomatic.
All right. So many thanks to Nikes for a very interesting talk. We, those who work on malaria, we apologize for always giving you not so exciting news when it comes to vaccine development. But Lars has already told us that he would resolve this before he retires, so we're waiting. All right. So I'll hand over to Peter for the next session. Thank you very much. Yeah.
Thank you, Gordon. Thank you, Nikes, for a great talk once again. Without much ado, I'll call up the next chairs, Dr. Lily Pemka and Dr. Kwabna Dankwa. And they will be taking us for human genetics and non-communicable diseases.
Going about human genetics and non-communicable diseases. We'll have the 12th plenary talk by Stephen Amoa, and it's going to be on brain region and sex-specific changes of circular RNA, SEC-HOMA1 in prenatal alcohol exposure mice.
Right. Thank you. Well, I think so. We'll switch a bit. We'll now move from the infectious diseases to brain and some, I believe, equally interesting fields. So we're looking at the brain region and sex-specific changes in circular RNA, SEC-HOMA1 in prenatal alcohol exposure. So the previous, our keynote speaker spoke of the placenta, telling us that you could have events during pregnancy, and I'll be looking at the consequence on the offspring. I'm in the University of Cape Coast, currently head of department for anatomy and cell biology. Right, so this will be my outline. And so, fetal alcohol spectral disorder. So from the name, this is when a pregnant woman ingests alcohol, and then, unfortunately, the offspring may now exhibit this. In the US, there is about 10% of pregnant women who use alcohol. In Ghana, much work has not been done, but those who have done this work indicates that between 20 to 30% or even higher of pregnant women are using alcohol. So that is certainly a major cause for concern. And still in the US, of the 10%, is that what some of these you can see, so it's a spectrum, but the most severe is the fetal alcohol syndrome. And what could happen is, in the nasal bridge, everyone has a nasal bridge, theirs appear to have a lower one, with their eyes getting to the medial region, the epicantral fold is there. And when you look at the lip, the philtrum area, that seems to be missing. They also seem to have a narrower orbital fissure, that is the opening of the eyes, looking a bit more Asiatic. That is for those of us in this part of the world. So unfortunately, all these children have the unfortunate outcome of their mothers using alcohol whilst they were in utero, and unfortunately they are exhibiting the symptoms. Of the various features, obviously the most devastating are the intellectual disabilities, whereby their cognitive functions unfortunately are affected. So that is what I'll be talking about. And then what the central dogma of biology poses that the DNA is transcribed into RNA, and then if that is translated into protein, then that is considered to be a gene. However, in humans, we know that only 1.5% of the entire DNA is coding for protein coding genes, and over 76% of the entire DNA is actually transcribed into various RNA species. And most of us, I believe, we know of some of these non-coding RNAs. So some of these include things like the ribosomal RNAs that I believe most of us are aware of, usually adapted for protein synthesis. And also importantly, the transfer RNAs, where we have the proteins attached so that they usually would help the codons, anticodons parent. But however, there are also, let's say, the new kids on the block. So there are circular RNAs, which I'll be talking about today. And there are also the long non-coding RNA. I'll be talking about this also. MicroRNAs, I think last year there was a poster on that by myself. So with these circular RNAs, they would also form similar to how our general mRNAs form. However, we know of the exons, right, whereby we have exons coming. So what is happening here, for example, is that we have a number of exons, let's say 2, 3, 4, and 5. If this will form the general protein, then we expect that 2, 3, 4, and 5 should come together, form your protein product. In circular RNA, however, there is what is called the back splicing. So what is happening here is that, in this case, second one, which I'll be talking about in much detail, we have the 5' end of exon 2 and then the 3' end of exon 5 coming together to now form a continuous loop. So what is happening here is that that splice junction has a unique sequence, and then you can now develop probes to either detect this or knock it down, which I'll be talking about in some detail. And this field of circular RNA has become a really exciting field, which I'll shed some more light on as we continue. Well, there are various functions by this circular RNA. However, one of the major ones is sponging microRNAs, and then various other phenomena. Because this is a new field, much more information is being found. Although some of this could be translated into proteins, that has been observed not to be the major functions of these circular RNAs. Now, so for this work, the method, so we use, this was done in University of New Mexico. During the time of my PhD studies, we collaborated with the Jonathan Brigman lab. And what happens here is that, so before the pregnant dams get pregnant, they are fed alcohol, and because the taste of alcohol is such that the mice usually don't like it, a bit of sugar is added, so that's saccharin. And then, and also during pregnancy, so what happens is, for about 4 hours every day, these mice are given ethanol throughout the entire pregnancy, and after that, once they give birth, then that's it. So they are just given a prenatal alcohol exposure. And then after that, well, this is the human brain. So we are interested in the frontal brain. Frontal brain, because that is the region where most of our personality is. So when it comes to your behavior, executive functioning, and so on, that is the part of the region. Occipital region, that is at the back of your brain. That is usually where vision processing occurs. Then there is also, the hippocampus is not showing here, but that is usually in humans found embedded within the temporal lobe, and that is important for learning and memory. These we investigated in here. And what we did here was mainly to use QRT-PCR. We designed primers for the splice junction so that only that circular RNA can be detected. And here we are using SEC-HOMA1. With the HOMA1 protein, it is usually important when it comes to memory formation because it is a scaffolding protein. However, the circular RNA appears to also have some distinct functions. So now to our result. So what we observe in this result here is, so we just want to pay attention on the highlighted ones. So while we have the adult hippocampus, and then, so this is the adult frontal cortex. So what happens here is, so after the prenatal alcohol exposure, between 80 days to 90 days, these offsprings were then harvested. They had their brains harvested. RNA, but this is a long non-coding RNA. That, however, was not changing. And when we also come to the H19, that is usually responsible for imprinting, so it's part of the silencing of the genome. And here we observe that whilst we are having a reduction in SEC-HOMA1, we are observing a converse upregulation in H19 non-coding RNA. So that would talk about in much detail soon. Then now when we come to the hippocampus, we are still here observing a similar outcome. However, when we look at the EIF3A, we are not having that significant upregulation, although there is a trend. But if you look carefully, look at the females here, we notice that we are not observing these changes. So this is telling us that this outcome is indeed sex-specific. And it's also important to note because it is now becoming clear that some of the outcomes being found in males and females are different. So we should not focus usually on males, which we usually do in animal research, but we should also consider the females as well. So now, when we now look at the various other genes in here, so here we are interested in the expression in the various brain regions. So we mentioned the frontal cortex, hippocampus, and then occipital cortex, and then the cerebellum. Cerebellum usually coordinates movement. So when it comes to learning, that is usually not considered to be a major, it's not considered to be a major role player. And when we look carefully, we notice that the frontal cortex, important for various higher order functions, that is much higher, followed by occipital cortex, that, as I've mentioned earlier, that is also for vision. And then we now have hippocampus for learning and memory, then cerebellum is lowest. And if you look carefully, you notice that that trend is reversed. So this is SEC-HOMA1, and then when we look at H19, we now observe that the cerebellum, which had the lowest SEC-HOMA1, that here has the highest, whilst the frontal cortex, which has the highest in the SEC-HOMA1 levels, we now observe a reverse. So we are observing these changes. And then when we further go on to look at the GAS5, that is also another long non-coding RNA, we are also observing that there are differential expressions in various brain regions. When we also decided to find out, okay, well, could there be any association, that is the expression of SEC-HOMA1 RNA against the H19, we are observing a significant inverse correlation, suggesting that as the H19 levels are increasing, the SEC-HOMA1 levels are decreasing, and this is happening for both the saccharin and also the prenatal alcohol exposure. So that is telling us that what is happening in the relationship is irrespective of exposure to ethanol. So having seen all this exciting work, we are now interested in, okay, what about the developmental expression? Are these non-coding genes, SEC-HOMA1 and also H19 and GAS9, are they differentially enriched? So when we look at the E18 mice, so these are prenatal, we observe that the SEC-HOMA1 levels prenatally, that is lower. But when we look in the adults, we notice that that is higher, telling us that as you age, these circular RNAs are increasing. And interestingly, we observe an inverse correlation with the H19, which was lower prenatally, and then in the adults, that is higher. However, GAS9, we are observing that that is relatively stable. And when we look at the comparison at the various levels, we notice that this circular, so the circular RNA, SEC-HOMA1, and then the age 19 and then gas 5, at the same age, those were not changing. So it's telling us that there seems to be something unique about the age and also the ethanol exposure. So we decided to ask a final question. Could there be a relationship between the EIF 43A and then age 19? Because some literature was suggesting that gas 19, for example, seems to promote the secularization. However, age 19 could prevent the secularization from happening. So we decided to use neuroblastoma cell lines, differentiate them into neurons, and then also see if we could knock down the, in this case, the age 19. And when we did that, as you can see in this case, the highlighted regions, we observed that. So here we have a number of clones. And we could see that when we were able to successfully knock down the age 19 long non-coding RNAs, interestingly, we have a significant upregulation, telling us that the upregulation of the circular RNA seq HOMA1 is indeed only happening when the Gas5 genes or non-RNAs were free to be available to then allow for secularization. And then when we also look at the correlation, we observe that indeed there was that significant inverse correlation, telling us that so what is happening here is that for the mechanism, H19 RNA, when present or in high amounts, will sequester the Gas5 gene, and that will prevent the secularization. This could be a putative mechanism that could contribute to some of the deficits observed in the fetal alcohol spectrum disorder. So to conclude, I've given data to show that the SEC-HOMA1 circular RNA is enriched in the adult brain, while the H19 long non-coding RNA is enriched in the prenatal brain. We've also had exposure to show that the rodent model of fetal alcohol spectrum disorder, we have a significant reduction in the circular RNA, in this case, in only the male adult mice, meaning that for females, there could be some other mechanism. And we also have unique enrichment of SEC-HOMA1 expression in which the frontal cortex had the highest, with the cerebellum having the lowest. We've also given data to show that the H19 is what is affecting the secularization, in that when we were able to knock that down, we then had a higher secularization expressed in the levels of circular RNA SEC-HOMA1 levels. So now, well, I've left the US, I'm back to Ghana, and some of this exciting work, naturally we have some challenges. So I'm now thinking of moving from the neuropsychiatric role, which I'm still interested in, into some of the non-communicable, but importantly, into the infectious disease arena. And these papers have all been published. So we have circular RNA in prenatal alcohol exposure. And this paper, this was in molecular psychiatry. That, because of the excitement of this work, it made a cover page. Hopefully, we'll do some exciting work here as well. And then, okay, good. So let me bring this one. So for my acknowledgement, my mentor, Dr. Nicholas Melius, he has taken this work into a very high level now, because currently he's a part owner and he holds a number of patents in the circular RNA genomics. And what they are doing here is they are using this as a precision medicine, whereby they are using circular RNA to investigate various neuropsychiatric disorders, such as depression, for example. And this is giving us a new frontier. For the PhD work, I was sponsored by the AIMS, and currently I am a Fogatti Fellow, and I'm really excited to have both DJ and then Gordon as my current mentors. I thank you all for your kind attention, and I'll be happy to take questions.
Thank you very much. Questions, please.
Thank you for your presentation. So though it's clear that you said total abstinence from alcohol intake for pregnant women is the way out. So, but I think in some cases it may not be possible. So can you recommend in terms of if total abstinence is not possible, do you do something like in terms of dosage, if you take certain amount of alcohol, what could be the outcome of the research?
Well, interesting question. I wish I could say yes, but I can't because of the training I've had. Well, but certainly, well, so to answer your question, if a person knows their status, obviously, it is just, well, I guess there could be some therapy given, but currently, the real difficulty is this. We know that the placenta is a barrier, but unfortunately, when it comes to alcohol, the placenta is not a barrier. The sad aspect also is that you could have alcohol being retained by the placenta and converted into products like acetal, sorry, I'm struggling with the chemistry today. Right, with some of the acetate products, and then these also affect the developing brain. If you look at the development of the brain from the 3rd week of development, by that time, a lady who is pregnant might have just missed her period for just a week. The brain starts development. And this would continue up until early adulthood. So that is why, especially in the Western world, alcohol is not allowed until after 21. Just so that you allow the brain to develop sufficiently well. So the sad news is there is unfortunately no safe dosage for alcohol, especially for the pregnant woman or a woman who wants to be pregnant. So sorry with this, although it may sound a bit religious, abstinence is still the key for a pregnant female or a female who wants to be pregnant. Thank you.
Yes, there's another question here.
Yeah, thanks for that question. In fact, I had to look that up because unfortunately in our parts of the world, some of these have not been done as we expect in a very good high rigor study. So the studies I've seen, I think I've seen a few of them, some in Jamestown, Ashanti Bekwai. But to answer your question, we need to have a deliberate policy of, for example, in the antenatal care of most of the ladies who go for that. Some of these could be asked routinely, and over a period of time, we could generate robust data to determine the scale of the problem, and then based on that, because to really do a good job, then we could even go a step further to find out, at least in the US, they are observing that of the 10% of those who are using alcohol during pregnancy, about half of them, unfortunately, are ending up with their offspring with this. Would it be the same in our region, in our country, or could it be high or low? And what could be some of the driving factors? Certainly, those are important questions to investigate. Thank you.
Okay, one more, please. And then there's one here.
Hey. Should I go?
Please go ahead, and then Dr. Kwashi.
Okay, thank you very much. This is interesting. So, with respect to placental barrier, you know that between mouse and human is different in terms of permeability of the placental barrier. And we've learned that from the tragedy of thalidomide. I don't know if you know about that. So to what extent do you think relying on mouse in your study will be reliable?
Okay. Thank you, very good question. So I think one easy thing about alcohol, unlike most of the other substances, is the size and also its penetrance in the blood-brain barrier. It's very easy, such that anyone who decides to binge drink would hardly escape the effect of alcohol. And you show symptoms when your brain, especially cerebellum, has been hit hard. So inability to maintain your balance, falling, and all those things. Those are really central nervous system effects. So when it comes to crossing the blood-brain barrier, ethanol, unfortunately, has no difficulty. So that is the real challenge. So most of these, and this model is actually a validated model. So what, for example, the Brigman Lab we collaborated with, what they do is they really have this touchscreen paradigm where they are able to let these mice, they train them, and then they are able to see, one, can they learn? Usually they're able to learn. But when it comes to altering their behavior flexibility, that is part of the difficulties they have, for example. So there are various aspects that are translatable to humans. But when it comes to alcohol use and its effects in humans, when it comes to the rodent models, most of those are translatable. So you don't really have that barrier.
All right, thanks. And we'll take one last question from Dr. Kwashi. Keep it very short, please.
All right. So I have two short questions. The first is unrelated to alcohol. I mean, do we know if low birth weight is a problem in Ghana? And then related to the data that you showed us, showing that this is very gender specific. Are these like Y-linked genes or something?
Are they?
Y-linked, like being expressed from the Y chromosome.
Oh, okay. Okay. Right, so with the Y link, then that one we have to be looking at the H19. Because H19, I think that is more expressed on the X chromosome. You then have the IGR, that is more on the Y chromosome. So what it seems to do is the imprinting. It seems to target that of the male so that the female more or less flourish. So you have that bit. With the low birth weight, many disorders will present with low birth weight. So the difficulty, for example, is that once you are not finding out whether, for example, the mothers have used alcohol during pregnancy, you may not catch that. Because, for example, severe malaria and various diseases could, depending on the exposure, give you low birth weight. But we have to find out to know whether indeed prenatal alcohol exposure is part of that.
All right. I wasn't sure about the H19. I recall it's an imprinted gene.
Fine, yes.
So it has parental, it's parental imprinted. So depending on the sex it's coming from, it may or may not be expressed. I'm not sure it's just on the Y chromosome. Okay. But I will check.
Right.
But yeah, I know it's an imprinted gene.
Right.
So if it's an it's imprinted, it should be coming from both sexes. So if it's Y, Y will just come from the male.
No, so sorry. You'd have it's usually silenced in one.
Yes.
Yes.
So it's imprinted, yeah.
Right, yes.
It's, yeah.
All right. A hand is furiously waving up. I don't know.
After. Please see me in chambers.
All right, thanks again for accommodating me.
Sorry.
Thank you.
Thank you very much. Our next talk will be done by Akiko Minoda on the topic Milking Mouse Aging Atlas. She has 20 minutes, 15 minutes for presentation and 5 minutes for questions.
Thank you very much, Gordon, and everyone for the invitation for this opportunity. It's my first time in Ghana. Got here a couple of days, yes, and I'm loving the food, the people, and music. So, yeah, I'm really enjoying my experience in Ghana. So, today I'd like to introduce our work. The data of which was generated in Japan at the lab, in my lab, that was at RIKEN. But I have since moved my lab to the Netherlands, so now I'm located in the Netherlands at the Radboud University. And I understand there is some interest in starting or establishing single-cell analysis in Ghana as well. So our data is all single-cell, and I'd like to introduce you what kind of things, how powerful the single-cell technology can be for your studies today. So let me just go back, actually. So I'd just like to quickly sort of explain the word mooking. So this is Mouse Aging Atlas. Mooking means germ-free in Japanese. And I told you why we call this Mooking Mouse Aging Atlas next. So my lab works on aging. We're interested in how cells age. essentially, because age really is one of the biggest risk factors for many, many different diseases. Osteoarthritis, atherosclerosis, obesity, there's so many that you can list. So what is it about aging that puts us all for developing these diseases? So that is something that we're interested in digging into. And there's been many, many hallmarks of aging that has been described. And this is the one on the right has been taken from a review recently. And there are some that you can consider to be coming from environment. So the diet, lack of exercise or exercising, amount of stress, all of these could be considered as an environmental and therefore extrinsic aspects, factors that may be driving or controlling the rate of aging. Now, there are other aspects like telomere shortening that is associated with every time the cells divide, that is very famous. That is one of the things that was first described for aging in yeast studies. And these can be considered as intrinsic cellular aging, because they are not really necessarily strictly coming from the environment. So I consider aging as something, it's a combination of extrinsic as well as intrinsic cellular aging. So, but it has been really, really difficult for the field to decipher between these two aspects. Very, very difficult. However, of course, feeding low-fat diet or low-protein diet or giving exercise to mice, these kind of things studies have been carried out in order to try to differentiate these things. So that's something that we are also interested in executing to look at the differences. One of the aspects that I thought it hasn't really been addressed in the field is the effect of the microbiota on the rate of aging. So, of course, we have a lot of microbes in our gut, skin, throughout the body, in fact, at the somewhat level. So, are they playing a role at all in driving the rate of aging? And to answer this, or at least try to gain insights into this question, I decided to deploy germ-free mice, compare them. So the germ-free mice are born and raised completely free of germs. And they're raised in a sterile vinyl protected cage. And so they're kept in a sterile condition their entire life. So we did this to age also the mice in this sterile condition. And then to compare between the mice that are born with the microbes. So this is the study design that we conducted. We took young, 9 weeks age of mice, young adult, as well as old mice of 19 months of age. We dissected tissues. We put them through single-cell dissociation of the tissues, and then put them into, in our case, a 10x genomics Chromium machine to generate the data of single-cell RNA-seq as well as single-cell ATAC-seq. And ATAC-seq looks, you can take a look at chromatin accessibility, in this case at the single-cell level. We conducted this for 10 tissues. We're also carrying out additional tissues, but the data, the analysis that we are really diving into is from 10 tissues. So this is what I will show you today. And we believe this is going to be an amazing resource for the community of aging, as well as the microbe, the field. Sorry. Okay, so these are the tissues. This is the list of tissues that we collected and generated the data. We also collected some other data sets, like lipidome and metabolome. The lipidome data set actually, in fact, has been recently published in Nature Aging. So for those of you interested, you can refer to that study, and it's very, very comprehensive. Our work data is only on female, but we actually obtained tissue samples from male mice as well as some other tissues, and our collaborator actually measured or did a lipidome analysis on male, female, germ-free SPF, 4 different ages. We, of course, have some other data sets but I will not go into so much detail there. But just quickly, if you're wondering, if you're not familiar with single-cell analysis, why bother doing single cells? What is the deal about it? Well, if you're interested in analyzing tissues, tissues are composed of a mixture of many, many different cell types. For example, liver is one of the simpler tissues in terms of composition of different cell types, but also, of course, it has many, more than 10. And when you are interested in figuring out where is the difference if you do RNA-seq analysis, bulk RNA-seq analysis, where is the difference coming from? You see lots of genes going up or down. Are they all coming from specific cell type, or are they coming, is it averaged out data of coming from different cell types? And this is, of course, important in the context of different various diseases, including cancer, which the field has taken up the single-cell technology very rapidly. So this is the sort of if you do the whole tissue, that's what we call as bulk RNA-seq analysis these days. Okay, so set all of this. And so we did this, basically we want to do this. A lot of studies in the past have looked at bulk RNA-seq between young and old tissues. Again, but we don't know, we cannot pinpoint which cell types are driving these differences. And another sort of way to sort of describe this is that, did anybody have a smoothie this morning? I did. But you really don't know, if you're asked how much of which fruit does a smoothie have, just by tasting or smelling, it is very difficult to really describe accurately how much of what is in there. So that's the sort of similar to bulk analysis, the smoothie itself. But really is, again, it's difficult to interpret the actual mixed data. So the single cell analysis tries to capture the original fruits without destroying them. So the single cell analysis basically takes mostly enzymatic reactions, dissociates the tissues, puts them into single cell suspension, and then you try to encapsulate individual cell, then take RNA out in the individual, either droplet or well. There are many, many different ways of doing this, and I won't go into details, but if you're interested in different techniques, please come and talk to me afterwards. So our data, single-cell RNA-seq and single-cell ATAC-seq, we have annotated roughly 46 cell types. When you go into a little bit sort of higher resolution, we can also see around 74 subtypes. So what kind of data can you see? So in our, for example, what we call cell type composition analysis, one of the questions is there a difference in, let's say, some cell type expanding with old age? And is that exacerbated by the presence of the microbiota? So one of the things that caught our eyes at the beginning is the CD8 positive T cells. And you can see that, okay, so this is each dot represents a cell. And it's colored by what kind of cell type it is. So these are all cells we obtained from the young mouse. And these are a mixture of all of the tissues, by the way. And this is all of the cells obtained that we annotated as either memory CD8 T or effector CD8 T cells. And you can see, just by sort of looking at it, that there is an increase in the number of CD8 T cells in the old. And if you look at, this is sort of natural aging with microbiota. This is what I'm calling natural aging. In the mice that don't have microbes, that I refer to as the process of germ-free aging, you can see that actually the expansion is not really observed anymore in the old germ-free. suggesting that this expansion can be attributable to the presence of the microbiota. All right, so I therefore categorize this kind of phenomena as extrinsic aging because it is exacerbated by the microbiota. Now, this is, on the other hand, an example that we have captured of intrinsic aging, because in this case, we see no difference between the old, germ-free old or SPF old. So this is B cells. We are seeing all the B cells here from the atlas, and we took just these two populations that we termed memory B cells and age-associated B cells. ABCs have been described in the literature to expand with old age, both in human and mouse. It's only been described in the last 10 years or so, and so with actually relatively little amount of information is known about this. And we think it is pathogenic, but actually we don't completely know it. It's been associated a lot with autoimmune diseases. And this population, especially, there's very, very little, hardly any in the young mice, and then huge amount in old mice. But even in the absence of the microbes, they still expand, and therefore we're calling these as intrinsic aging. And of course, we can do some sort of if the identity of these cell types are not known, then you can pick these up separately bioinformatically instead of sort of enriching by FACS and sorting. We can carry out sort of pseudo-bulk RNA-seq analysis of that particular population to do the differential gene expression analysis, for example. All right. Okay. And these ABCs as well, so the ABCs here shown in red, they're actually found in many different tissues. So these are the tissues that we have found ABCs in: bone marrow, PBMC, spleen, lung, mesentery, a little bit in the intestines. And we also have done sort of pseudo PCR analysis which suggests that they're actually circulating. And I won't go into details because I'm also probably running out of time. You can imagine you can do this for any cell type of your interest because there's a very comprehensive and non-biased set of data. And you can see, okay, in which tissue of interest does your cell type of interest increase or potentially decrease. This is just a proportion. It's not the absolute number of cells. It's a proportion, percentage of that particular cell type in a tissue. But you can sort of gain a hint into, is it going up? Is the trend going up or down with old age? And is that trend actually seen in or across different tissues? We don't see that to be the case, in fact. For example, you can see that this memory CD8 T cells expansion in the old natural aging is like comparing what's going up in old age is going up here, it's going down in germ-free, but that trend of red and blue is not really seen in every single tissue. So it can be very tissue-specific phenomena as well. Okay, thanks. All right, so then very quickly, aging. There is a huge thing about senescence. I won't go into the details, but senescence is a way of cells arresting cell cycle, and that can be pathogenic to other cell types as well. And we just did a quick sort of senescence scoring by using a set of genes called SEM-MAIO, and actually we find that alveolar macrophages has highest score across all cell types. And we are following this up a little bit more, and we can see that it's a combination of intrinsic aging, but that is exacerbated by the microbiota. So I won't go into more details, but the take-home message for today, I would like you to, you don't need to remember the details of which cell types, but in the end, the take-home message I'd like you to remember is actually we are trying to sort of figure out what age-associated changes are intrinsic versus extrinsic. And we have been able to determine some of these things so far. All right. Okay, so I'd like to finish up. Oh, well, okay, I'll spend 2 minutes on this. So this data was generated at RIKEN when I was in Japan, and has been almost 10 years since we started this project. And so many people have been involved, including and all of the collaborators, different labs and their members, in handling different tissues for us. And these are the people in my lab in Japan, especially Tommy, who really started this project with me, ex-postdoc mine, then got passed on to Renchen, another postdoc, and now Ward, a PhD student at the Radboud University, is really doing the analysis. Yes, thank you for your attention. I'd like to take any questions.
Okay, thanks for the presentation. Questions? Okay, you will take 2 or 3 questions.
Thank you, really interesting talk. I have 2 questions. The first one is, when you say aging in mice, so what are you comparing adult mice with really old mice, or what is your, is it juvenile mice?
How old is old?
And is it, so are they juvenile mice or are they young adults? How does that, And then the second question is, you said your age-associated B cells increased in number. So that's not a proportion, I guess that's an absolute number. And if so, how do you control for changes in size of the animal or weight with when you look at numbers? Thank you.
Yeah, so the first question, how old? So I also sort of asked around at the beginning, what is sort of appropriate old age? A lot of people in the field think, and I agree with them, when they're too old, they start to develop tumors and other things, so then you are really starting to potentially seeing disease as well on top, which of course makes the analysis difficult. And so we took 19 months, which is sort of a human age of like maybe 60s. If you look at different plots, they're all different. But I would say it's in the range of like 60s to 80s, potentially 70s maybe. And no, for the young is young adult, which is 9 weeks old. Yeah. Oh, sorry, I didn't answer your second question. We don't really, that's true. Obviously the mice get bigger and the tissues therefore get bigger. I don't know if this is really, really true, but what I've heard is that the number of cells don't necessarily go up, like increase a lot with age once you mature. It's the size of the cells that makes the organisms bigger. I've never done the actual absolute number of tissues, so there could be some difference. But when it's sort of this drastic in terms of the change with ABCs, I think it is, regardless of maybe there is some difference that we haven't taken into account for, still I think it would be significant, yeah. And it's also been seen in PBMC, in human, and there it's, more consistent, I think, more or less.
The last question.
Yeah, and thank you. Yeah, such an interesting talk. One very straightforward question and then one kind of practical one. So the straightforward one is, which strain of mice did you use? And
Black 6 N strain, yeah.
Yeah, so Black 6s have some peculiarities, right? Anyway, Are there plans to look at other strains of mice?
Not so at the moment, yeah.
I mean, it's a huge endeavor, of course. And then my other question was, if I understood correctly, this is something that people can query this database themselves. So I just wondered if you could talk a bit more about that.
Sure. At the moment, we haven't published it yet. So, of course, once it is published, it is will be accessible for everybody, and we're trying to make not just the raw data accessible, because which is minimum we are expected, right, at least by the journal standard. then you would have to run a lot of further analysis. So we're also going to be providing the processed files.
Idiot-proof.
Sorry?
Idiot-proof.
Yeah, at least of what we can, people who are very bioinformatically trained can start with from scratch and then do their own processing, because we have cleaned it up a lot. We have probably filtered out some of the cells that people might not want to, but we're kind of taking the conservative route of really going for high quality cells. And so, for example, neutrophils is known to have lower RNA count, and they might have been filtered out a bit more. But if you're interested in neutrophils, for example, should go back to the raw data and then reprocess to rescue them. And if you have very specific interest in looking at our data before publication, which will probably take more than a year, please come to me and we can collaborate and we can share the data with you just by signing MTA. Yeah.
Brilliant, so interesting, thank you.
Thank you. But yeah, I know we are done, but I think I can ask one question. Sure. So these mice that don't have they lack the microbiome. When you allow them to age and you basically eyeball them, do you see the obvious phenotypes you see in older, naturally aged mice?
Yeah. Yes, very good question. We do. Definitely the mice, germ-free, not happy mice. They have a lot of issues. So one of the things that I noticed that we had to actually put them down even before coming to the 19 months age, because they had enlarged cecum. And this is a known, described phenomenon where the cecum is just becomes so big that they're just dragging their body because it's so big and they're in so much pain. So enlarged cecum is one of the phenotypes that they have. Yeah.
Okay. Thank you. Don't go getting rid of your microbiome. It's not good. Thank you very much. That's fascinating. Now we have a fellow session by Fatima Oyawe, and she's going to speak on generation of cellular model systems of red blood cell diseases. So Fatima is an alumnus of WACCBIP, and she happened to be my student. So I'm happy to see her.
Good afternoon, everyone. I'll be presenting on generating cellular models for red blood cell disease. Today, anemia is insufficient LD red blood cell, and today I'll be focusing more on sickle cell disease and beta thalassemia. This is because these two anemias affect the beta globulin gene, while sickle cell disease is more of a structural form of anemia where a single nucleotide change has resulted into the change of the red blood cell. Beta thalassemia is an issue that has to do with quantity, where there is rather insufficient production of beta globulin protein, or better still, no production of beta globulin. These two anemia are currently being managed by chronic transfusion or use of disease-modifying drugs. But the issue with this current therapy is with blood transfusion, there's risk of alloimmunization and also iron overload. While when these individuals are treated with disease-modifying drugs, there are damages to vascular cells, or there's also variability in response based on genetics. And then these two conditions affect places with low income, so these drugs are quite expensive and not the best option. Hence, there's need to develop new therapy. When it comes to drug discovery, there's also the need to find the appropriate model to study these drugs in. Currently, we use mice models to study some of these conditions. However, it is not an ideal system, partly because the human erythropoiesis is completely different from what is seen in the mice. And if you're looking at drugs that specifically target erythropoiesis process, this is not a good model. And it's kind of unfair to be taking a lot of cells from patients who are already anemic. So my work really relies on the fact that we need to create a model line for studying this disease. At our lab, we've been able to make what is called the Bristol erythroid adult line, which is basically an erythroid line. which continues to expand in the pro-erythroblastic stage until put in a differentiation media where it eventually differentiates into reticulocytes at day 12. This line is especially special because it doesn't express fetal globulin. It doesn't have any issues with terminal differentiation, like it being unable to lose its nucleus to form reticulocytes or go on to form red blood cells. And then erythropoiesis in this line is less complex and is also a pretty straightforward process. So what we've done is to edit this beautiful line to form the disease line. So we do this by using CRISPR-Cas9 to edit the Bella lines and then keep them in culture for 24 hours in NEDI. So what NEDI does is when we CRISPR-Cas9 causes a double-strand break in the DNA, and when there's a double-strand break, there are two mechanisms of repair. One of these is homology-dependent repair and non-homologous end joining. What NEDI does is it inhibits the non-homologous end joining process, ensuring that the repair is solely dependent on the template that we provide for the cells. Then we go on to single sort these cells and then select for clones. Now, here is a situation where we are trying to create a disease line that properly mimics what we see in patients. So we go on to characterize this cell line by determining their viability during differentiation, and then determine also cell morphology, and we test for ineffective erythropoiesis, which is what is seen mostly in beta thalassemia. And then we go on to check for the globin expression profile. Okay. So one of the classical characteristics of beta thalassemia is ineffective erythropoiesis. Ineffective erythropoiesis is there will be normal production of red blood cell until it reaches the polychromatic stage, and at this point the cells then begin to die. So which is what we see for our beta thalassemia line, our homozygous beta thalassemia line, where at day 6, where we normally expect polychromatics and autochromatics to be formed, there will be a sharp decline, and eventually there won't be enough production of reticulocytes. So this properly mimics what we expect in patient samples. Then we went on to also determine the globin expression profile using HPLC. And we noticed in our beta thalassemia line, as expected, we do not expect them to express any form of beta globin. Then we went on to do the ineffective erythropoiesis assay. So what this assay does is we check for levels of CD36 at certain intervals during erythropoiesis to see how the CD36 is expressed, and also we check for annexin. We stain for annexin. During differentiation, the cells begin to lose expression of CD36. So a cell line that is progressing towards erythropoiesis will significantly start reducing as a reduction in CD36, which is what we see in our Y-type cell at day 5 and day 7. So if you compare from day 5 to day 7, there's been an increase in cells that have low CD36 from 2.1% to about 78% in our wild type. However, in beta thalassemia line, we have the cells increasing from 0.57% to 5.31%, but rather we have a higher population of these cells being expressed in the annexin quadrant, indicating that these cells are significantly dying, which is a feature of beta thalassemia. Here is also a morphology data where we look at the cells at day 6 and at day 10. For the wild type, the cells at day 10 then start to have a few reticulocytes and polychromatics, and this is missing here in the beta thalassemia line. As I said earlier, this line is characterized by ineffective erythropoiesis, so normally they are unable to form reticulocytes, so they end up dying. So you would see in this chart that these cells are more apoptotic than in the wild type. Another feature of beta thalassemia is presence of insoluble aggregates of alpha globin. So here we try to detect alpha globin in the cell pellets, and then this was missing in wild type and the heterozygous form of sickle cell, beta thalassemia, which was present in the beta thalassemia. moving on, for the sickle cell disease line, sickle cell itself does not necessarily have any issues when it comes to differentiation. The red cell is formed, but the formability comes after red cell is formed. So ideally, I didn't expect to see any issues during differentiation, so which is quite similar to what we see in the wild type. We also went on to do HPLC to determine the hemoglobin profile, and then used Western blot to detect the hemoglobin S. So the antibody we use here is actually very specific for a sickle cell beta globin subtype, and that's why we're only able to detect it in the sickle cell disease line. We also try to see if there's any ineffective erythropoiesis with our sickle cell disease line. There's some level of ineffective erythropoiesis, but it isn't as pronounced as what you see in beta thalassemia. And ineffective erythropoiesis is a classical characteristic for beta thalassemia and not sickle cell disease. This just further confirms the ineffective erythropoiesis not being a case in sickle cell disease, because we see a few reticulocytes here. Then we keep these cells under hypoxia to see if they are able to sickle, because that is essentially the characteristics of sickle cell disease. And then we see a few sickle cell here. So far in our lab, we've been able to characterize and validate disease models for beta thalassemia and sickle cell disease that properly recapitulates what you see in human. And then this line serves as a sustainable supply of disease cell line for studying underlying molecular defects or identification of new therapies. So currently what we are doing is to validate an heterozygous sickle cell disease line, and then using this line to create an high throughput tool for drug screening for both sickle cell disease and beta thalassemia. And we're also characterizing certain mutations that are known to alter disease phenotype. I'd like to acknowledge everybody in my lab and my supervisor, Professor John Frayne and Dr. Daniels. Thank you.
Thank you very much, Fatima. It's really good to see you. So after introducing her the first, I saw Peter was looking very hurt. He was a co-supervisor. So, co-supervisor. All right, time for questions. Yes, just one.
That's really interesting. Can you tell me, now you've established the models, what's the next steps for your project?
So now that we've established the models, there are certain mutations that are already found in these individuals that are known to either increase their severity or reduce the severity. So what we're trying to do is further characterize those mutations in this our established line and see how we can target some of these for therapy, especially in the case of beta thalassemia, where these individuals, they are unable to make red blood cell and they are constantly dependent on blood transfusion. So beta thalassemia, for example, some patients experience high level of gamma globulin, so they don't have issues with ineffective erythropoiesis. So if we have mutations that are specific in patients that do not really show symptoms, this can then be channeled towards therapy.
Thanks. Any other questions? Yes, in the back.
Hello. Thank you very much for your presentation. But why beta thalassemia and sickle cell only? How about the other thalassemias?
How about the other thalassemia? So there are other people in the lab working on that, but for me, I'm focused on beta thalassemia and sickle cell disease.
Hi, Fatima. Great talk. I'm just, maybe I missed it, but are you looking at potentially where you have a heterozygous with beta thalassemia and sickle cell?
Yeah, so with that, we started working on that as well, but we're trying to reduce how much complications we start with at the beginning.
All right, thank you.
All right. Thank you so much.
presentations. I don't know how many sponsors.
Yeah, Dr. Kwashi, how many?
No, it's just me.
Okay.
One sponsor, only one topic.
Okay, I just want to see my presentation. But are they not
Oh, please go ahead.
Are they
Oh,
not
do you have
gonna-
slides?
Yes, I've got slides.
Oh, okay. Is someone working on it?
There we go. There we go. Okay. So I am aware I'm standing before your coffee. So I'll try and make it quick. It is a little bit of a speed dating session. So my name is Joanne Mizrahi. I'm here today representing Thermo Fisher. So I don't know, maybe just a show of hands. Anybody know Thermo Fisher Scientific?
Oh, yeah, everyone. Everyone.
Okay, that's good to know. That's good to hear. So let's see. Okay, so Thermo Fisher Scientific, our mission is really to ensure that we enable our customers to make the world cleaner, healthier, and safer. And I think part of that mission is really to ensure that we give access to all of our customers in Africa, making sure that they can access these innovative products. So we have partnered with a local supplier, LabMart. So I also just wanted to thank Richard. He's really passionate about science, and he's made it possible for us to actually be at this conference and to be able to make sure that everybody in Ghana and West Africa can have access to Thermo Fisher Scientific products. So we do have a stand. If you want to come and discuss with us in more detail some of the products and where we can support you, please feel free. I only have 10 minutes, so it's not even going to be able to touch on everything that we do. But I'll try my best just to give you guys a high-level overview of some of our newest and innovative products. Okay, so Thermo Fisher Scientific, we really are the leading science supply company in the world. We pride ourselves on investing $1.3 billion annually in research. We have over 120,000 colleagues across the globe. So when you're using Thermo Fisher Scientific products, you really do have a big team behind you to support you and make sure that you can be at the cutting edge of science. I'm sure many of these brands that we have up here as part of the Thermo Fisher umbrella are familiar to you. I think many of you would have grown up with Applied Biosystems, Invitrogen, GIBCO. I remember when I was in the lab, some of the first products that I used were the GIBCO products. So we do have a powerhouse of tools which you can use to make sure that you are successful in your research. We do service 4 main segments: life science solutions, specialty diagnostics, analytical instruments, and then laboratory products and biopharma services. My focus and what I'd like to talk to you a little bit about today is the life sciences solution segment. In the life sciences solution segment, we cover biosciences, bioproduction, and genetic sciences. So my job today is to tell you a little bit about the genetic sciences portfolio. We offer the top tier genetic analysis products, and some of our products which we have available are for digital PCR, real-time PCR, Sanger sequencing, next generation sequencing, and then microarray. Okay. Sorry. Okay. So one of our latest innovations is a digital PCR instrument called the Absolute Q instrument. And the Absolute Q... Sorry. I feel like it's my wedding day again. Wow. Okay. So I'm sure many of you would have heard about digital PCR. It is one of the current buzzwords going around. And digital PCR is really quickly becoming the gold standard in precision, absolute absolute quantitation of nucleic acids. So if you're wondering where digital PCR could fit into your research, you can use it when you need accurate measurements of targets in low abundance. If you're monitoring small changes over time, so routine monitoring, absolute quantitation as opposed to relative quantitation, answering complex questions like CRISPR drop-off assays. I don't know what that is. But if you need a solution that is also resistant to inhibitors. And then some of the other applications that you'd use digital PCR could be for rare targets quantification, absolute quantification, copy number variation, gene expression, and then wastewater monitoring. You could also use digital PCR if you're looking at generating reference standards. If you want to know how much viral load you have, or perhaps confirmation of NGS variant detection. Oops. Okay, so the Absolute Q really designed with innovation in mind. It's a single instrument, so it's a very easy to use workflow. You just have one instrument, not a whole range of different instruments. It takes about 90 minutes to do a run. And it's very consistent, so we have 20,000 consistent microreactions, and you can multiplex up to 4 different targets at a time. Okay, then, oopsie, as I mentioned earlier, Thermo Fisher Scientific is really one of the leaders in terms of sequencing technology. So Sanger sequencing is still the gold standard when it comes to capillary electrophoresis. We brought out the first automated sequencer in 1986, and since then we've really just worked on improving our sequencing offering. So our latest addition to the capillary electrophoresis family is the 6 Studio Flex instrument. 6 Studio Flex has some really unique features, so you can keep the reagents on the instrument for longer. So it was always a big struggle for our customers. You had to replace your buffers, your POP every couple of days. Now you can leave them on for at least a month. You can prioritize urgent samples, so if you're running a set of sequences, you can choose which sample to run first. You can continue loading the samples while you're running sequencing, and it also has integrated barcode reader. So the 6 Studio Flex also allows you to move between 8 or 24 capillaries, so you have a flexibility in terms of throughput. Then I'm sure many of you are involved in real-time PCR research. So over the years, Thermo Fisher Scientific has really been a leader when it comes to real-time PCR products. And we really do position our real-time PCR portfolio as an ecosystem. It doesn't help if you just have a good extracted DNA, RNA sample. You really need to have a quality instrument, an assay, and a great master mix to make sure that you have a successful run on your real-time PCR instrument. We do offer more than 20 million pre-designed assays in a flexible range of formats. To go with our real-time PCR portfolio, we also have analysis software and support to make sure that you get the best results every time. We also have a fast run mode, so you can do a real-time PCR experiment in as little as 40 minutes. We have a range of instruments which cater for different throughputs, different budgets, so you can start with the QuantStudio 1 and work your way all the way up to the QuantStudio 12, where you can look at 3,000 different targets at the same time. The beauty of the TaqMan assays, I think one of my favorite products to sell, is that you can start from a single tube You can scale up to our open array plates, which allow you to have up to 3,000 throughputs with pre-spotted assays. So I think a really great fit if you're looking at lots of different targets and you've designed an assay and you want to do a lot of routine runs. Okay. To go with our assays is a high quality master mixes. So we have a range of master mixes that go together with assays for different applications. So if you're wanting to do maybe cyber green, or if you want to do fast experiments or working on viruses and you want a one-step master mix, we have lots of different options available. Okay, and then the last technology I just wanted to share with you is our next generation sequencing technology. So we offer two options. One is the Ion Gene Studio S5. So this is a scalable, flexible throughput instrument, or the Genexus, which is designed more for a clinical setting. With the Genexus, very little hands-on time and quick to set up a run and super fast. So you'll have your, for example, COVID virus sequenced in 24 hours. Okay. And then another innovative technology that we're very proud of is our AmpliSeq panels. So we have a range of off-the-shelf panels designed for targeted sequencing, and you'll see we have a lot of different options covering bacterial, viral, fungal, and parasitic options for you to look at. Again, many options that are off the shelf, specifically designed for infectious disease research, but you can also design a custom panel for targeted sequencing. And together with that, we do realize that when you're doing next generation sequencing, you'll be generating a lot of data. And to ensure that you have the support that you need, we do offer bioinformatics training, bioinformatics support, as well as remote monitoring of your instrument. Okay, and I think that's all for me.
Thank you. All right. So next, no questions, right?
There's a question.
There's a question? Okay.
I think so.
One.
Our procurement law is atrocious. And it's atrocious, and it involves a third party. So is it possible to have, say, a warehouse for consumables, and then the cold chain, that if a researcher wants something, he could just go into this warehouse instead of going to Europe and through a third party? Because it takes about 6 months if you want to do any work over here, and you need simple consumable like tests. It's just crazy.
Okay, so I think my colleague Lindy will be.
Right, as Joanne articulated, we actually have a partner with LabMart, and the primary reason we've decided to do that is to really address some of the challenges our customers in Ghana have expressed. The first is really around the availability of products. The second is really around the cost associated. And we do have a table outside with LabMart, and in conjunction with them, we are ensuring that we're bringing our products directly to the local market. So I hope that has explained your question a little bit.
I hope it's this.
Is it possible to cut the third party?
They are an extension of us, therefore they are our partners. And so at the end of the day, we want to ensure, because sometimes the legislation in the countries dictate that you need to be able to have a local supplier to provide the products. Right? And taking that into account, we also want to be able to empower the economy of Ghana, as opposed to the money's always going abroad. And yet you're still able to get the exact same quality of products.
But the prices marked up. The
Excuse me?
prices.
What about the prices?
Yeah. Are they marked up?
believe you me, if you guys are here on Friday, I will highlight on my discussion some of the things we're doing for you.
Okay.
Thank you.
All right, thanks. So I'm sure people have questions, but it's coffee time, and we're coming back at 4:40. Right? Yes. So can have your coffee. And thank you so much.
It's a pleasure. Thank you.
Don't forget to get copies of your pictures. Your pictures, yeah. I think you took the best pictures. Yeah, okay.
All right. We're running a little late today. So, as much as I'd like to wait for everyone to come in, I think we're going to move on with the next session. And the chairs for the next... Oh, yes, okay. The chairs for the next session will be Dr. Janelle Biman and Dr. Samuel Dodu.
Joining the last session. So we're going to start off with the first plenary talk from Irene, who actually just spent 5 weeks in our lab in South Africa with us. I'm very excited to hear about her study on neutralizing antibody responses in SARS-CoV-2 infection and vaccination.
Good evening, everyone. I am Irene Owusu, a postdoc in Peter's lab here at WACCBIP, and today I'll be sharing with you work that we've done to show how COVID-19 patients neutralize different variants of SARS-CoV-2 with and without vaccination. Okay, so last year, sometime when the WHO announced the end of the COVID-19 pandemic, I'm sure a lot of us were relieved and we were excited to do or go back to our normal works. But then one thing we are supposed to always keep in mind is that the end of the pandemic did not mean that the virus had been eradicated. It will still be with us, maybe till the end of the world or for a very long time. But then would admit that the presence of the vaccine played a crucial role in putting the COVID-19 pandemic at bay. But then questions still arise, or we still have questions, even at the time that the vaccines were being administered, that were they necessary for us? And even now that, especially in Ghana where we didn't experience a lot of deaths, we wanted to know, people kept asking, was it necessary for us to have the vaccines? And even if it was at the time, when there were new variants that were emerging, we started asking if these vaccines that were produced from the ancestral strain, if they were able to protect us from the emerging variants. And these are some of the questions I'll try to answer at the end of this talk. So speaking of vaccines, there are different parameters that we use to measure vaccine success. Among some of them are the induction of immune memory, as well as production of neutralizing antibodies. Now, for the sake of my talk, I'll be looking more into neutralizing antibodies, and how they work is very simple, or this is a very simple explanation of how neutralizing antibodies work. So for for SARS-CoV-2 to infect a cell successfully, the spike protein on the surface will have to engage with a cell surface receptor, which in this case is ACE2, for a successful infection. What neutralizing antibodies would do is bind to the spike proteins and then prevent the spike from interacting with cell surface receptors and so inhibit infection. Now, when this happens, then a person is protected from infection and the downstream effects. How do we measure these neutralizing antibodies? There are different assays and platforms. In our lab, we use two main platforms, the pseudovirus assays and then the live virus. But data I'll be showing is data that we generated from live virus assays that we use the microneutralization assay. So this is a relatively cheaper assay, and it uses the live virus. What we will do is just introduce patient plasma and allow it to interact with the virus for an hour before we infect the virus onto cells. So the whole idea is that if there are any antibodies in the plasma, they'll neutralize the virus before we introduce them onto the cells. And then after 3 days of allowing infection, we measure the neutralization titers just by staining the cells and looking for complete. So here what we are measuring is complete neutralization. So we tested, so data I'll be showing, we've tested a lot of samples, but I'll be showing just data from 199 plasma samples from COVID-19 patients. And these patients were sampled across 5 different pandemic waves. And most of them were not vaccinated, while just about 36% of them were vaccinated. And talking about the waves, we grouped them according to these periods. So from January to November 2020 was wave 1, and it peaked in July 2020 in Ghana. And then wave 2 peaked about 6 months later and was dominated by the Alpha variant here in Ghana. And then some 7 months later, we had the Delta-dominated wave 3. And then just 4 months later, which was really short, we had Omicron and all its lineages that we still have now, taking waves 4, 5, and whatever waves we have now, if we have any at all. So for my study, I looked for neutralizing antibodies against these 4 SARS-CoV-2 variants. So we have the wild type and 3 other variants that we looked at. And I have beta in red because in Ghana, we did not record cases of beta variant. But then I added it in the mix just to see how we responded to a variant we were not introduced to. And this diagram is just to show you an appreciation of how the variants work. So, or how the variants look like. For every new variant that emerges, there are more amino acid substitutions or mutations. So you see that beta, which was earlier, has few mutations, and then Delta increases, and then Omicron has like 58. And for even the Omicron lineages, it increases. So this is just for you to appreciate. So I tested these 4 variants for neutralizing antibodies in 199 samples, and then I found that, so these graphs are neutralizing antibody titer levels against the different variants. So we have wild type, beta, Delta, and Omicron. We found that even patients or donors from the early waves were able to neutralize, or some of them were able to neutralize viruses of the later waves. So you see for wave 1, for all the different variants, you see that we have a couple of people who would neutralize the virus. And then for wave 2, we had a few. And if you look at wave 3, you see a lot of neutralization against Omicron. And what was also fascinating is that most of our donors neutralized the beta variant, which we did not have in Ghana. So that was an interesting thing to look at. But then one thing we found was that in wave 2, most of the donors from wave 2 had very low levels of neutralization, okay? So you see that we would expect that as the waves increase, we'd have more people having neutralizing antibodies. But then it dampens at wave 2 before it picks up momentum. And so we were wondering if variants in wave 2, like the alpha, which was more dominant, if they are immunogenic, or if they are less immunogenic, or whatever it is, but then we would have to test that. And so we picked these 10 donors, or we have these 10 donors who neutralize completely all 4 viruses, okay? So for donors whom we were able to test all 4 variants, we had about 91, and we see that about 11% of them neutralize completely all 4 of the variants. And most of them, as you can see here, were vaccinated. We have a few. Of course, this one was not vaccinated, but then he had consistently high levels of neutralizing antibody titers, which is an interesting case to look into. But then you also find that most of the donors who had who neutralized all the viruses came from the later waves 3 and 4, except this one, which is also an interesting case to look into, who is from wave 1, but then was able to neutralize all 4 of the viruses. That's the only donor we had from wave 1. And so seeing that vaccination, most of the vaccinated people had more neutralization, we decided to group and analyze our data according to vaccination status. And then and to see whether vaccination increases neutralizing titers or not. And truly, we found that for all the variants that we tested, donors who were vaccinated had higher levels of neutralizing titers against all the variants. And we wanted to see, so these are neutralizing titers. What about neutralizing titer positivity or antibody neutralization positivity? These are the titers. But I want to know the percentage of people for vaccinated or non-vaccinated who would neutralize. And when we did the percentage positivity as well, yes, we found that vaccinated persons, you'd have about 80% of them expressing neutralizing antibodies, where just half of them would express if they were not vaccinated. And when we did a chi-square, we found that truly there's a positive association between vaccination and neutralizing antibody titer production. And when you look at the data, beta is still featuring high. Beta, a variant we did not have. So our question is that this beta so the beta positivity that is featuring high, was it localized to a particular wave or it's spread out in all waves? So we broke our data into waves, okay? Vaccinated and unvaccinated for the different waves. You find that unvaccinated persons would have them up to wave 4. And then for vaccination, all our donors were started from wave 3 because we had our vaccinations later, okay? And then still, you see that even when you break the data into waves, beta is still featuring as the highest for everything. So now this is where we are at, asking the question whether how beta is easily neutralized. Is it as strongly immunogenic? And then if it is, if it's used as a vaccine or if it's introduced to people, will it also produce broadly neutralization as would expect? But these are questions that are left to be answered. And so that is where I would leave you. And so my last data slide will further buttress the point of how vaccination may be contributing to broadly neutralizing antibody production. And this was work done by Cassie, Peter's doctoral student, who it would have been nice for her to present more of this data, but unfortunately she couldn't make it here. So here, she took cells from some of our donors and then ran single-cell sequencing, single-cell B-cell sequencing to look at the clones and other receptors. So when she analyzed the B-cell clones or data, she found that people who were vaccinated had increased unique B-cell clones. When you have increased unique B-cell clones, it means that you have more B-cell expansion, meaning you are likely to produce antibodies that are broadly acting. But then when we looked at the data between symptomatic and asymptomatic, there was no difference. So it's really pointing towards vaccination status more. When we broke the data into waves, you see there's a steady increase in the B-cell clones with waves. And of course, the waves also indicate, so as the waves were going on, we were having more people vaccinated. So this data is still pointing towards vaccination for B-cell clones. And so at this point, I believe that I have shown you data that indicates that persons who are vaccinated are more likely to express neutralizing antibodies. And then the vaccines that we had, although they were from the ancestral strain, are still able to protect us against even the newer variants that emerged, and that vaccination could also be contributing to broadly neutralization. Now, our end game for this project is to isolate monoclonal antibodies that are broadly neutralizing, and the point is to have something for therapeutics if necessary, but then also, more importantly, for diagnostics and research. So on this note, I'd like to say thank you to these people who have contributed immensely to the work. My boss, Peter, for hiring me and trusting me with money to do research. And Cassie, with whom I started the project, Elichem, who helps a lot in in the assays, Becky and Nanama, who curate and process our samples, and then my colleagues, Crystal and Michael, for the good company, and then my study participants and sources of samples. So the COVID-19 WACCBIP team led by Prof. Gordon Wandari, and then the Ghana COVID-19 response team led by Prof. William Ampofo, and then all the hospitals from which the samples came from, and then our collaborators at Noguchi and then University Medical Center led by Professor George Che, and collaborators from University of Glasgow, where Kessie did the single-cell sequencing, led by Thomas Otto. We thank them all for their contribution. And I thank the University of Ghana and WACCBIP for providing the table for us to operate on, and of course, these funders who put money on the table for us to work. So on this note, I'd like to thank you and have questions if you have any.
Thank you, Irene, for the beautiful presentation. We'll now take 2 or 3 questions for Irene. Yes.
Hello, Irene. Beautiful presentation. And as you know, I'm a public health specialist, so I'll bring you back to the epidemiology. Your sample size was about 200 and something patients. And I was wondering how valid is this? Because I want to know what was the prevalence in Ghana, and can your findings actually be extended to the whole of the country? Can we actually say that this is externally validated?
For the sample size, thank you. So for the sample size that I'm presenting and to extrapolate for the whole country, no. Because even we didn't cover all the regions, although we took some of the samples from a lot of the regions. But for samples from Wave 2, actually, we're all from Cape Coast. So I can't extrapolate for the whole country. If we want to do that, we'll have to expand. But we have more samples we could test. Now, the goal for this main work was to identify patients with broadly neutralizing antibodies to produce monoclonal antibodies. So for us, even one patient with a successful isolation of monoclonal antibodies was a success for us. So it's not a public health study. It was more of looking for fishing for patients for neutralizing antibodies production here. Okay,
All right, thank you. Okay, sorry.
You're welcome.
You know, across the different types of vaccines.
Okay, so I'll repeat your question if for those who didn't hear. He wants to know if there were different vaccines in Ghana. So instead of me putting vaccinated and unvaccinated, if we could look at the specific types of vaccines. Yes, we had a lot of vaccines, about 5 of them. And so for this small sample size, if I had decided to break it, it would have been difficult to analyze. And so they were spread across the different vaccines that we had. But then Yao Bediakon yesterday presented on just AstraZeneca and Pfizer. So yeah, all right, thank you.
All right, thank you very much. I think, let's give her another round of applause. Thank you, Irene. So now we move to the fellow section. Is Emilia here? Is it online? She's presenting online. Okay, so we have an online presentation by Emilia, and she's going to look at molecular characterization of SARS-CoV-2 variants in patients with COVID-19 in different provinces in Burkina Faso. Emilia, if you can hear us, please load your presentation and start your talk. Can you hear us? All right, okay, so you have 15 minutes for, 12 minutes for presentation and 3 minutes of questions. So please, you can go ahead. Please, can you put your slides in the presentation mode? No. No, not yet. Okay, all right. Yes.
QIAGEN. Areas. From our results, we note that Omicron variant was found in about 45%, followed by the Delta variant at 35%. Only 3% of recombinant was found. These are classified as a probable co-infection of Delta Omicron variants. In previous study carried out in Burkina Faso and Malawi, the Beta variant was the most common at a given time during the pandemic. In another study in Western Burkina Faso, Alpha, Eta, Delta, and Omicron variant were present. In this study, the most common clades are 20A and 20B for patients from whom samples were collected between 2020 and 2021. After this, clade 20G and 21G and 21K were the most common. The figure four shows that Omicron and Delta variants were more present in suspected cases. In a study in Singapore, symptomatic cases were more infected with Alpha, Beta, and Delta variants. However, travelers were more likely infected by Delta variants. Taking account the 9 waves of the pandemic in Europe and by superposition, you note that Delta variant was predominant from wave 2 to wave 3, but since the event of Omicron variants, it's the ones that persist over time. Areas. The figure 6 shows that the Delta variant was predominant in rural area. In urban area, on the other hand, 2 variants were more prevalent, Delta and Omicron, and the most prevalent was the Omicron variant. To conclude, we can say that there were several variants of concern of SARS-CoV-2 virus during the COVID-19 pandemic in Burkina Faso. Omicron was the most common variant in subject presenting as suspected cases. Delta variant was the most common in rural in rural area and Omicron the most common in urban area. This study provides additional data on SARS-CoV-2 variants in the country. At the end of this presentation, I would like to thank all the people from whom the SARS-CoV-2 samples were collected and all the facilities from which these samples were collected. I would like also to thank my institution, WACCBIP, and all the rest I can mention for fear of forgetting someone. Thanks to the presentation and thanks to all for your kind attention. Thank you.
Okay, Peter's got a question.
Thanks, Emily. Nice presentation. I had a question about your urban-rural divide. Were these samples collected at the same time? I don't know if I missed that. Can you hear me?
Emily, can you hear me?
Yes. Oh, okay.
That's me.
Hello, Emily. Can you hear? Can you hear? If you can hear, say hi. Can whoever is manning the computer repeat the question? No? Can you type the question? So I'd like to know if the samples were collected at the same time for the urban and rural cohorts. And if not, in what sequence were they collected? Spelling for what? Sequence? Yeah, they should have.
She's not hearing actually.
I know.
Shall we continue with the program?
Yeah.
You can ask her your question when you see her again.
Okay.
Okay, so our next speaker is Francine. I don't know how to say his surname, Yuso, who's going to be speaking about the molecular and serological evidence of Crimea Congo hemorrhagic fever virus in livestock and ticks in Cameroon. Over to you, Francine.
So my name is Francine Kenyusu. I'm a postdoc at the Center for Research in Infectious Diseases in Yaoundé. So my presentation is entitled Molecular and Serological Evidence of Crimean Congo Hemorrhagic Fever.
Can you share your presentation, Francine? We can't see your slides.
Let me share it now because I'm trying to share it.
Or if you can't, can the...
Let me share it.
Okay.
Please, can you see my presentation?
Yes, we can. You can continue.
Okay. So I was saying that my name is Francine Kenyusu. I'm a postdoc at the Center for Research in Infectious Diseases. And my presentation is entitled Molecular and Serological Evidence of Crimean Congo hemorrhagic fever. Prevalence in livestock and ticks in Cameroon. This presentation will follow this outline. As an introduction, human Congo hemorrhagic fever is a tick-borne hemorrhagic zoonosis that is asymptomatic in domestic and affects.
We can't actually hear you. Can you, is there some background noise?
It seems that somebody...
Maybe you can just ask them to be quiet while you're presenting.
No, it's not here. So there is another person that is on mute.
Then IT team, can you mute other participants that are online, please?
Okay. So I was saying that human Congo hemorrhagic fever is a tick-borne viral hemorrhagic zoonosis that is asymptomatic in domestic ruminants and affects humans. In animals, it is mainly asymptomatic, as I've said, and it affects cattle, sheep, and camels and goats. And in humans, it is mainly linked to occupation, such as occupational disease, such as veterinarians, shepherd farmers, slaughterhouse workers, and also clinicians. The incubation periods vary from 2 to 13 days, and we have several phases of the disease. We have the pre-hemorrhagic phase, that is mainly mild, where we can find mild symptoms, and the hemorrhagic phase with a case fatality rate of up to 40%. And this, there is no vaccines and no treatment available for veterinarians and human use for this disease. And I want also to mention that human Congo hemorrhagic fever is one of the top priority diseases for research and development in an emergency context by the WHO. It is mainly due to a virus called human Congo hemorrhagic fever autonavirus. That is a three-segmented negative sense RNA virus made of the S segment, the medium, and the large segment. And the virus belongs to the autonavirus genus and the Nairoviridae family. This virus presents the greatest degree of sequence diversity of any arbovirus, and with divergence ranging from 20 to 31% amongst the S, L, and M segments of virus isolates. The virus is mainly transmitted by chicks of the genus Yaloma, and that chick serves as a zoonotic reservoir of the virus. The human Congo hemorrhagic fever remains largely unnoticed until human cases are diagnosed. The transmission is mainly through horizontal transmission from a nymph or a larvae of the chicks to birds and small mammals. And after this horizontal transmission, there is a vertical transmission from adult chicks to the offspring. There's no horizontal transmission when the chick is already adult, from adult chicks to big mammals and livestock. And there is also a transmission through direct contact with infected tissues of those animals, such as cattle, sheep, goats, and camels. And it has been also reported in nosocomial transmission in hospital settings. The disease is mainly endemic in Africa, in Western and Eastern Europe, the Middle East, and in Asia. In Cameroon, there are mainly seroepidemiological studies that have been conducted in animals, and this was a retrospective study where samples collected in 2013 in the southwest region of Cameroon were used to detect IgG and IgM of human Congo hemorrhagic fever, and they found a seroprevalence of 56% of the different antibodies. And in the human population, we have one study that was conducted in 1989, where they found 0.22% of IgG directed against human Congo hemorrhagic fever virus, and another one from retrospective study on samples collected by a team of Centre Pasteur of Cameroon in 2005 and 2012, and there they found a seroprevalence of 4.4% of IgG. In view of this limited data concerning this virus in the country, in humans, domestic animals, and chicks, and we wanted to assess the epidemiological study of the virus in the country, the recent epidemiological study of the virus in the country. Thus, to answer this preoccupation, we have as general objectives to determine the prevalence of human Congo hemorrhagic fever virus in domestic ruminants and identify its potential tick vector in Cameroon. Specifically, we wanted to describe the animal species according to their origins, determine the seroprevalence of anti-human Congo hemorrhagic fever virus specific antibodies, and determine the prevalence of human Congo hemorrhagic fever virus in chick species, characterize the virus circulating in Cameroon. To achieve these objectives, after obtaining the different authorizations from the different ministries, the Ministry of Livestock, Fisheries and Animal Industries, we conducted a cross-sectional and descriptive study in the central region of Cameroon, where we chose two different markets, a market of cattle and a market of sheep and goats, in 2019, 2020, and 2021. Blood samples were collected from cattle, sheep, and goats in EDTA tubes, and they were then processed in order to obtain the plasma, and the plasma was stored in -20 until further analysis. For the analysis properly, the plasma were used to detect specific antibodies directed against human Congo hemorrhagic fever using IgVet kit, and negative human Congo antibodies and positive samples were also used in order to confirm using a seroneutralization assay. These were simultaneously collected on the same animals in the different markets, and then they were then morphologically identified and preserved into a 1.5 milliliter tube containing RNA later into -80. During the analysis, chicks were pooled in 3 to 8 chicks per pool, and a fragment of the air segments of autoneuroviruses were amplified in order to detect the different autoneuroviruses that are present in the samples. Then the sequence were analyzed using Vigen and IQ3. As a result, overall, 756 animals were collected, 692 males and 64 females. We have an overall 441 cattle, 168 goats, and 147 sheep. 73% of the cattle were from Cameroon, as we can see on the figure on the right. There we can find that all those cattle were coming from the Adamawa, the north and the far north region of Cameroon. 22 samples were originated from Chad, and 4% of the samples were from Sudan. All sheep and goats were from the north region of Cameroon. We have seen that during this study that most of our samples were adult males, and this is due to the cost effectiveness and because females are mostly preserved for reproduction and also milk production. And similar observations have been made by other authors that were working on the marketplace. Concerning the distribution of human Congo hemorrhagic fever virus seroprevalence, we found 61% of seroprevalence in all animals, 98% in cattle, 15% in sheep, and 6% in goats. And the cattle seroprevalence in Cameroon were 98.15%, 100% in the far north, 97% in the north, and 97.6% in the Adamawa region of Cameroon. Concerning cattle originating from Chad, we have a seroprevalence of 97.96%, and from Sudan, a seroprevalence of 100%, that you can see on the figure on the right. And in view of this high seroprevalence of human Congo hemorrhagic fever antibodies, in a context where no epidemics of these viruses have been declared in the country, we asked ourselves if this was due to possible cross-reaction with other members of the same serogroup, such as Azar virus, as it has been mentioned before. Thus, we just proceed next by confirmation of these results using seroneutralization assay. Considering the seroneutralization results, we selected some samples, 10% of each positive and negative samples, and we sent for seroneutralization at the Centre International de Recherche en Infectiologie at Lyon, France, and there they found that most of the samples that we had were also positive using the ELISA analysis, the GA ELISA analysis, were also positive using seroneutralization, confirming the high seroprevalence that we observed using ELISA test. Interestingly, there was also 2 samples from cattle that were negative with GA ELISA that become positive using seroneutralization. That is stated as the gold standard technique for ELISA analysis. So these results confirm the circulation of human Congo hemorrhagic fever in cattle and small ruminants in Cameroon. After finishing with the plasma samples, we just go through the detection of the autoneuroviruses in chicks. Thus, we collected 3,757 chicks belonging to 11 species, and the description of those tick species and also the polymorphism analysis was also done on those species that we found, and paper was published in 2021, and it's available on PubMed. And on those 3,000 chicks, we used 1,500 in order to form 300 chick pool. And on the 300 chick pool, we got 2 positive pools. One positive pool were made of Yaloma truncatum, the species Yaloma truncatum. And we found that after sequencing that the positive pool were positive to Crimean Congo hemorrhagic fever virus. And the sequence is available also on GenBank. And we found the second pool were the pool of Ambioma variegatum. And this pool were positive to Jugbe, to the mildly pathogenic Jugbe virus. The sequence is also available on GenBank. So this shows the current activity of the virus in the country, and also it highlights the need in order to the need of more epidemiological study in order to detect the whole of Yaloma truncatum and also Ambioma variegatum in the transmission of these viruses in the country. For the phylogenetic analysis of Crimean Congo hemorrhagic fever, we found out the strain that we obtained were from the African tree genotype. And these have been also found in 2 Central African countries. And it has been stated that there is 2 different genotypes that are circulating in Central Africa, the African 2 and the African 3 genotypes. In view of the small portion of the L segment that were amplified and sequenced, this represents only a little genetic information of the virus. So for further study, we need to do the whole genome sequencing of this virus in order to fully characterize the virus in the country. In conclusion, we found a high seroprevalence of Crimean Congo hemorrhagic fever in cattle with various origins, Chad, Sudan, highlighting the necessity of the establishment of a transborder surveillance of zoonosis using the One Health approach. And the detection of the virus in chicks shows the active circulation of Crimean Congo hemorrhagic fever virus in the country and highlights the necessity of more epidemiological studies in the main cattle production society, considering domestic ruminants, at-risk human population, and also the vector of chicks. This study was also published in the Frontier in Cellular and Infectious Microbiology in 2023. And as perspectives, we want to continue with the whole genome sequencing of Crimean Congo hemorrhagic fever, metagenomic detection of tick-borne pathogens in the remaining chicks, as we have seen that there is more than 1 virus circulating.
All right. Francine, thank you very much for the presentation. We are open to 1 or 2 questions. Okay, there's a hand here, please. Can you hear me, Francine? Hello, can you hear me? Hello, can you hear me, please?
Yes, I can hear you.
Okay, so there's a question for you. All right, go ahead.
Bonsoir Francine, merci pour ta présentation. After your presentation, I was so scared, and I really have like 2 main questions. Since Ebola, since COVID, we are afraid, and people are wondering what will be the next pandemic, and we think it might be a zoonotic disease. So my question to you is this: when you come up with this Crimean Congo hemorrhagic fever virus, do you have any information about its lethality in humans? That's the first question. And then in your perspectives, you talked about following this disease, but I didn't see about informing any policy-making institution or influencing institution. My question is, have you thought about informing, and what are the things you've been doing to connect this information, this very important information, to these institutions in order for us to intensify surveillance of this virus? Thank you.
Okay, thank you for your 2 questions. For the first one, concerning the lethality in humans, as I've mentioned, there are studies that show that the lethality in humans vary from, it depends on the context first, and also on if the population is still naive or it's the population that have been exposed to the virus, but the lethality ranged from 5 to 40% in humans. For the second questions, have you thought about informing the different institutions? In our institution here in Cameroon, the Center for Research in Infectious Diseases, we have developed a group. We call it CIVICAC. It's a group where we found different institutions of the Ministry of Public Health, the Ministry of Livestock and Fisheries, and Fisheries and Animal Industry. So we found the representatives of all the ministries in that CIVICAC group. So each year, from our institution, there are presentations of the different findings that we have in that typical groups. So we keep informing them on the situation of everything that we found on the field.
All right, thank you. We'll allow a very short question for. Yeah.
Thank you. Francine, I wanted to ask, the ELISA that you did, what did you use as your negative control and then your positive control? That's one. And then also, was this negative control unique for both cattle, sheep, and or you use one negative control for all of them? The second question is the seroneutralization. Is it a live virus neutralization or a pseudotype neutralization? If it's pseudotype, what antigen were you using for your VLP? Thank you.
Okay, thank you for your two questions. For the first one, the ELISA that we were using was a kit made by IGVET, and they were providing the negative and positive control. And on the kit that we use, they said that it can be used for cattle, sheep, and goats. So we're using the same kit for the 3 species. And concerning the second question about the seroneutralization serotype, we did it not in Cameroon, it was at Lyon, and there they used one cattle that were experimentally infected with the Crimean Congo hemorrhagic fever, and they used as negative control a cattle that were not exposed. So the cattle were found in France. It was not here in Cameroon. So they used both, and it was a VLP technique that they use for seroneutralization assay.
All right. Thank you very much, Francis, for the good presentation and also for the responses.
Thank you.
So we'll move to the next speaker. I think Ms. J. Oh, Ms. J is here. All right. So Ms. J is going to talk about dissecting the molecular barriers to virus replication and disease in natural reservoir species and humans. Ms. J, you have the floor.
Hello, welcome everyone. It's been a long day, I know, also for me. My slides are relatively simple, but please, if you have any more questions than allowed afterwards, just come to me and we will figure out if I can answer them. It's my second time here in Ghana, second time here at WACCBIP. It's an amazingly organized conference, and really congratulations to all scientists who prepared the data and presented it. It's been an amazing day so far. I would like to present on a topic that's, I think, typically not very much studied here on the continent, which is sophisticated in vitro models that allow us to address biology otherwise not accessible. But I usually like to start my talks with sort of an unreferenced, out-of-context image, and I think this is a beautiful micrograph that was taken over 50 years ago from one of the one of the deadliest viruses known to humankind, which is Ebola virus. And the reason why I'm showing you this is because 10 years ago it was the largest recorded outbreak from Ebola virus seen on the world, with imported cases around the world, and we're still scared of these viruses and its cousins, the Marburg virus. And roughly the same time, we have seen the emergence of another deadly coronavirus. It's not SARS-CoV-2, that was just now, but MERS-CoV, which reported a case fatality rate substantially greater than for SARS-1 and SARS-CoV-2 coronavirus. And none of us was around that time, but I can assure you, H1N1 influenza A virus, and now the emerging maybe H5N1 highly pathogenic avian influenza virus is quite deadly. Although the case fatality rate is low, it's much more contagious and resulted in about 100 million deaths in 1918 and 1919. So in 1 year, 100 million of people around the world, one of the largest pandemics that has been recorded. So if I ask you this simple question, are these dangerous pathogens? I guess from a human healthcare perspective, we would say absolutely yes. But if you look into the animal reservoirs that naturally carry them, actually the answer is a bit more complicated, because you don't find these animals ever develop any form of sickness once they have been exposed, and they are pretty much exposed all the time. The reason why I'm telling you this is because about 75% or even more of emerging pathogens that can infect humans are found as zoonosis, meaning that they circulate in an asymptomatic fashion in reservoir species, and upon spillover into humans cause a varying degree of disease, ranging from asymptomatic disease to death. And I think it's been extremely difficult to predict what actually makes a virus pathogenic, and even within us humans, why are some people protected and others not? So that leads us to some of the central questions we'll try to address with our research, which is again, what determines pathogenicity? Is it something encoded simply in the virus? Is it something encoded within the host? Or is it maybe a combination of the two? And second, and I think this is really exciting, can we learn something from mammals that have naturally evolved to become disease resilient to some of these pathogens? So basically understanding how does one adapt to an emerging virus that is otherwise lethal. And our leading hypothesis is actually that a co-evolutionary adaptation process has shaped the genome both of the virus as well as the host to essentially make them disease tolerant, because that would make them fit. Whereas humans, in that case, we consider as a non-adapted host that has not lived with this virus for long. And in fact, for some viruses like filoviruses that have been around for at least 20 million years, that exceeds human existence by at least a factor of 2. So I think being non-adapted is part of the problem. But in reality, we're looking actually only at a subgroup of mammals, namely bats, because these are anciently diverged mammals that carry a lot of highly prolific viruses that can make us quite sick. And we try to understand what is the genetic basis, meaning not that we think there is a genetic basis, but precisely dissecting where does disease pathogenicity or resilience lie. Is it something, again, just found in the genome of the virus and or the host? So our research objectives are to develop in vitro models that would allow us to basically study infectious disease processes in a dish. Why is this important? If you think about human experimentation or animal experimentation, such as with bats, it's pretty much impossible to do them in parallel. So we have been developing in vitro models that will allow us to do cross-species comparisons under controlled laboratory condition. And we're currently focusing mostly on barrier tissue because we consider these sites as sort of the primary site of infection. So the site of your body where you first encounter the virus, and from it may spread to other parts of the body. And we are investigating these in vitro models by making them or infecting them with viruses that can cause a variety degree of sickness in humans, ranging from asymptomatic disease to high pathogenicity as seen with the filovirus Marburg. And ultimately, of course, we think that from comparison of, in that case, bats and human, and their response to virus, we can learn about adaptation strategies that maybe can exploit it for therapy and improving human health. The in vitro model I'm talking about is not the conventional 2D cell lines that you might have seen, but in fact it's a more sophisticated model that we call organoids. You can think of your tissue sort of as an ensemble of cells that all emerge from a stem cell. Not the stem cell that comes from an embryo, but actually some of our organs carry adult stem cells that can regenerate our organs, the liver, the lung, the gut, and we can isolate these adult stem cells and culture them in vitro to make miniature versions of organs that we call organoids. And this is just an example of organoids that can be kept in culture, they can be passaged just as cell lines, and they can recapitulate cell type diversity and function of human organs. And while I'm not talking about this today, we also have a collaboration with scientists here on campus to develop an affordable liver organoid system to study liver stages of Plasmodium infection, which I think is critical and currently only addressed with humanized mice, and that's simply not accessible for the majority of research labs. But today I'm focusing on organoids such as this small intestinal organoid that we developed from bat. We call them sometimes mini-guts, although this is not really a scientific term. And these organoids, they emerge from a tiny fragment that over the course of just 7 days develops into this sophisticated 3D structure. Well, this, I think, to me is already astonishing that something can form from a single cell or a small tissue fragment. We, of course, try to understand also whether these organoids have the cell type diversity and gene expression profile found in natural organs. And for this, we like to use single-cell transcriptomics, first because it allows us to compare data across species. Antibodies are not readily available for bat species, so we use single-cell transcriptomics to understand what are these organoids composed of. And we mostly use 10x genomics or droplet-based single-cell sequencing. I'm not sure whether you have ever heard about this, but in simple terms, you're essentially mixing your cells that are sitting in buffer with an oil emulsion, and when these two come together, they form droplets, where individual cells are contained within these droplets, co-mixed with beads, and these beads carry oligonucleotides that can hybridize to the mRNA that comes from a lysed cell within these droplets. And this happens to tens and hundreds of thousands of cells in parallel, which means that in a single tube, you basically have 20,000 individual RT reverse transcription reactions, that then when you combine them and sequence, you get sort of a huge table where you have columns representing one cell and rows representing different genes, and sort of a gene expression matrix that's quite large. And I'm not going to go into single-cell sequencing analysis, but I will show a plot that looks like this, where basically the degree of variability between cells is projected onto a two-dimensional space, a conventional XY spot, where similarity between transcriptomes of individual cells makes them cluster together. So cells that are basically pretty much the same type will stay close to each other and separate out from other cell types. So we've done this exercise with gut organoids. This schematic is a bit pixelated, but I hope you can still see it. Here's a cross-section of a small intestine, and below sort of just the cell types one would expect. At the bottom we have the crypt, where these adult stem cells lie, then differentiate into the villus, which is largely composed of enterocytes that are important for taking up food, but also the one cells that are actually infected by a majority of enteric viruses. So when we did single-cell RNA sequencing, we of course expected that enterocytes will make the largest proportion of cells, but we do detect stem cells and rare cell types that are important for example, innate immune processes. Here's just focused on the epithelium. Of course, the gut contains stromal cells, it contains immune cells, but for the sake of this talk, I will focus on the epithelium because our organoids are simply composed of epithelial cells. And I hope you can appreciate that the organoids resemble a large degree of the cell type diversity found in vivo. When organoids cultured under expanding conditions, they are largely composed of stem cells and to some degree of cells that are highly differentiated. But when we change the media to a differentiation condition, we can actually recapitulate cell type frequency and function, largely what is seen in vivo. So this is a model that, although in a dish is tiny, it's actually quite complex in itself. So not just a 2D cell line composed of one or maybe two cells. We've also done this for airway. So we have reconstructed the bat and human airway, focusing on the nose, the bronchus, and the alveolus. You can't unfortunately see it on the bottom. This should have been live imaging of differentiated airway organoids, but they polarize into a basal apical form with ciliated cells on the top. You may have heard about ciliated cells, but these are interesting cells with sort of beating cilia on the top that are important for transporting mucus out of your airway. And these can also be made in vitro and usually sit on the top, and this would be the top right. And by single-cell sequencing, without going too much into detail, I can assure you, we also have a large degree of cell types, even very complicated cell types, that we don't even know how they emerge in vivo present, which means that the organoid might also be a model to understand how individual cell types actually emerge, especially in otherwise inaccessible species, such as the bat that I'm going to talk about. Okay. Having established organoids, we would like to understand how, again, maybe bat organoids respond different to a virus that they host, such as Marburg virus, than human organoids. And for that, we used organoids and generated organoids from the Egyptian fruit bat because it's one of the best understood reservoir relationships. So the Egyptian fruit bat is the known true reservoir, and I think the only natural reservoir known to harbor Marburg virus. It's a cousin of the Ebola virus and is similarly deadly to humans. In the Egyptian fruit bat, it does not cause any disease, whether it's in the wild or under experimental conditions where these bats have been bred for decades, never been exposed to Marburg in the wild. That means they don't have an antibody response, yet if you give them Marburg virus, they still don't develop disease, which really argues for not an adaptive immune protection, but sort of maybe an innate immune protection on top of that. And it's not as large as the Ebola outbreaks that we have seen, but nevertheless, they have been deadly. And it's one of the viruses that's prioritized by the WHO because there could be an emergent emerging virus that belongs to the Marburg virus. It's not as contagious as Ebola, so human-to-human transmission is very poor, and that's largely the reason why the outbreaks have been relatively small. And this is also a virus that can be directly transmitted from a bat to human without going through an intermediate reservoir. So first we confirmed whether our organoids are naturally susceptible to Marburg virus, and for this we used the strain, Musoke strain, one of the reference strains, and we find that in the bat organoids it actually replicates in pretty much all of them, including the airway as well as the small intestine, and we compared it also to human airway organoids, simply for the reason because we believe that the respiratory route might be an initial site of replication. But I will maybe not go into too much detail, but I want to show you here is that once bats organoids are infected, they elicit an antiviral response measured on the transcription level, which is here shown on the XY plot, where upregulated genes are shown on the top, and the ones that are also highly expressed further segregate out to the right. And gene ontology enrichment analysis shows that enriched genes are largely found in the defense response genes. Surprisingly, human organoids do not show any form of upregulation, suggesting that despite being infected, despite shedding virus, they do not respond to an infection with an antiviral response, while they do to a non-pathogenic virus. So this is a clear example of the same organoid type responding to a different virus with a strong innate immune induction and not to Marburg. We also focused on type 3 interference because they have been strongly upregulated during the infection, and they are mostly relevant for epithelial tissue. And we just needed to confirm whether they're indeed protective, so we recombinantly made them. We stimulated organoids and then infected organoids with a GFP encoding vesicular stomatitis virus, and indeed, compared to type 1 interference, bat type 3 interference also protect the organoids from infection. So we also wanted to see whether when given together with the virus, they are able to protect bat organs from an infection, and they clearly do. This is infectivity level normalized to interferon untreated cells on a log scale, so I hope you can appreciate that the level of reduction, not pre-treatment, but given at the time point of infection, still is largely protective. So our model for Marburg virus, currently in organoids at least, would support a scenario whereby bat cells are protected by eliciting an antiviral response that leads to largely local restriction. Here on the right-hand side, a published data set of in vivo infection of the Egyptian fruit bat, where you basically see viral loads predominantly found at the site of inoculation. In primates, that does not seem to be the case. We know that from our in vitro studies, cells do not elicit an antiviral response. We know from in vivo studies that Marburg virus, even when given aerosolized form by inhalation, it spreads from there into the entire body. You see the heat map here. You find Marburg virus at the onset of hemorrhagic fever in these macaques in pretty much every tissue, including the brain. And you only find very late induction of interferon alpha, I think supporting our in vitro data. This is also true for SARS-CoV-2. I don't have time anymore to go through this, but we basically find the same thing. Bat cells, in that case primary lung cells, are able to allow the virus to enter, and they elicit an antiviral response that we don't see in human lung cells. At least we don't see it early in the infection point where we think control needs to happen for the virus to not propagate in amounts to spread all over the tissue. And we confirmed this by knocking out a key transcription factor that is required for signaling, interferon signaling downstream to the antiviral productive genes, which are called interferon stimulated genes. So in a control cell, we've hardly, if any, find after 3 days virus positivity. If we block pharmacologically interferon signaling, we see strong positivity suggesting no protection. The same is true when we knock out again this transcription factor, and it's exacerbated by dual combination where we see even cytopathic effect. That means the cells are dying from the infection, which we otherwise don't observe in bat cells at all, while we do see this in human cells. So I think some of the take-home messages, and then I'm done. I think organoids allow us to model complex biology in a dish across species for species that are also otherwise not accessible for research, such as humans, and in our case, the Egyptian fruit bat. These organoids are quite complex, and we show that they are susceptible, at least from the Egyptian fruit bat, to Marburg virus, which allows us to study this virus in more depth, also test therapeutics, of course. Again, we see an antiviral response in bat organoids, not in human, and we believe that type 3 interferons play a major role, especially in the mucosal epithelium. These have not been explored in Ebola virus disease, but I would not be surprised if type 3 interferons, interferon lambda, might have an important role as a therapy, because they are less pathogenic than type 1 interferons, interferon alpha, but they have not been explored in the context of primate disease. So we believe currently that maybe enhanced innate immunity with concurrent limitation of pro-inflammatory buffer protects a case in the Egyptian fruit bat, it from excessive inflammation and disease against Marburg virus. And our data on SARS-CoV-2 suggests that it may be a general strategy employed by the Egyptian fruit bat, and we believe also for other bat species. Our work is published in an early version on bioRxiv, currently doing revision experiments, so we hope to see it at some point in Cell, hopefully. Yeah, these are the acknowledgements. There are a ton of people who worked with this. We're doing our BSL-4 experiments with Marburg at the Public Health Agency of Sweden. We have a lot of in-house services, and I really thank the support from everyone, including you all for showing up to my talk and the whole WACCBIP staff for organizing this amazing conference. It's one of the best organized conferences that I've seen, and I've just come from a Keystone conference, and I think that says a lot. So amazingly, without charging any fee, I don't think you see this at all, at least from a European perspective. So amazingly organized conference. Thank you for showing up, and please any questions.
Thank you very much, Max. I think you have so much stuff to give, and you can take it on coffee break.
Yeah, absolutely.
Thanks. So I think because of the time, we'll allow one question. If there's a pressing question, just for Max. Yes, okay. Can we give it to? Hi. Yes, please. You can shout. Okay.
So for the organoid, I'd like to know how you infect. You go into the lumen, or you?
No.
Just use
Yeah. So.
The medium. Okay.
Exactly. So for the airway organoids, we reconstitute them on the air-liquid interface, as we call them, where we can expose the apical surface, just as in the airway, to the virus. So we know exactly if you add it on the top or the bottom, you can address this. So we hardly ever work with luminal organoids because it's difficult to assess the site that would normally be infected. Yeah.
Okay, thanks.
All right, so maybe we will take one last short one from.
Thank you. Yeah, that's really cool. Do I understand that you're doing this with wild bats?
No.
No, these. Okay.
Yeah, so our starting point was a bat breeding colony in Germany that have Egyptian fruit bat colony for over 20 years. We are collaborating with the Helmholtz Center now, who have been doing longitudinal studies in Côte d'Ivoire, where they can sample bat species, but there it's a bit of a concern about bringing maybe infected tissue into.
Yeah, that was going to be my question,
so
because they're known to be pretty riddled,
exactly.
I think
so
is
for
the
those
technical term.
We do thorough testing and of course establish them at least under BSL-3 conditions.
So, would wild bats, would they have, my understanding was that certainly in SARS, the wild bats, their interferon responses are always on because they're just under attack by so many viruses, or is that not quite right?
No, I think, I know it's been cited 7,000 times. It's the only paper that has shown this. The majority of people have actually disproven that interferons are actively on, and I think this is exactly the problem with wild bats. Because they may encounter virus infection in the wild, and at the time point of sampling, they may have an active infection which you measure by interferon activation. No sterile breeding colony has been able to recapitulate this.
Okay.
We don't see this in the organoids. We don't think it's a general trend. It might be true for the black flying fox, where this was demonstrated, because it has a contracted interferon locus, but it's certainly not the general feature in our understanding.
So you could do lots with co-infections, couldn't you? I bet that would be really interesting in your model.
We tried to step away from co-infections right now because of potential biosafety issues, and I don't think it's something really that happens in nature. If you look at Marburg virus and its close relationship with the Egyptian fruit bat, I think it's one species, one pathogen sort of situation, and I'm not sure how co-infections would help us. Yeah.
All right. Thank you so much, Max. Max. So we are just at the end of.
Just a really quick comment. We appreciate your beautiful talk. You may not have received the memo, but the conference fees go directly to me, okay?
That was great. Thanks, David. So we are almost there. So this is the last talk for this section of today. We are calling on Ellie Kim. She's going to talk about, on investigating. Ellie Kim is going to present on investigating potential cross-reactive immunity to SARS-CoV-2 from acute febrile illness causing pathogens. Ellie Kim, you have 12 minutes.
Hi, everyone. So my name is Ellie Kim, and I'm presenting my master's work, which was investigating potential cross-reactive immunity to SARS-CoV-2 from acute febrile illness causing pathogens. Yeah, so in the beginning of the pandemic, it was estimated that Africa would be one of the hardest hit places However, that was not what was seen, as we recorded about 1% of reported cases and about 1% of mortality. And this was attributed to a few severity modifiers, such as climate, vaccinations, genetics, age, comorbidities, and I am especially looking at exposure to certain pathogens. So acute febrile illnesses are endemic in Africa. They are a set of diseases that are characterized by the onset of a fever between 2 days to 14 days, and then the fever subsides. So these acute febrile illnesses usually present overlapping symptoms, similar to what was experienced during SARS-CoV-2 infection. And we know in immunology that when you are exposed to a new infection, rather than mounting a whole new immune response, your body engages already existing cross-reactive lymphocytes. This helps decrease the response time and then save your energy and resources. So the hypothesis of this study was that exposure to these acute febrile illnesses gives you some cross-protective immunity to SARS-CoV-2, at least in the Ghanaian population. So COVID was mild in Ghana, and acute febrile illnesses elicit similar responses. So we want to see if maybe this is a possible explanation as to why that trend of COVID was seen in Ghana. So this is the aim of the study. Specifically, I was going to predict potential cross-reactive immunity to SARS-CoV-2 from 26 acute febrile illnesses causing pathogens using in silico methods. And then secondly, to validate a protein microarray chip and detect antibodies specific for these 26 AFI causing pathogens. And then finally, to determine if there was an association between spike and nucleocapsid antibodies and how well you neutralize the virus. So here is my workflow. I worked with 2 sets of samples, pre-COVID samples from 30 healthy individuals and 65 AFI positive samples. I also worked with COVID samples, 52 cases, so people with active infections as at the time their samples were collected, and 40 people who were vaccinated. For the protein microarray chip, this work was begun by Dr. Gloria Megache, where she designed a protein microarray chip that has antigens from these organisms printed on it. And so these antigens are printed in spots, and then the spots are in blocks and are repeated 16 times. Basically, this chip was designed to help detect antibodies against these 26 acute febrile illness causing pathogens. So for the first objective of my work, one thing I failed to mention was that the peptides printed on the chip are known immunogenic peptides from these acute febrile illness causing pathogens. So I took those peptides from the chip and blasted their sequences against the SARS-CoV-2 proteome. And then for sequences that were greater than or equal to 70% sequence similarity from the SARS-CoV-2 proteome, I checked if there were possible B-cell epitopes. So here are the results I got. Out of 128 peptides that I looked at, 9 of them were had a greater than 70% sequence similarity. And then out of that 9, 5 of them were predicted to be possible B-cell epitopes, which was interesting. So I was excited to see what I would see in the experiments. So for the validating the protein microarray chip, I used samples that I knew were seropositive against some of the organisms that were printed on the chip. And then, so for that part of the work, the protein microarray chip comes with a polio control. So all of the samples that are used, we assume that the individuals are going to be vaccinated against polio. So that's a control in the chip. And so there we see over here that, yes, we see that there are hits from the vaccination because they have antibodies against the polio antigen. And this is a glycine chain, which is supposed to serve as a negative control. However, so it's supposed to be like a 0, but then we can see some negative readings. For my West Nile virus, if you see over here, this peptide here was was from the West Nile virus. And here, this sample is supposed to read positive, but it didn't. And similar was seen for the Plasmodium falciparum sample, and same for S. typhi. So in conclusion, I couldn't go on to detect the antibodies against the 26 acute febrile illnesses using this chip. So I went on to perform an ELISA to find out if these individuals had nucleocapsid antibodies and spike antibodies against SARS-CoV-2. And here, for those with active infections, there was only significant difference in their antibody production at the day 28 and day 56. And then for those who were vaccinated, their antibody titers did not change over time. So comparing the pre-COVID samples to COVID samples, we can see that healthy individuals I didn't record any positive, they weren't positive for nucleocapsid antibodies. But however, for those who were AFI positive, some of them were positive for nucleocapsid. This could be because they have been exposed to maybe a previously circulating coronavirus, since the nucleocapsid is a relatively conserved a relatively conserved protein in coronaviruses. And so I went ahead to find out if there was an association between your SARS-CoV-2 exposure and then your nucleocapsid antibody titers. And it was interesting to see that about 40% of the pre-COVID AFI positive samples showed nucleocapsid antibody, which could not necessarily be antibodies against the nucleocapsid, just that they may have produced antibodies that are cross-reactive to this nucleocapsid. And then also you see that the healthy individuals pre-COVID, 100% of them did not show any antibodies against the nucleocapsid. So I did the same thing for the spike antibodies, comparing pre-COVID samples. But here you can see that the pre-COVID samples showed significantly lower antibodies against the spike compared to the COVID samples. And then when I did the association, you can see that spike antibodies production is associated with your exposure to SARS-CoV-2, though about 20% of the pre-COVID healthy individuals did show antibodies against the spike. I went ahead to find out, sure, you have antibodies against the SARS-CoV-2, but do you have neutralizing antibodies, right? So using the same experiment as Irene described earlier. So here in people with active cases, their antibodies, their neutralizing titers didn't change except on day 28. And then for the vaccinated, over time, their antibody titers didn't change. And here we see that pre-COVID samples, so both healthy and AFI positives, show lower neutralizing titers. They were not able to neutralize the virus as well as people who have been exposed. So I went on to find out if there was an association between virus neutralization and exposure to the SARS-CoV-2 pathogen, which we see here, except that again we see some healthy pre-COVID samples that are able to neutralize the virus. Then I finally went on to find out if there was a correlation between how well you neutralize the virus, your spike and nucleocapsid antibody titers. And as expected, we see that there's a high correlation between spike antibody titers and then how well you neutralize the virus, which makes sense since neutralizing antibodies have to be against the spike, right? So in summary, 9 out of the 28 peptides that I looked at showed sequence similarity to proteins from SARS-CoV-2, and out of these 9, 5 of them were positive B-cell epitopes, which gave hope that if you're exposed to these AFI pathogens, then you could possibly produce antibodies that are cross-reactive. And then we see that where the pre-COVID samples, the AFI positive samples did show a significant antibody titer against the nucleocapsid, but then it wasn't neutralizing. And then finally, there was a positive correlation between spike antibody titers and how well you neutralize the virus. For the recommendation of this work, I think that the problem with the chip was that there could be an optimization of the number of peptides printed for each antigen. And then finally, I would like to investigate the potential function of these antibodies that we're seeing in the AFI positive samples. I would like to acknowledge my supervisors, Dr. Bediako, Dr. Peter Kwashi, Dr. Gloria Megache, and everyone on this slide. I'm grateful to WACCBIP and all of the funders for this opportunity. Thank you.
Okay, we can take a question from Chinadu over there.
Thank you for the good presentation. This sounds like a eureka moment for me, because I spent my entire time in Dakar arguing this case with Peter, and it's good to see the same data that we both agree for once. So thank you. I have two questions. One is on what strain of the SARS-CoV-2 were you neutralizing?
Wild type.
Oh, the wild type. Then the second question, I like your chip. I wish it was working. So you used a non-positive West Nile sample. This is a serum.
Yes, the serum.
So what do you think why that positive sample is not picking up the peptides?
Okay, so generally in the design of the chip, it is the same amount of antigen that is printed for each peptide, right? But then, even in ELISAs, you still have to optimize the amount of antigen that you put there. So I think that's the problem.
Did you run it individually before multiplexing it?
No.
Yeah, I think probably that's...
Yeah. I'll ask a follow-up on that. So do you know that those peptides that you selected for each of those are antigenic?
Yes. So they were derived from literature.
Okay.
So peptides that people were already working with, yeah.
Okay. Any other questions? There's one more question here.
I have one question about peptides that we find across other pathogens that resemble the SARS-CoV-2 peptidome, as you can call it. So that would maybe also imply the converse is true. Once you have a SARS-CoV-2 infection, you might be protected against some of these, which for us Europeans, maybe in the case of Plasmodium, might be relevant.
Yeah.
So have you considered this too? Whether serum from Europeans might be protective against some of these pathogens?
I mean, that's something to look at, right? Yeah. So I could look at it. Thank you.
Thank you.
Okay, I don't think there's any more questions. Thank you very much. And
Thanks, Sam and Janelle. Today's been a long day. And as a reward for everyone staying, we have lodger for the guests. So I think the car will be, as last time, will be ready to take people to lodger and then to the hotels thereafter. But Thanks. I think today has been a great day of talks. It's been long, but I believe it's been worth it. So I'm not going to talk too much. Tomorrow we'll have a very interesting day. I mean, ordinarily we would be done by now. But because we're doing 5 days, tomorrow is going to be very interesting. We'll have, again, 2 great keynotes. One who from Mrs. Elizabeth Beatty, Irene Beatty, who actually graduated from the department and is now, has gone on to do great things and is now head of Patholab. And then in the afternoon, we'll have Dr. Tepo Tekwa, who will kick off the biotech entrepreneurship section, and then we'll have a pitch competition. So it'll be something different. It's the first time we're doing it. We'll see how that goes. But have a good evening, everyone. Bye.